In the video conference, call some.
No, thanks. No? No? Hello, okay ಆಗಿದೆ ಎಲ್ಲ ಸೆಟ್ ಆಗಿದೆ ಎಲ್ಲ ಸೆಟ್ ಆಗಿದೆ ಹಾಂ ಸರಿ ಹಾಂ ಹಾಂ ಎಲ್ಲ ಎಲ್ಲ ಸೆಟ್ ಆಗಿದೆ ಹಾಂ ಮಾಡ್ತೀನಿ ಸರಿ ಎಲ್ಲಿ ಸಾಕು
ಏನಿಲ್ಲ ಮೇನ್ ಇಂಟರ್ನೆಟ್ ಪಾತ್ರ ಮಾಡ್ತಾ ಇದೀವಿ ನಾವು
ಓಕೆ ಸರಿ ಸರಿ ಅದು ಓಕೆ
2 Mbps ಇದೆ ಸ್ಪೀಡ್ ಇದೆ
ನಮಗೆ ಡೌನ್ ಆದರೆ ಆಗ್ತಿದೆ ಸಾಕು
ಇಲ್ಲ
ಆಗಲ್ಲ ಆ್ಯಕ್ಚುಲಿ ಇದು
ನಾವು ಫೋನ್ ಮಾಡಿ
ವೈಫೈ ಇದ್ರೆ ಡೌನ್ ಆಗುತ್ತೆ ಸೊ ಇದು ಡೌನ್ ಆಗುತ್ತೆ ಡೌನ್ ಆಗಲ್ಲ
ಇದು ಡೌನ್ ಆಗಲ್ಲ ಮ್ಯಾನ್ಯಲಿ ಡೌನ್ ಅಂತಂದ್ರೆ ಔಟ್ ಸೈಡ್ ಏನಾದ್ರು ಇಶ್ಯೂ ಆಗುತ್ತೆ ನಮ್ಮ ಕ್ಯಾಂಪಸ್
ನಮಗೆ ಆ್ಯಕ್ಚುಲ್ ಆಗಿ ಬೇಕಾಗಿರೋದು 100 Mbps
450 Mbps ಮಾಡುತ್ತೆ 2 days ಆ ಕಡೆಯಿಂದನೇ all successful
admit it
doubts can overall presentation Bangalore அது ஆன்லைன்ல நான் ஒரு PhD ஒரு presentation ஆரம்பிச்சிருக்காங்க காலேஜ்ல தான் கண்டக்ட் பண்றாங்க sound level down. Why? There is no sound in this one. On the mid on the mid down the
Hello? Hello? Good.
Hi, good morning all.
Very good morning, sir.
Not 12.9. 32. Yes, sir.
नाही करायचं आहे पण आता नाही जमत जाणं. हम्म. मैंने नाश्ता ही नहीं किया.
Hello.
Virtually connected. Okay, it's connected. The live is going on.
Jai Shri Gurudev. I am Rakshit and this is my co-host Trisha.
On this wonderful day, we welcome you all to the International Conference on Innovation and Advances in Pharmaceutical Sciences, organized by Sri Adichunchanagiri College of Pharmacy, together with Association of Pharmaceutical Research, APR.
To kickstart today's day, we have an energetic speaker, Dr. Roshan Palewar, MS, MD, PhD, CEO and former global leader, Doctors Global Solutions, Healthcare Tech India.
We kindly request you, sir, to conduct the session on revolutionizing healthcare, the power of artificial intelligence in the pharmaceutical industry.
Yeah, thank you so much. And it's indeed a great pleasure to be a part of today's session in this wonderful international conference. Very fine. Good morning to all of you. I hope that this day is starting with absolutely amazing, positive energies, and throughout the day we're going to experience that with the various, key sessions. I must congratulate, first of all, the BioLeagues for having this mega event, and on top of that, the active support and coordinations and the entire organization organized by Shri Adichunchanagiri College of Pharmacy and the University. And indeed, it's a great pleasure to present my thoughts, experiences directly from the industry. And we'll talk about much detail how the AI is playing the critical role in the entire segment, right? So I hope I'm audible to all of you.
Yes, sir, you're audible.
Okay, great, thank you. So, friends, let's get started. I'm going to share the insights on the AI. And here is like now I wanted to present the slides to you. Let me share the screen. Right. Yeah, I hope the screen is visible. Yes, I can see that as well. Okay, very fine. Good morning once again. And yes, this is Dr. Roshan Palewal coming from the hardcore pharmaceutical industry, having more than 17 years of rich experience in the sector. And so far I have developed more than 400 medicines, that you possibly studying or developing at your front, but across the world, almost 172 countries that I operated, developed more than 400 medicines. And on top of that, currently leading a team of more than 650 global scientists. So today, with the pleasure, I'm going to touch upon the revolutionizing the healthcare, and more on top of that, how the power of artificial intelligence in the entire pharma sector is booming. What are the scopes? How the AI is going to transform altogether the traditional way of doing the business versus the modern way of doing the business in terms of the discovery, development, bringing the new formulations in the market, so forth and so on. So let me take you. Yeah, so when we talk about the pharma industry altogether, there are the few aspects that we need to understand. It's just not about the medicines, The pharma has three major aspects. One is the science behind the any product or any disease. Second is the technology which uses those, and then ultimately a lot of data get generated. So there is a smart areas of the intersection. And here, when the science comes into the picture, of course, we talk about the diseases, we do a lot of research for the medicines, drugs, and obviously the diagnosis part, which is the core of the science, right? And in a way to utilize this current the science, we take a lot of help of the tools, technologies, which includes very modern high-end technologies like artificial intelligence, machine learning, NLP, big data analytics. So that's the second side of it. And third important, while doing both the aspects, a lot of real world data get generated throughout the process from early phase of development till its commercializations. And when it gets structured out, so it carries a heavy information, and that has a tremendous opportunity as well as the importance when it comes to the data mining or the big data itself, right? So that's the intersection we are into. So what I would like to highlight from this point is that we need to have the balance of the science, the traditional methodologies, the traditional approaches versus the advanced technologies, which is going to shape altogether the new world in a very smart and sophisticated way. And that is how the altogether timeline of the product development, whether it's the generic formulations or whether it's a discovery, the timeline has been reduced because we are today talking and using, not only talking, but we are using the technologies in the real world in the pharmaceutical industry, and therefore it carries the humongous importance. Having said that, since we are going to talk much depth on the pharma, AI and everything, but broadly, we must also understand what is that the pharma industry all about, where we land today, and what are the key important healthcare ecosystem. If you understand this aspect, this is going to take you to a different level when it comes to you're working from day one itself. So when we talk about the healthcare, there are the 8 key principles, the P's, what we say is a P's. It's all centric, P for the patients, of course. Whatever we are doing, the development is ultimately for the patient itself. For the providers, the payers, there are a lot of stakeholders get involved into it. There's a policymakers, the patient advocacy groups, there is a pharma now, and then obviously the pharmacy chains, right? And ultimately the packaging companies. So in the world of pharma, the P has a lot of importance because we touch upon every single stakeholders who are the core part of the healthcare ecosystems. So if you understand this aspect, then whatever the product that you're about to do the development, bringing the new molecule in the market, you're going to do it in a much better way because you have the understanding and the the responsibility of taking care of the stakeholders who are part of the healthcare ecosystem. Now, having said that, now this is important to understand altogether the the global pharma industry, and I welcome all of you here. So the global pharma industry, all of together, if you look into, it's all about 1.5 trillion US dollar as of now. And as you are seeing in this particular graph, when it was in 2001, it was just $390 billion. And by 2024 to 2014, it reaches to 1 trillion USD. And already by 2020, in the past, $1.25 trillion has been crossed, and today we are standing at $1.5 trillion. What this graph indicates is that industry is growing rapidly, steadily, and there is a promising future in the coming days. And therefore, it's very important role that we are going to play as part of the pharma industry, whether I'm talking all together in a nutshell, whether it's the industry or the academia or the education sector or even as a hospital segment. Every one of us are playing a critical role to contribute in the overall well-being of the healthy human beings across the world. And therefore, as we know how the graph is growing steadily for higher run, we have absolutely the secured the future. So especially the students of B Pharmacy, M Pharmacy and the PhD, nothing to worry about your future. Your future is secured because the industry is growing rapidly, and this is a testimony of the real world how the industry is performing, right? So this is on the global perspective. But how can I miss about the Indian pharma? I certainly need to discuss and highlight about that. And interesting fact is that the Indian pharma industry is all about $55 billion as of now. Okay, when you look into 2005, it was around 6 billion. By 2015, 30 billion, and now the $55 billion. That means the Indian pharma industry is growing rapidly, having a consistent growth with the CAGR of around 15.92%. And this is another testimony. The Indian pharma industry carries a due importance the entire the world of pharma. By the way, India is known as the pharmacy of the world, and whereas India is already ranking a top 3 in terms of the pharmaceutical market, and it's incremental growth by the incremental growth, right? Whereas the 6th largest market globally in absolute size. That's the potential of the Indian pharma industry, and this particular point will attract all of your attention is that the entire world is looking at the India. Why it is so? because the India has that ecosystem, the atmosphere, the culture, the talent, the skill is ample enough. We have the world-class infrastructure in place, and most importantly, the entire cost of manufacturing or the development of the pharmaceutical products in India is absolutely economical, and that's the reason now India becomes the pharmacy of the world. Here, you should pay attention that how you are going to contribute when it comes to the pharmaceutical industry as well, right? Having said that, when it comes to the the segmentation, basically the entire Indian pharma has segmented into three areas, and definitely after this, I'm going to talk much on the technology side of it. The major portion is almost like 70%, which is of the generic formulation, generic drugs, right? Whereas the 21% is the OTC formulations, right? And most importantly, the remaining 9% is of the patented drugs. Now, if you see this particular, the segments, you will come to know which particular the pharmaceutical segment is, doing the excellency or doing the great in terms of the development. And why it is so, if you look into the 70% generic formulation, why it is because India is exporting to more than 200 countries, the generic formulation. And if I give example of, for example, the US market, India is supplying almost 60% of the total demand of the US market. And that's why we have many generic manufacturing facilities, R&D facilities in India. And of course, there are the, to cater the Indian market, we have the 21% of the OTC formulations that you already, where you can simply get the medicines over the counter, right? So this is the important aspect, to understand when, and this is going to take you how and where you're going to nurture your career in the coming days, right? I think all of you are with me this still point, right? So I was talking and highlighted about the manufacturing of the pharma products as well as the export. I just mentioned like the third largest in the world in terms of the total volume, and it's really proud moment for all of us. Why it is so? Because India is already at the $22 billion economy in the export segment, catering to more than 200 countries, and becomes accountable for 10% of the world's total demand. And that's everything coming from the Indian manufacturing facilities. And every single dose of the vaccine administered across the world is from the Indian manufacturing facility. And that is how we get the pride, we get the honor, we get the motivation that we are fortunate enough to be part of the pharma industry. And I congratulate all of you that you are going to be part of this industry in the coming days, especially to the students, and you should highlight that how you are going to contribute in the coming days as well. So on that perspective, now let's get into the core of the cycle. I'm just going to touch upon, I'm not going to dwell into the more of the technicality of the discoveries or the development, but on broader perspective, I'm letting you know that in the life of the drug discovery, which is almost a 20 years life, 14 years goes for the hardcore, discovery, which includes the pre-discovery, right from almost 10,000 more compounds, which is get screened, comes hardly to 50 for the preclinical stage. In the clinical trials, which has a different phases, phase 1, phase 2, phase 3, right, which is very lengthy period of 6 to 7 years. And ultimately, out of 10,000 compound, hardly one single compound, which is now turned as a pharmaceutical molecule, which has the pharmacological actions that get approved in the US FDA, and hardly the molecule or the product gets approx 2 years to do the commercialization in the market, So almost that 16 years is the hardcore discovery. So in this entire phase of development of the new molecule or the discovery, a lot of technologies are being used in every single steps. And now that is how the legacy of the data that we do have, the legacy of the scientific tools, techniques, and the technology, we are able to get the product in a very potential time, early time, I can say. And that is the importance of the technology. But yes, when you talk about the generic formulation, especially the drug development, and especially it's the cycle. It's the cycle of the product, because the product that comes to the market, before that, as I mentioned about the discovery, the early discovery to launch, which is the development phase again. And when it reaches to the market, which is we call as a launch of the market, from launch, there is a certain time it reaches to the maturity, and from maturity it goes down, which is we call as a loss of market exclusivity. And from this particular point, the generic formulations or generic medicines starts coming into the market, because this is the phase where the generic companies get the opportunity to do a development just a lookalike of the innovated product, okay, and thereby the generic market is get open for the industry as well, right? In very nutshell, when it comes to the development of the generic formulation, there are a lot of processes that get involved, start from the research, which is drug selection, pre-formulation, development, and then the development phase has the further optimizations. We do the pilot bio studies, followed by what we call the technology transfer to the pilot plants, and we perform the exhibit batches or the registration batches or the pilot batches, and then we generate the stability for almost 6 months from the 3 different batches. And this is that the pivotal bio studies get performed, and from this entire process, all the data which got generated, almost this time takes almost 12 months to 18 months, depending on the complexity of the molecule. But more or less 1.5 years to 2 years is more than enough to do the generic formulations. And we compile all those data, make a doses, and file to the regulatory authority. And ultimately, after approval, the product goes to the commercialization phase, manufacturing, and ultimately to the market. So even in the generics development processes, there are advanced technologies have been used. Now here I would like to request your attention. When I talk about the technology, there are 2 major categories of the technology which I'm highlighting here. One is the scientific technologies which has been used in the development process. That's one set of the technologies, right? The other aspect, now here the technology, for example, machine learning, artificial intelligence, big data, mining, the software, the tools, these are the IT-based technologies which is helping us to do the development with the minimal efforts and to avoid any sorts of risk involved during the processes, right? So this is the importance of the IT-based technologies, which now in the coming days is massively going to be used, and this is going to transform the whole segment of the traditional way of doing the development versus the modern way of doing the development. And since we are in the 21st century, we must definitely use these technologies in our day-to-day life, and especially in the pharmaceutical sector, because the world is changing, because the future has completely different way, and we are driven with the technologies, and therefore smartly we need to utilize the technology and keep a balance in the entire healthy ecosystems. So I think so far all of you are with me, right? It's very well understood. Now the interesting fact I would like to share, because we are in the world of 21st century, right? And all of us are using the technology in day-to-day life. So let me tell you, based upon the recent data, 60 seconds, that means hardly 1 minute. What happens in the entire world in the technology? 60 seconds, for example, Facebook's more than 1.4 million scrolling happens. In WhatsApp, more than 21 million texts get sent. YouTube, 500 hours content get uploaded every single minute, right? Apps get downloaded, more than 4 lakhs. Look at these numbers. from the Insta to Twitter to Tinder, the email, even the TikTok, Google, and WhatsApp, so many other stuff, including the Netflix, LinkedIn, and everything across the world of the digitalization. 60 seconds is transforming our life, and therefore, yeah, therefore, pardon me if somebody is
like this.
Mr. Prasun Singh, do you have something to ask?
Like that, I think. While I'm grasping, is actually about
Hello.
the reason is
Okay,
sorry.
think there is a background discussion is going on perfectly all right. I will skip that. Okay, so 60 seconds is something we are living in day-to-day life, and therefore we need to watch what and how we are doing it, because we are changing the phenomena in the world of technology. And when it comes to the obviously the Indian segment, and here I would like to highlight about the healthcare market. The entire, here is a pure IT-based technology is playing a critical role in the ecosystem of the healthcare, because the global healthcare IT industry or the IT market, I'm talking about the healthcare IT, it's not just the IT, but healthcare IT exclusively for the healthcare segment is as of now going to be $390 billion. By 2024, we already crossed almost 190 billion, and in 2024 is going to be $390 billion. Okay, and this is going with the speed of 15.8 CAGR, and this percentile of CAGR itself is a very promising for all of us, that how the healthcare technology is grabbing the entire global market, the global ecosystems of the healthcare industry. And when it comes to the Indian healthcare market, we already at growing speed of almost 27.41 the CAGR, and from $116 billion is going to be what do you call the $485 billion. That is how the Indian healthcare IT industry is also going to be revolved completely. And if I talk about 2023, you might have already witnessed advanced artificial intelligence is already plugged into the systems, right? And just a quote, like you might have heard about the ChatGPT, and this GPT-based process technology in the AI is shaping. Google is coming with the Brad GPT, its own in-house language programming system for the AI. And when I say AI, the artificial intelligence, it's the intelligence which has been using the existing information data, the psych and the pattern. The machine start learning the entire processes and builds the program in a fraction of seconds and gives the results the way human mind is behaving, or the human mind start working in the similar fashion. The AI is also working in a real life, in the real world. And therefore, no matter what we used to do in terms of the development of the medicines and as well as various processes, AI is going to be instrumental to give the precise, accurate and legitimate information about the rest of the process, and thereby we are going to transform the complete system in the coming days, right? And that's what is very interesting. Now, precisely as AI is a core of our discussion today, if you look into the altogether the market of the AI, by 2025, it's going to be $126 billion. Now, this is the, again, amalgamation or the combination that the technology is going to be used. For example, AI, machine learning. Again, these are the human intelligence being used in the real world of the drug and the pharmaceutical development, right? And so far, if you look into the global AI software market, why I'm highlighting this, because this is the complete new era or the new segment of the industry, where the industry is transforming themselves The global AI market altogether, $10 billion already, and going to be $126 billion in 2025. So these are the actually advanced algorithms which machines take cares, which the software takes cares, and gives the powered tools to streamline the entire drug discovery processes or the drug development processes. This actually uses the intelligence tools which has been designated or identified for the certain pattern or the certain large data sets which is got generated, and thereby giving the advanced output. Now this is actually solving today's lot of complex challenges, and there is a complicated biological network associated with it. So it solves those challenges as the way the human minds certainly do it, the intelligence do it. But again, it's beyond that. Why it is so? Because it is giving the output in a fraction of microseconds, not even the seconds, fraction of microseconds, right? And that is how the importance of the AI is in today's world, right? Having said that, now let's come to the core aspect of how the AI across the pharma value chain is being used when it comes to the drug discovery, high throughput screening, where the AI has been plugged, where the screening of, if you look into I talk about the 10,000 compounds, which is called the screening of the compounds to bring the very potential molecule having the pharmacological effect. So it helps to identify those rather than now studying all 10,000 molecule compound. We have the upfront feasibility that okay, this out of this hardly 5 are giving the potential outcome. So AI is instrumental in that way. The drug repositioning, how the drug should behave. So that also the AI is instrumental. Indication expansion, like for example, just to quote paracetamol, like antibiotic drug. But whether do you have the further, the associated responses for the other biological effect to give the pharmacological action? So the expansion of the indication, that is how the AI is going to also correlate the information data and the data minings, and is instrumental, by the way. Okay. Also the polypharmacology. So now look how you used to spend the massive efforts earlier in the drug discovery. With the help of the AI is going to be even smarter now. On the other side, when it comes to drug development, it's been used in the clinical trials for the designing and the optimizations, right? Further, the combination therapies to treat the various diseases, right? For the patient satisfaction or the stratification, this AI is also instrumental, and the biomarker identifications. On the other aspect, if you look into the AI in the healthcare segment, it's used for maintaining the electronic health record, which is called also as the EHR or the EMR. Okay, maintaining the entire patient data, personalizing the medicines being used for the certain specific diseases, treatment to the patient. There is also been used, and on top of that, the patient medications, right? Now, there are the other uses or the benefits of the AI is currently being used and going to be further expand on the novel technologies, where gene editing, CRI, SPR, the package innovations, the patient compliance, even from the marketing perspective, the sales force effectiveness. Here, the AI is going to be instrumental. And what we are seeing or the discussing as of now, these are already been implemented, right? And going forward in 2023 onward, AI is going day by day much more powerful, building the intelligence. Now it has a certain algorithms which analyzes the existing data, processes, technologies, information, algorithms, and the data sets. And the most important, why I say AI is a powerful, because it has the capability as well as the capacity to generate its all new algorithm basis the existing one and behave the way human minds going to be, and therefore AI is the core part of the ecosystem now. And for example, those students who are doing their research during the pharmacy or during the PhD scholarly research, I would insist you, or I would request all of you to pay your attention to club your research with the AI or with the machine learning or the blockchain technologies, because this is the future. And if you have your research along with this amalgamation of these technologies, then definitely those research carries a due value in the industry, which carries a lot of commercial importance at the same time, so you can explore that possibility for you. Yeah. Okay. Across the pharma value chain, as you saw, but if we wanted to put the numbers, how this drug discovery, the drug development, AI has been used and how is it transforming? To use this technology, obviously heavy investment comes from the industry, almost $200 million to almost $2 billion for a drug to get through the complete process of clinical trial and get FDA approval, whereas approximately 14% of the drugs are successful in passing the clinical trials. So these are the two main reasons why the pharma industries are increasingly adopting the AI, artificial intelligence, machine learning to improve the success rates of the new formulation, new drugs, and create the affordable medicines and therapies for the humankind. And since spending $2 billion for the drug discovery, and at the end, the risk factors are very high. To optimize all the processes, AI is instrumental because it's going to give the outcome in a very shorter period of time with the reliability higher. This is how even the cost of manufacturing is going to be reduced in the coming days. Reason is very simple. As of now, we are spending heavily on the development, heavily on the discovery. So R&D cost of the product itself is a high. And now bringing those R&D costs into the commercial side of it, obviously the product which is in the market comes costly, right? Now, whereas understand your 60 to 70% of the total cost gets saved because of these technologies, and then definitely there is a larger window to play, and the product which going to come in the market is going to be affordable, economical, and is going to be patient-centric, which is going to balance the pocket of every single patient. And that is the importance of the AI. Now, in nutshell, how we are currently the data science and the biological science are using this real AI. Data from the previous research, which I already mentioned, but this slides gives a precise, the aspect, right? The real world data, okay? The computational models, machine learning, deep learning, AI tools, this is called the data science. Now the pharma is no more only a pharma, is actually the world of science, having the combination of data science as well as the biological science. So from the biological perspective, the pathways, disease pathophysiology, the scientific correlations, insight, experimentation. So all these, heavy information, heavy data get scanned or get the new algorithms get built up. This is this all data, and we get these smarter tools, which is currently core part of the industry now. And apart from that, when it comes to the emerging trends in the industry research, apart from this AI, what else we have which is going to transform the industry coming forward? Artificial intelligence is one of those. Then blockchain technology. This has been massively been used in the current date when it comes to manufacturing, productions. Not just that, but blockchain is like, where we have this certain sequence of processes which is got repeated, whether it's a research, whether it's a manufacturing, or whether even it's an administrative jobs. There are certain processes are repetitive, and this technology uses those algorithms and builds the new way, and we get the very smart outcome without spending much in that way. So blockchain is another boom in the industry now. Mobile technology, as most of you have seen or might be observing, that everything what we are seeing is available on a fingertip, whether is the delivery of the medicines from the pharmacy, whether it's the delivery of the drug, which is called the targeted drug delivery systems, using even the mobile sensor-based technologies. These are there. IoT, Internet of Things, which are currently we are using it, and automation. Automation is actually a very close friend in the manufacturing segment, and now rather than only sticking to the commercial side of it in terms of the manufacturing, productions and supplies, now the automation is being used in every single day-to-day operations, whether administrative, management, research, discovery, or commercialization, or even the marketing. The automation is by default there, and this is how, from the sense of human touch, we are getting into the completely technology touch, and this is where we are standing in between, and we are experiencing this new phenomena, new technologies. This is actually the new future. This is actually the new 21st century. This is actually the future is going to be like that. We are actually keeping a balance of both our existence as a human being and the existence of the technology, keeping the balance and giving the best. When it comes to advantages, there are many, but at the same time, every single technology, it has own limitations or only or you can say drawback. But yes, we have to weigh both the aspects, and we have to bring the best out of it in the best interest of the entire world. Therefore, these are the emerging trends, and we should definitely keep a watch on that. And of course, the big data is part of that. Okay, there are a lot of other technologies or visualization, interactive reporting system is also there. I'm not going to touch in much of depth, but just understand it's all depend upon the kind of algorithms is being set. Everything is possible in today's world. These are the reality where the big data, AI, machine learnings, algorithms are playing a critical role, including the blockchain technologies. And therefore, this is very instrumental, helpful in today's world of science. And the modern way of doing the development is already there. We are very much available on that. These are few technologies already been used. For example, in the big data streaming solutions, there is a Hadoop technology, open source platform, Cloudera or AWS, big data. Although it might look very surprising to you, but these are the fact where, you are not just a mere pharmacist going to be. You are actually having the combo of technology and the core science that you have gained during your course of time. So here is the opportunity for us to connect every single dot and bring the best healthcare ecosystems where already the transformation happening with respect to remote monitoring, mobile health, general healthcare with respect to IT, telemedicines. There are a lot of things are already there. But what we certainly need to see how smartly we are, using those technologies in today's world. And as my say for the all participants today, we have to be much more flexible because we are transforming, the traditional way of doing the entire thing in the industry versus the modernization by using the very advanced technologies. We are transforming ourselves. We are experiencing new things, right? And therefore, the flexibility is the key to going to be a success in the near days. And therefore, you need to also acquire these tools, techniques, skills in a way to stand unique in the crowded market, because today you are studying, but tomorrow you are going to change the ecosystem by your contribution. And when you going to join the industry, if you come with this advanced skills or the techniques, definitely your presence is going to create an impact in the industry. Thank you all. I always say that mind is just like a parachute. It works best when it is open. So open your parachute. Sky is not the limit, because we can fly above the sky. And as a young generations, definitely you have a zeal to do many things. You have humongous capability to transform the life of thousands, millions, and billions of the people in the coming days. And universe is always there to, to bless you with ease, joy, and the glory. Thank you all, and wish you all the best for your growing career in the future ahead. Thank you all. Thank you. Any queries or questions or any things that you would like to ask, we can take up for another couple of minutes if time permits. Yeah, over to you.
Hello, Dr. Roshan. This is
Yes, please.
from Bangalore. Dr. Roshan, I think what I wanted to ask you was, you mentioned about the drug discovery processes where they normally traditionally cost you around $2 billion. That's I would say that's not more important than the timelines. Like you're talking about 10 to 12 years by the time you start your proof of concept or the phase 1 trial, and then it comes to the market. So we are looking at basically 10 to 12 years of time, and currently the success rate is less than 1%. So do you think that artificial intelligence is going to compress that time from, I think, more than the money? I think what we are also looking at, let's say, from 10 to 12 years, will we be able to bring out the product to, say, in 3 or 4 years time with the success rate of, say, maybe a double-digit success rate? What's your view on that?
Yeah, a very interesting question, sir. This is very what do you call the hot topic in today in the industry, how the AI is shaping, the new phenomena altogether. When it comes to the discovery, obviously, what the scientists look into is all about the safety, efficacy of the molecule, right? On top of everything, for the scientists, cost is not the factor, right? But while doing so, we used to do screening of thousands of compounds, the candidates, right? Now what happens? AI is not stopping or inhibiting those process of screening. What AI is doing is that now we have the already available data sets, the information, the history, the case studies, the outcome of the past so-called researches. So those available data is being used smartly by the AI and giving us the probability of the success or the right approach for that. So the formulator or the scientists are going to use these tools to derive to their conclusions, right? And obviously the phase 1, phase 2, phase 3 studies are part of the core discovery that cannot be overlooked or cannot be compressed at all. These are going to be there, but yes, the initial phase of the pre-discovery, which is very what do you call the complex in nature as well as time consuming, where we spend heavily screening so many the targeted candidates. So here, basis the availability of the information, AI, technologies, tools, definitely that process get optimized, that outcome get very fine-tuned, and then rather than spending on so-called thousands of molecules, we come up with the a very likely candidate having the pharmacological the benefits. So therefore, it's not only going to save the time, but most importantly, efforts as well as the cost. So it's what do you call it? It's a balanced ecosystem now, where we are completely, rather than depending upon the day-to-day practical aspect, we are using the available research, available scientific information and the data. Basis AI is going to give the outcome. And yes, at the end, human mind to take a call. Human mind, intelligence, the formulator, the scientists are going to, define the outcome. AI is just a tool. So we have to have the balance from both the sides. But in a nutshell, as a bottom line, yes, it is instrumental. Rather, the earlier the success rate was around 18%, 13 to 14%. Now with the AI, the success rate is higher. Yeah. Thank you, sir. Yeah, over to you.
Sir, good morning, sir. So along with your presentation about showing the significance of artificial intelligence, you also highlighted the scope and hopes of the pharmacy profession. Thank you for that. And in one presentation, you highlighted that the kind of or the category of drugs which are available in the market, like said that around 79% are generic drugs and 9% are the patent drugs. So,
right.
Out of these 9% of patent drugs, which kind of drugs are majorly patented? Either antineoplastic agents or cardiovascular drugs or anti-diabetic drugs? Can you just highlight that the category of the drugs which have been patented and available in the market, sir?
Okay. Thank you so much for bringing this point. Very, very interesting one. And yes, no, the patent is like, it's all depend, we can patent anything and everything. Okay. If there is an innovation, there is a different way of methodologies, processes. The patent has a different what do you call categories. You can patent the process, you can patent the formulation, you can patent the design also. A lot of things are there. So having patent is one aspect, but industry is never just behind the patent. What they certainly do, or how the industry is working on the intellectuals to safeguard their entire research, is they identify the key molecules as per the market. So if you look into the Indian multinationals or the multinationals, the pharma company, they have the segments to operate. Okay, and this segment is derived by the market. So there are a lot of categories of the drug, which is what you call driving the market. For example, the cardiovascular drugs, okay. On top of that, the diabetic drugs, the anti-diabetic cardiovasculars, on top of that, the oncological drugs. So these certain drugs has the larger market value because there is a heavy demand, because there is a many cases of the diseases. So these are the drugs which has been performing well in the market. So basically, the industry looks on identification of the molecule versus the commercial value, right? And this is that the entire portfolio get designed or developed, and this is further they do the the patenting. So coming back to like the 9% of the patented drugs or innovated drugs. So this is basically derived basis the what is the value is going to contribute in the market, and this value depends upon the total the cases, the total disease conditions, and what is the solution available. So there is an algorithm to design the portfolio, and this is that the industry does their development altogether and does the patenting framework to safeguard the entire intellectuals. And therefore, although the development or the discovery is pretty long phase, and therefore the cost of product is also high as compared to the generic formulations.
Right.
Yeah.
So thank you very much, sir.
Right.
So along with your effective presentation, all the delegates are impressed with the hairstyle also, sir. Thank you very much, sir. Sir, one more question.
Yeah, please.
Good morning, sir. I am
Good morning.
So I wanted to ask, like now, ChatGPT is a new technology where we can give some question and it will give us a detailed answer. So can this technology replace writing jobs like scientific writing, regulatory writing?
Okay, very interesting and smart question, by the way. Thank you.
Thank you.
As I said, as the technology evolving, it has a pros and cons at the same time. Specifically, if you talk about like you quoted this ChatGPT. So what is ChatGPT now? These are the programmed language which has been designed basis the available data sets, basis the available information, basis the available, the content, right? And the chat GPT is going to give the result only on the existing data. Now that there is a difference between Google search versus the chat GPT. The Google search gives the result in the form of various unlimited links of the data. Okay, so here we need to go each and every particular article, particular publications, or particular website link to see what is what type of content it is there, and then we filter out certain content as per our need. Okay, that is how the Google search is doing it. The ChatGPT, what it does, it uses the same information, but the presentation is like as a fine outcome in the very comprehensive way, very conclusive way, the way we actually expect, that is the difference. Okay, but having said that, now Google is coming with the Google Brad, which is ahead of going to be ChatGPT. Why it is so? Because it's going to design the algorithm on the existing info and building the new algorithm set to give the future perspective of the search content. That is interesting. Now, coming to your point, whether it's going to impact, let's say precisely the what do you call the jobs of the people who are in this particular segment, writers and all that. I can't say, we can't say that is going to kill the jobs of thousands or lakhs of the people. But one thing is very clear. If we don't build ourselves or improve our skills, then there is a limitation. Because ultimately, in a way to operate ChatGPT or Google Brad or any such technologies, including AI, ML, and every other advanced technology, we need the human source to operate that. Okay, so the message is very loud and clear to all of us. No matter whether we are hardcore technical experts or not, we also need to learn the technologies. Okay, at least in a way we are enabler to perform these operations using this technology, right? So that's the message. On the other side, rather than killing the jobs, is going to be segregate, rather than 10 people working on the scientific writing, for example, section. Now this chat, what do you call, rather than saying the chat GPT, possibly the Google Brad is going to give the better outcome, right? So we need the certain resources. Human is non-replaceable. Okay, we can't replace a human being. But yes, we are going to deploy the only resources which are equipped, which are skilled. So the message is that we need to equip ourselves in today's world, because tomorrow is even going to be much more challenging, because we are living in the world of technology. And we have to, along with what we are learning, BPharm, MPharm, PhD, my suggestion, recommendation is to also acquire the knowledge of the, so you can perform, you can do the advanced courses on the AI, machine learning. On top of that, drug regulatory affairs, formulation development, intellectual property management. So because that is how the industry looks into the specific candidate, and if the candidates are equipped with the skills, definitely their presence is nobody going to, impact. Yeah, I hope it's a satisfactory answer to you.
Yes,
Thank
sir.
you.
Thank you.
Thank you. Yes, thank you all for giving the opportunity and to be part of today's conference. Indeed, a great pleasure. It was thoroughly, I enjoyed, interacting with you while giving the insights about the artificial intelligence. The power of artificial intelligence is very instrumental. Yeah.
you, sir, for this wonderful session. Jai Shri Gurudev.
By seeking the blessings of His Holiness, Parama Pujya Jagadguru Shri 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 Dr. Bala Gangadharanatha Mahaswamiji, and rendering our humble pranams to Parama Pujya Shri 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 Dr. Nirmala Nandanatha Mahaswamiji, we welcome you all to the International Conference on Innovation and Advances in Pharmaceutical Sciences organized by Sri Adichunchanagiri College of Pharmacy together with Association of Pharmaceutical Research, APR.
This is Visham and Rakshit, feeling extremely glad to host this program and share this pleasant morning with you all.
Shubham Karoti Kalyanam Arogyam Dhana Sampadaha Shatruputti Vinashaya Deepa Jyoti Namutsate.
It means I fold my hands before the light that brings prosperity, auspiciousness, good health, abundance of wealth, and destruction of the enemy's intellect. Light destroys the darkness.
Now, I request the dignitaries to light the lamp.
आराधयामि मणिसन्निभमात्मलिङ्गम् मायापुरे हृदयपङ्कजसन्निविष्टम् श्रद्धाभिषेकै नित्यं समाधिकुसुमैर्न पुनर्भवाय नित्यं समाधिकुसुमैर्न पुनर्भवाय ज्ञानपूर्ण निर्मलवादा ज्ञानपूर्ण निर्मलवादा
Thank you, dignitaries. In a society that worships love, freedom, and beauty, dance is sacred. It is the prayer for the future, a remembrance of the past, a joyful exclamation for thanks for the present. Here is Team Vajrakaya to welcome you all with your dance performance.
Thanks for the dance answer. Okay. ಅದಾಗತ್ತಲ್ವಾ ನನ್ನ ಮೊಬೈಲ್ ಬೇಕಾದ್ರೆ ಅದಾಕ್ ಕೊಡ್ತೀನಿ ಬೇಕು ಅದು ಬಜರಂಗಿ ಸಾಂಗ್ ಅಂತ ನೋಡಿ
ಮೊಬೈಲ್ ಕೊಟ್ಬಿಡಿ
ಇದೆ
ಮೊಬೈಲ್ ಕೊಟ್ಬಿಡಿ ಬೇಕು ಕೊಡಿ ಲೋಕಲ್ ಕೊಡಿ ಹಾಂ ಮೊಬೈಲ್
Thank you. That was indeed mesmerizing.
A multidisciplinary Adichunchanagiri University came into existence in 2018 under the stewardship of His Holiness Sri Dr. Nirmalanandana Mahaswamiji, a great visionary and an ocean of knowledge. The Honorable Founder, Chancellor, is giving a new dimension to the university with a cultural, spiritual touch and a scientific look.
Currently, more than 5,000 students are pursuing their education in medical, pharmacy, nursing, engineering, management, commerce, and education. The university adds to its credit a center for research and innovation and a multi-super specialty teaching hospital with 1,200 beds.
We are highly privileged in presenting you the teaser of Sri Adichunchanagiri College of Pharmacy, Adichunchanagiri University.
Let it be, no? Oh, Zoom, let it be.
Okay. Just attach.
Adichunchanagiri University, one of the mainstream of education that constitute is the Sri Adichunchanagiri College of Pharmacy.
Adichunchanagiri College of Pharmacy was established in 1981. Currently, we offer B Pharma, M Pharma, PharmD, and PhD programs.
SACCP is an acknowledged institution for quality learning and as an outcome of the various facets that give ACCP its characteristic value, difficulty, enterprising students, infrastructure facilities, and industry partnerships. To achieve this vision, the department is committed to providing a course of study for undergraduates which combines curriculum, scholarship, and service engagement opportunities that are high quality, innovative, and intellectually challenging, and employ state-of-the-art technologies.
SACCP is a world of dream to be explored. Our college has built on the belief of knowledge is power. I am very proud to be a part of SACCP.
We are happy to share that Sri Adichunchanagiri College of Pharmacy is MBA accredited and listed in NIRF amongst 100 colleges in India for 2 consecutive years. And Sri Adichunchanagiri College of Pharmacy is also ISO certified for quality management systems.
Audio, video, media room, communications, and broadband-enabled Wi-Fi campus. Library, which is open round the clock. Cultural programs, student fests, festivals, national celebrations make life at SACCP truly colorful. The sports and games facilities at the campus make the students attain physical growth and sportsman spirit.
Our college is completely covered with greenery and comfortable, relaxing, and peaceful environment.
I appeal to all stakeholders to make use of these wonderful infrastructural facilities created to cater the need of academic, research, co-curricular and extracurricular activities for diploma, degree, and various other programs offered by Sri Adichunchanagiri.
Jai Sri Gurudev.
Thank you. Very good morning. This is Lohita joining as co-host. I would like to welcome Mr. Harish KJ, President, Karnataka Drugs and Pharmaceuticals Manufacturing Association and Director, Embryological Laboratories Private Limited. Dr. M.A. Shekhar, Vice Chancellor, Adichunchanagiri University. Dr. K. Bhairappa, Pro Vice Chancellor, Adichunchanagiri University. Dr. C.K. Subbarao, Registrar, Adichunchanagiri University. Dr. B. Ramesh, Organizing Chairperson, Dean and Principal, Sri Adichunchanagiri College of Pharmacy. Dr. Prakash Gowda Navar, HOD, Department of Pharmaceutics, Organizing Secretary and Convener to the dais. We welcome you all. We welcome Mr. Rudra Bansakpati to the dais. We welcome you, sir. Freedom in mind, strength in words, pureness in our blood, pride in our souls, and faith in our heart. Let's salute the spirit of Karnataka. I request you all to raise for Naragiri.
A day without prayer is a day without blessing, and a life without prayer is a life without power. I welcome Pranita and Deepti for invoking the Almighty's blessings through invocation song.
ಸದ್ಭಕ್ತನು ಸಂಪನ್ನನು ಅತಿ ಸರಳನು ಗುರುದೇವನು ಎಲ್ಲರಂತಲ್ಲ ಎನ್ನ ಗುರುದೇವನು ಸದ್ಭಕ್ತನು ಸಂಪನ್ನನು ಅತಿ ಸರಳನು ಗುರುದೇವನು ದುಷ್ಟರನು ಕಂಡೊಡನೆ ದೂರ ಸರಿಯುವನು ದುಃಖಿಗಳ ನೋಡಿದೊಡೆ ಬರುವವನು ದುಷ್ಟರನು ಕಂಡೊಡನೆ ದೂರ ಸರಿಯುವನು ದುಃಖಿಗಳ ನೋಡಿದೊಡೆ ಬರುವವನು ಶರಣರನು ಬಿಡದೆ ಆದರಿಸುವವನು ಶರಣರನು ಬಿಡದೆ ಆದರಿಸುವವನು ಗೈದ ತಪ್ಪನು ಕ್ಷಮಿಸಿ ಸಂತೈಸುವವನು ಎಲ್ಲರಂತಲ್ಲ ಎನ್ನ ಗುರುದೇವನು ಸದ್ಭಕ್ತನು ಸಂಪನ್ನನು ಅತಿ ಸರಳನು ಗುರುದೇವನು ಒಂಟಿತನವನು ಇಷ್ಟಪಡುವವನು ಗದ್ದಲಕ್ಕೆ ತಾನೆಂದು ಅಂಜುವವನು ಒಂಟಿತನವನು ಇಷ್ಟಪಡುವವನು ಗದ್ದಲಕ್ಕೆ ತಾನೆಂದು ಅಂಜುವವನು ಗುರುಕಾಲ ಪೂಜಿಸುವನು ಗುರುಕಾಲ ಪೂಜಿಸುವನು ಭಕ್ತ ಜನಕ್ಕೆಲ್ಲ ಭಯದಾಯಕನು ಎಲ್ಲರಂತಲ್ಲ ಎನ್ನ ಗುರುದೇವನು ಸದ್ಭಕ್ತನು ಸಂಪನ್ನನು ಅತಿ ಸರಳನು ಗುರುದೇವನು ಎಲ್ಲರಂತಲ್ಲ ಎನ್ನ ಗುರುದೇವನು ಸದ್ಭಕ್ತನು ಸಂಪನ್ನನು ಅತಿ ಸರಳನು ಗುರುದೇವನು ಅತಿ ಸರಳನು ಗುರುದೇವನು ಅತಿ ಸರಳನು ಗುರುದೇವನು
Thank you, Pranita and Deepti, for the melodious song. I request Dr. Prakash Gowdanavar, HOD, Organizing Secretary and Convener, to welcome the guests and give the opening remarks.
Jai Shri Gurudev. My humble pranams to Puja Sri Dr. Balagadhar Mahaswamiji, Puja Sri Dr. Nirmalanandanath Mahaswamiji. A very good morning to everyone. It's my privilege to deliver the welcome address for this international conference. This is the first hybrid international conference we are organizing on the topic the innovation and advances in the pharmaceutical sciences, current scenario and future perspectives. in association with the Association of Pharmaceutical Research. At the first, I would like to welcome our Honorable Vice Chancellor, Dr. M.A. Shekhar sir, and I request Mr. Rudraban Satpathy, the CEO and founder of APR, to felicitate him with the token of love and memento for the conference. Thank you, sir. Thank you. It is my pleasure to welcome Pro Chancellor and Research Director, Dr. K. Bhairappa. Welcome you, sir. I request the organizing chairperson, Dr. B. Ramesh sir, to welcome him with the token of love. It's my privilege to welcome our chief guest of today's inauguration function, Mr. Harish KJ, who is the president of Karnataka Drugs and Pharmaceutical Manufacturing Association, and is also director of Pharmaceutical Laboratories Limited, Bangalore. I request our Honorable Vice Chancellor. Thank you, sir. Now, again, it's my pleasure to welcome the respected Registrar of Adichunchanagiri University, Dr. C.K. Subbaraya, sir. I request Dr. B. Ramesh, organizing chairperson, to felicitate him. Thank you, sir. I also welcome Mr. Rudrabanu Satpathy, who is the CEO and founder of Association of Pharmaceutical Research, and also our co-organizer for conducting this international conference. Now I request our Honorable Vice Chancellor, Dr. M.A. Shekhar sir, to felicitate him. Thank you, sir. Now it's time to felicitate the backbone of this international conference, Dr. P. Ramesh, who is the organizing chairperson. I request Mr. Rudrabanu Satpathy to felicitate, please. Yes. Now it's a most important time to welcome all our eminent speakers, of course, who will be on virtual as well as digital mode, and who will be dealing with all the key points, the latest trends, and various facets of pharmaceutical sciences. I welcome all the speakers who are physically present and also virtually present to this conference. I also welcome the principals and deans of our sister institutions of Adichunchanagiri University. Firstly, I'd like to welcome Dr. Ramu sir, the Principal Commerce College. I'd also like to welcome Dr. A.T. Sivaram, the Principal of College of Education. Welcome you, sir. Dr. P. Narendra, Principal, BJS IT, Adichunchanagiri University. Welcome you, sir. Dr. Prashant Kalapa, who is Dean, the Faculty of Natural Sciences, Adichunchanagiri University. Welcome you, sir. And at the last, the delegates. Actually, we were in a dilemma to hold this international conference at this location. We were hesitant to organize the international conference because of our location. But I'm very much happy to see the tremendous response of registration. So totally we have received 440 registration, out of which 265 are physical mode and 175 will be on the virtual mode. So my wholehearted welcome to all the delegates who have come across the country to attend this international conference on innovation and advances of pharmaceutical sciences. Welcome you all. We also arranged the research presentation in terms of oral as well as a poster. I'm also happy to announce that there will be around 225 research presentations. So all the delegates who are presenting, right? So we'll be forwarding their all the research thoughts in terms of the poster as well as oral presentations. So at last, I again welcome. Have a pleasant learning for these 2 days of conference. Thank you.
Thank you, sir. We welcome you too, sir. I request Dr. Ramesh to welcome Dr. Prakash Gowda.
Thank you, sir. Association of Pharmaceutical Research, APR, is one of the world's leading professional association. Today, we have Mr. Rudra Banu Satpathy, CEO, founder, APR. I welcome you, sir, to give us your intellect with regard to APR on this occasion. Welcome you, sir.
A very wonderful good morning to everyone. My most respected His Holiness Jagat Guru Sri Nirmalanandanath Mahaswamiji, a revered Vice Chancellor of this esteemed university, my most respected principal of this leading college, dignitaries, honorable chief guests, faculties, organizing chairpersons, organizing committee members, scientific committee members, and all dynamic faculties of this college, along with my loved students. Last year, we celebrated 75th years of our nationality. More, at this point of time, our effort towards pharmaceutical innovations not merely encompasses the scientific aspects, rather do prioritizes on the spirituality verticals as well. In fact, the great deals of science encrypted in forms of Vedas and Upanishads like Charaka Susruta that says Dirgham Jivitam Manavicham Bharadwaja Upamagata. Even the science and healthcare studies encrypted in form of regional dialects like Sanskrit, Pali, and other languages still remains not translated or studied. With due course of time, with the progress of different scenarios, ups and downs, celebrating 75th years of independence last year, our country has emerged as fourth largest economy, surpassing United Kingdom. Today, the effort that were given by our ancestors of in time, we generations have come forward because it says Karmanya Vadikarastu Ma Pareshu Gadashana. And India is today third largest producers of pharmaceutical productions in world in terms of volume and 14th in terms of value. If you look towards generic medicine production, we are 20% contributors of generic medicines all across the globe, and we rank 12th among other countries. India nurtures more than 3,000 pharmaceutical industries and 10,000 plus manufacturing units, thus creating its presence in global market. Our attitude, our commitment has always been towards welfare of humanity. Our tradition has always focused on Vasudhaivam Kutumbakam, that has always emphasized to make the globe a peaceful place of living with harmony and brotherhood. That says, Sarve Bhavantu Sukhina, Sarve Santu Niramaya, Sarve Vadrani Pashantu Makaschita Bhavav. And the result is that our country contributed COVID-19 vaccines to more than 71 countries with 580 lakhs doses in time of pandemic. This could be possible because a strong higher education system we have created. Our country inculcates more than 4,000 pharmaceutical colleges and universities. And it's high time to industries to come forward to establish the center of excellences in these institutions. Even United Nations organization has been focusing on capacity building. As millennia academic entrepreneur, we personally believe from the team of Association for Pharmaceutical Research APR that capacity building towards next 50 years is the most important aspect, and this could be possible if proper skill development and capacity building could be brought at students level. We are trying hard to bring industries and research laboratories very close to academics to make our studies and educations synchronized with practical trials and applications. We are creating an environment that will fulfill all your needs. Looking into community management, it's very important. If we look into professional studies, community management is the most important aspect. We create an environment, we speak about, we talk about, we do implement globalization also, because we strongly believe learning beyond classroom is a very important aspect. And our students in our pharma colleges do deserve the access to the global content, global knowledge sources. So we facilitate this thing through our conferences and events we do. We operate in more than 70 countries all across the globe. In fact, last night, in mid of night, we inaugurated a chapter at Peru that will deal with South American and Latin countries. This could be possible if proper faculty exchange programs, student exchange programs could be brought into existence. We are in a verge of creating the knowledge centers, knowledge source, by connecting world-class technical and professional leaders and facilitate organizing knowledge-centric activities like conferences in nook and corner of the nation. I express my hearty gratitude to the faculties, especially the principal, Prakash sir. In fact, I had a plan to go back to Chennai today, but looking at the hospitality and affection, I postponed my travel for tomorrow. And it's really a amazing experience. Even I express my gratitude to my entire team of APR, those who have relentlessly worked for last 4 months to make these 2 days of conference valuable, so that some values and comfort zone could be added to your life. And I believe this conference, including thought-provoking keynote speakers and experts, chairpersons, will have amalgamations of thoughts, beliefs, and ideas, and will be a great opportunity for learning and networking. I wish everyone a wonderful conferencing with each other, networking for these 2 days, and wish you all the best. Thank you very much.
Thank you, sir, for being a role model and inspiring today's youth. And thank you for enlightening us with your valuable words.
Now it's time to release the souvenir, which is a compilation of scientific abstracts. May I now request the dignitaries on the dais to release the same. Thank you.
May I now request our chief guest, Mr. Harish Keche, President KDMPA and Director of Embiotic Laboratories Private Limited, to share his insights with us.
Good morning to all. Ellarigu namaskara. At the outset, I always wanted to come to. I've been traveling on this road to Bangalore a few times, and always when I come here, I pass through this road. Always wanted to get into Adichunchanagiri. And what's the occasion like? I think this is the first time I'm stopping by at Adichunchanagiri. Wow, blessed to be the blessed by the divine presence of Param Pujya Jagatguru Sri Balgangadhar Mahaswamiji. I think blessed I am by him. I think whenever I remember almost more than 25 years back when I used to travel on my scooter from my home to factory, and there was a construction on Mysore Road in Bangalore which was going on. I think of the flyover, the famous Sissi Road, Sissi Circle flyover. And when the flyovers were inaugurated, and after a few years it was named after BGS, then I was, that's when I actually, although I'm Karnataka by birth, but then I'm Marwadi, but then this is when actually I came to know about Dr. VGS, and then I started more research and knowing about the work, the kind of work, what it has been done today. And this is what I'm seeing today. Since last time night when I came over here, it's been a huge medical college with 1,250 beds. Wow, my God. A hospital and medical college with 250 seats, pharmacy college, nursing college, engineering college, PU college. That's in this place. And then Bangalore, you have BGS hospital, then BGS residential school, which is right behind my factory. And this is the kind of work, I think, amazing. The Karnataka is blessed to have the blessings of Mahaswamiji and the kind of work which has been taken forward by the present Jagat Guru Sri Nirmalananda Swamiji. I think great and the kind of benefit what the people of this country and people of this state are getting it in terms of education, it's unparalleled. Education is God's work. And I'm happy and honored. Today I'm standing on this amongst you, the August gathering, and delivering my thoughts on innovation and what's the way forward for the. No, coming to the innovation. My first point, I think wherever I go, I talk about. I think the biggest innovation happened maybe almost a century back, not today. Can anybody tell? I think except for the few of us who already heard me earlier, I think the biggest innovation. Can anybody say that? And because of which we are all sitting here today. I can see a smile on some of the faces who know the answer. I think the biggest innovation what happened, this whole profession of medicine dispensing and medicine administrating to the patient was in the doctor's hands. And in the modern and present, we talked about mixtures, powders, and all that. And sterilization used to happen in the crude way, injectables were given. And the biggest innovation which happened was that we took the profession from the doctor's hands to the pharmacist. I think that's the birth of the pharmacy profession. I think we are all today sitting here celebrating it. So when you're talking about the next growth of next phase of innovation, what this profession can give to the society, I think this is what we will look back to the history. It's all because of that particular one incident. And today, if this whole mankind is benefiting because of the medicines unit dosage forms, it's all because of we pharmacists. And today we are talking about artificial intelligence, machine learning. We're talking about big data analytics. All this can happen only if we are ready with the multidisciplinary approach. We as a pharmacist, if we are ready with the multidisciplinary approach, we can take this profession to a greater heights, bringing respect to our profession, and also we are talking about the benefit to the billions and billions of the patients across the world, and this can happen only through pharmacists. Now, when you talk about innovation, what's innovation? We need to distinguish between a discovery and invention and innovation. When you talk about innovation or even a discovery and invention is something which has come newly, which we have brought in newly to this world. Innovation is improvement upon what we already know. I think that is actually very important on the basic stage, whether it is the kind of processes, the services, the kind of innovation or the manufacturing product, whatever, it's an innovation. Now, I'm happy this Adichunchanagiri College of Pharmacy is conducting such a wonderful program on international program on innovation and advances in pharmaceutical sciences. I congratulate the Chancellor, Pro Vice Chancellor, Dr. Ramesh, and the whole organizing team. I think this is the need of our. I think since on 15th of August, our Prime Minister mentioned about, from the Red Fort, mentioned about the Jai Anusandhan. And this is the kind of push I think this is just words. This is the kind of motivation what our Prime Minister gives. Just with that was the kind of revolution has started in this country on research and innovation. And this is more applicable to the pharmaceutical industry than any other industry I would talk about. If you have to really look at the benefit which can bring, the incremental benefit it can bring to the humanity and this particular country. It is always said that about 1% one rupee investment on the health brings about 5% increase in the GDP of this country. I think this is the kind of incremental benefit this program of research and innovation will bring to this country. I think if you over the course of next 2 years, I've been seeing that there are huge number of programs which have been and the lectures which have been organized, whether it's a modern pharmacology, we are talking about drug regulatory affairs, pharmacovigilance, machine learning. I think before this inaugural program, here a huge thing about, I think many of the students who are keen on looking at what lies them for the future is artificial intelligence. We know artificial intelligence. We use this on a day-to-day basis. We talk about Google Maps. It's artificial intelligence. We talk about lot of things, machine learning, data analytics, big data, the kind of data which is available on your mobile. How do we analyze? Do we have that? It can happen only by machine learning and the kind of artificial intelligence which we can brought about. So having said that, I always say, what is the benefit of innovation for a pharmaceutical industry? When I talk about innovation, it's as I said, it's an improvement about the things which we already know. Now, what benefit it brings to the industry and to the profession is, I am competing with my competitors on quality rather than the price. At the same time, I am bringing multiple benefits to my patients. Now, what sets us apart as a pharmacist or the organized allopathic medicines? What sets us apart from quacks? Anyone? Many of your friends would have told you, whenever you are, especially when you have a chronic disease like diabetes, you have an orthopedic disease or a cardiac disease, many of your friends would have told you who are into non-medical, non-healthcare field would have said you, okay, this particular small-time doctor is there, he will give you some medicine and which brings about your disease gets cured. Isn't it? At some point of time, many of us would have gone through that. Now, what sets us apart is, our whole profession is on 3 aspects. I think many of you would have learned it, but then probably have not dwelled upon it in deep, is safety, efficacy, and quality. For us today, efficacy, everybody can prove it. It's not a very big thing. Now, what takes us apart is safety. Safety of the medicine. That's why you will see that any new drug which is being developed takes around 10 to 12 years for development, because efficacy studies, when you're looking at phase 1 and phase 2 trial, doesn't take much of a time. It's a pre-discovery or a discovery phase of the development phase. It's a phase 3 trial and the phase 4 trial. Even after the product is commercialized, the phase 4 trial, which takes years and years altogether. And this is where, when you do an innovation, that's what I think I was talking about. When you do an innovation and with this multidisciplinary approach, we can bring not only the cost of discovery and invention, we can also bring down the timelines. Today, what we are talking about, 10 to 12 years, $2 billion, success rate of less than 1% for a new drug. Can we make it into a double digit? Can we do it within, say, $200-300 million? Can we do it and compressed into 5 to 6 years with the use of analytics and all that? Now, this is innovation. Imagine the kind of benefits it can bring to the humanity. Today, what medicines, people will get in 10 years from now, we can get in 5 years from now. Now, some of the newer, if you look at the newer products, newer drugs which are coming into the market, are basically very complex, big molecules. We are talking about biologics and biopharmaceuticals, which has its own complexities. Now, what Again, when I talked about innovation, when we are competing in terms of quality rather than the price, it also brings you one more benefit. The benefit is you're creating an entry barriers for your competitors. For example, the innovation can be as simple as how do we reduce the impurities in a chemical compound. Now, in US FDA, and increasingly when you look at, I don't know how many of you have looked at the new Indian Pharmacopoeia of 2022. When you look at that, the impurities which have been defined in the pharmacopoeias have been tightened. What was, say, for example, the limit of 0.5% has been brought down to 0.2%. Now, US FDA gives it, I think, unless you see, for the pharmacopoeia drug, we know the impurities are defined in the pharmacopoeia. But for the non-pharmacopoeia drug, for an innovator, US FDA is very clear about the impurity profile of the drug, and they will not allow higher amount of impurity. The reason is very simple. When we do a safety and efficacy studies, we just study the active ingredient. We don't study about the impurities. And when you are looking at impurities, which we are talking, when it's a long-term therapy, whether it's for say cardio, diabetic, most of these impurities are carcinogenic. Whether you are looking at nitrosamine or any other kind of thing, most of them are carcinogenic. Now, if you are taking this drug on a long term, say 5 years, 10 years, your drug itself will give you cancer. So if you can, by some way of innovative process, if you can bring down the impurities, that becomes the entry barrier for other competitors to get it. The innovation can be in terms of processes. I'll give you an example within my own company. Ranitidine tablet, it's a huge volume product. Earlier, we used to manufacture with a wet granulation method. I'm talking about 20 years back. And 20 years back, when we realized the kind of opportunity what we have, now 20 years back with the wet granulation, I could make only 1 million tablet a day with my small facility. Without changing the facility, without changing any investment, we spoke to the API manufacturers and make the changes in the flow, adequate changes in the flow properties and the angle of repose and all. I think that many of you have already studied that. With that simple innovation, we converted that formulation to direct compression drug. Result, instead of 1 million tablet, I'm making 2 million tablets a day. My cost is coming down by half. And we have done crores and crores rupees of worth of business. Huge. Second thing is, when you do a ranitidine, it's a very sensitive molecule. So during the processing, the impurities develop. Now we were able to, although in those days the pharmacopoeia, the impurities were very relaxed, but because of that, our innovation in the processes, our impurities started coming down. My stability became better. Wow. Similarly, we have been, I think, as we are concerned, we are one of the pioneers in doing dispersible tablets when you're talking about more than 3 decades back. Today, it is very common. We were the pioneers in doing dispersible tablets. So instead of having a dry syrup, a bottle, we can carry a kid's tablet in your pocket. And this is the kind of small, when there is a big scientific conferences that we talk about drug discovery, invention, but then it's a big ticket game. But for us, for a SME or for somebody who is in a startup, these kind of small innovations are very important. I can give you a number of examples. Know about this acne drug called as isotretinoin? Okay. Now this drug, an innovator had this regular soft gelatin capsule. What this Ranbaxy did more than 2 decades, it was in the US. 2 decades back. Now isotretinoin to be effective, it has to be taken by a high-fat milk. High-fat meal. And this is given for acne. Now imagine a girl having acne have to take a high-fat meal. Now what this Ranbaxy did was, when you look at innovation, it's like you have to look out for opportunities. You have to keep your eyes and ears open. So when you look at those opportunities, what this company did was, they developed a patented lipose technology. Means the base was a lipid base in the capsule. Okay, so you don't have to, when it was launched, you don't have to take with a high fat milk, you can take as a regular capsule. And within no time, this innovator's drug was out of the market. And the US, it became the #1 brand in the US. For 2 decades back, an Indian company, a medium-sized Indian company to become big in US was quite an achievement. Like that, you can see when I talked about you build entry barriers. There are a lot of, you develop your complex drugs by innovation. There are a lot of things can be done. Some of the innovations, I think what we are talking about is sustained release formulations. And sustained release formulation, we have developed indomethacin. We take a regular indomethacin, you have what you call as gastric irritation. With sustained release, you can always minimize that. With iron supplements, ferrous sulfate capsules and all that, you not only increase your, decrease the gastric irritation by a Spansel technology, but you also, what you do is, you also increase the extent of absorption of iron supplements in your body. I think that's a kind of huge innovation what we are talking about. And when we are talking about innovation, what is more important is ecosystem. Do we have a research ecosystem? This is what this country is going through. Research ecosystem, when I'm talking about, was the kind of funding which is available. I think very important for the universities. Whether what's the kind of funding which is available, whether it's from the government sources or the private sources. And not only the funding, we are also talking about what are the high-ranking universities in the country? What are the number of patents? The academics and the number of PhDs what we are having. And what are the kind of publication, high-impact publications we are making? I think all this makes kind of a, I think today I'm not carrying that slide with me. I think whenever next time we are coming, I have a slide with me where I have compared the 14 countries in this world who is going to be next big in pharma. And what we are seeing is countries like China and Israel, not only the funding is available, the ecosystem, the number of high-ranking universities, these are all 14 emerging countries. Now, India is still that we have the ecosystem, but we don't have the kind of sufficient kind of a funding. If we can bring up that funding thing, I think nobody can stop us from becoming #1 in this world. I think my previous speaker talked about we are third in terms of volume and 14th in terms of value. We want to be third in terms of volume and third in terms of value. And that can come only about by innovation. It cannot come by, the country has a target of $130 billion in pharma, only manufacturing. I'm not talking about, I have not included nutraceutical, I have not included cosmetics, I have not included Ayush, I have not included services. These are the kind of opportunities we have. $130 billion only in pharma drugs manufacturing business I'm talking about. And this can happen only by innovation. And I'm excluding, I'm not a big fan of a new drug discovery or something, because for today's, for Indian context, it's not a viable option. Yeah, I'm talking about incremental innovation, whether it's a process innovation or the new drug delivery system, or we have the new dosage forms. Fix those combinations. Huge opportunity for India if we can prove this. Scientifically, we can prove the safety and efficacy of the drug. It also brings down, when you talk about fixed dose combination, it also brings down the cost of compliance. The patient will be more happy to take one tablet rather than 3 different tablets of a 3 different drug. Especially when you have a chronic disease, the patient will dread to take 10 tablets a day. Give them 2 to 3 tablets, he'll happily take it. This is where the fixed dose combinations will be very helpful. This is going to be the incremental innovation this country is looking at. There are huge kind of opportunities. Services, innovation in services. And I'm telling you, in this kind of artificial intelligence, machine learning, and big data analytics, sir, it can happen in a multidisciplinary university like Adichunchanagiri. It can happen only in Karnataka. If it has to happen anywhere in the world, the kind of innovation, anywhere in the world, I'm not talking about the country, it can happen anywhere in the world, it can happen only in Karnataka. And it will happen, whether you like it or not, the world likes it or not, it will happen. This is the kind of opportunities where we are very, very bullish about this country and our state. The services, world is looking at, I think I spoke about those services just before the budget on the national TV, and I talked about Services, it's going to be the bigger than the current pharmaceutical industry, whether we are talking about stability studies, contract research, pipeline studies, clinical trial, whatever you are talking about, whether it's a pharmacovigilance, again, very big pharmacovigilance program. It's going to be big, and this is the future of pharma, what we are looking at. Innovation, new age learning, this is what will happen. I think one last point what I want to tell you about is, we have seen many multinational companies. They innovate the drug or invent a drug and go all across the world, and wherever they get a manufacturing site, they manufacture and sell in those particular countries. This is what lot to learn from there. And I have given a word called technology indigenization. We need not do manufacturing within this country. We can do manufacturing all across the world wherever it is possible for logistics and supply chain. But technology indigenization, technology should be in our hand. Look at US, look at Europe. They are the owners of technology. They may not be owners of the manufacturing sites. They may not be owners of their marketing teams. But this is what is important. If India has to become the first world country, I think this technology indigenization is extremely important. I think with that, thanks Adichunchanagiri College of Pharmacy, especially the Honorable Vice Chancellor, Dr. M.A. Shekhar, Vairapaji, and my great friend Ramesh. And also thank all the audience for patient hearing. Thank you very much. Jai Hind, Jai Karnataka.
Thank you, sir, for your remarkable words. We are truly lucky to have you here. Now, I request the APR founder, CEO, Mr. Rudra Bhanusathpathy, sir, to hand over the memento to Sri Adichunchanagiri College of Pharmacy for organizing the APR Conference 2023. Thank you, sir.
Success is not final. Failure is not fatal. It is the courage to continue that counts. Now, I request our Honorable Vice Chancellor, Dr. M.A. Shekhar, to address the gathering.
Right. Jai Shri Gurudev. ಕ್ಷೇತ್ರ ಪಾಲಕರು ಕ್ಷೇತ್ರ ದೈವರಾದ ಶ್ರೀ ಗಂಗಾಧರೇಶ್ವರ ಸ್ವಾಮಿ ಶ್ರೀ ಕಾಲಭೈರೇಶ್ವರ ಸ್ವಾಮಿಯ ಪಾದಗಳಲ್ಲಿ ನಮ್ಮ ನಮಸ್ಕಾರವನ್ನು ತಿಳಿಸೋಣ, ಅವರನ್ನು ಪ್ರಾರ್ಥಿಸೋಣ. ನಮ್ಮಿಬ್ಬರ ಪರಮ ಪೂಜ್ಯ ಜಗದ್ಗುರುಗಳ ಪಾದವಿಂದಗಳನ್ನು ನಮಸ್ಕರಿಸಿ ಅವರ ಆಶೀರ್ವಾದವನ್ನು ಕೋರಿ ನಿಮ್ಮೆಲ್ಲರಿಗೂ ಇನ್ನೊಂದ್ಸಲ ನಮಸ್ಕಾರ. I will not say thank you, welcome. Well, dignitaries on the dais, respected resource persons and invited speakers, and more importantly, my dear students. The registrants are our students. I'm proud to say you are all our students. With this, we'll go forward. And I was talking to Dr. Ramesh, what I'm supposed to talk to the pharmaceutical company people or the budding pharmacists and pharmaceutical scientists. And he gave me, not he, Dr. Harish Singh. You are a doctor, yes? Okay, you are a doctor. Doctor is to teach. He has been teaching. Gave me a pointer stating that in the last century, the innovation, if at all that happened, was pharmaceutical people or pharmacists became the front line people and not the doctors. And do you know why from the last century to this century I've become thin? You people took away my practice in mind. Anyway, it worked out very nicely because it prevented me from becoming a diabetic and hypertensive and having a heart attack at the age of 63. I'm now 64. Thank you for wrestling away all the money from us. It doesn't really matter. Okay? Now, I keep telling the young minds and young souls that one of the important concepts that you should develop is the power of observation. Okay? Power of observation. Keep looking around, look out, etc., etc., and develop a power of observation. I'll put 2 observations. Then we'll go further, and I will not take much of your time. Ramesh and I were talking about in any conferences or in any important event, the bottleneck is inauguration. There is a big bottle where all you are waiting for the escape from the bottle out into the open space. What is the bottleneck? Inauguration. We will see to it that it's done in another 10 minutes or 5 minutes. It's okay? Right. Do you know looks are deceptive? There is a statement called looks are deceptive, and there is another statement called aspirations. So let me tell my observation when I walked in. As we were walking into the hall, I saw somebody talking. I thought it's a female. Then the voice did not register. The voice was a male, the look was a female. Looks are deceptive. The second one is aspiration. You saw a very beautiful rendition of Sri Anjaneya Swami's aspirations. Did you not see? I was given to understand they got fantastic accolades in Midday University recently. They stood runners up in dance. The aspiration is, I was given to understand when I was a small child and I was growing up, Gadha Yuddha, mace battle, is always done by people who are well built. Bhima, Duryodhana, Ghatotkacha, and all that. Any one of them are well built? They all look as if they were starved. This is aspiration. They are praying to Anjaneya to give them physical as well as mental strength. It was fantastic. So do you understand the power of observation? I've just given you a simple concept. Please inculcate in your life. Now, International Conference on Innovation and Advances in Pharmaceutical Sciences. A lot of things have been talked about innovations and advances. I will tell you a simple story to start with. A middle class parents put all their efforts to make their son a very good aeronautical engineer. As usual, when you get a lot of education in India, you walk out of India. You don't work in India. You go out. He went to NASA. After some time, they did a very, very innovative spaceship. And he was proud and asked the parents to come to America for the inauguration. During inauguration, the father asked son, Beta, kitna deta hai? How much does it give per liter? Innovation should be, if you ask me, as a very, very good researcher, should be about cost effectiveness, cost benefit, and benefit risk. Otherwise, innovations, you bring out innovations, all sorts of things that it has been spoken, and it costs about 50,000 rupees per dose. Do you think that is a good innovation in science? I don't think so. So keep this in mind, youngsters. When you become good scientists and good researchers, keep innovation. Yes, innovations bring advances, but advances should also help the population or the populace that you're going to help as far as cost effectiveness and benefit risk ratio is concerned. Classic example, if you ask me in a non-communicable disease thing is aspirin and statin. Remember, cost effectiveness, aspirin wins hands on, 75 paisa. Effective, fantastic. Benefit risk, as you grow older, above the age of 60, 65, you give them aspirin. Above the age of 75, you have a call from the emergency room, they're bleeding. But when you get cost effectiveness today of statin, benefit risk of statin, it is fantastic. So a small example, and as far as science is concerned, how many of you believe in God? Stand up on the bench. I keep asking everybody that. Everyone, if you don't believe, at least I'm sure you believe in some supernatural power in nature, right? So there was a small discussion or a debate or whatever you want to call about science and faith. Science and faith. Science says if you can't prove it, you don't have faith in it. But faith says if you can't prove it, better you have faith in it. So please have faith in your own strength. Please have belief and faith that something can come out of your life. Only then science will prosper. Only then innovations will work. This is a great innovation. Don't you agree? I remember in 1997 when I bought my first mobile, it was this big. I could fend off my from attackers. It was like a small maze. Today it's so small, it does every damn thing. So frugal engineering or frugal medicine is the way to go, and we go beyond that. The second thing is, there is a statement that says, you can get a lot of inspirations from many people. Don't you agree? This is one of the podium where you get a lot of inspirations by the speakers who have done a lot of work in life. But determination is entirely yours. So after 2 days, when you go out of the hall after a very good 2-day program that you go through, mental food and also physical food, I hope, you should have the determination to do the best to the population and youths. Can you do that? Finally, I will not talk about sciences and all that, don't ask me. I just wrote it down as we were sitting there. Finally, youngsters, my dear students, your smile is your logo. You're all talking about making industries and making money, bringing India to the best, blah, 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 blah. Your smile is your logo. Your personality is your business card. And the way you treat and make others feel is your trademark. Logo, business card, and trademark. It is you, not anything else. Thank you. Yes.
Thank you, sir, for turning your thoughts into beautiful words. We really appreciate your time with us. No duty is more urgent than that of returning thanks. Gratitude can transform common days into Thanksgiving days, turn routine jobs into joy, and change ordinary opportunities into blessings.
Now, I welcome Dr. Yunus Pasha, HOD of Department of Pharmaceutical Chemistry, to propose the vote of thanks.
Jai Shri Gurudev. So good morning to one and all. Gratitude is not only the greatest of virtues, but the parent of others. A warm and cherished welcome to our Honorable Chief Guest, Mr. Harish K. Jain, President, KDPMA, and also a Director of Embiotic Laboratories Private Limited, Bangalore. Respected speakers, APR members, faculty members of our institute, and also the faculty members of other institutes, students, delegates, and everyone. It is my privilege to propose a vote of thanks and acknowledge the contribution of those who work hard to make this international conference successful. I, Dr. T.Y. Pasha and Dr. Bharti, as in charge for the organizing committee of this international conference, and on behalf of entire fraternity of faculty member of the institute, first of all, my sincere thanks to Almighty God and Reverend Swamiji for making this international conference a grand success. By His blessings and grace, we are able to make what the event is now. On behalf of my institute, Sri Adichunchanagiri College of Pharmacy, I give a real heartfelt vote of thanks to our chief guests, speakers, and for our Adichunchanagiri University respected official bearers, office bearers who spent their time for gracing the occasion. Today, we have the opportunity to hear your thoughts, and this is definitely encourages us in our future events and days to come. I'm also thankful to the anchorings like Risha Sneha, S. Lohita, and Rakshita for their efforts towards the anchoring. The elegance of my thoughts and the interpretation of everything, I need to mention my deep sense of appreciation to all the staff members and the in-charge members of the hospitality committee, stage committee, scientific committee, food committee, and for all other members for delivering their duty. Last but not least, on behalf of our institute, I want to express special thanks to one of our faculty members who, from the date of finalization of this international conference to till today, constantly coordinated with APR members, delegates, speakers, committee members, and also he is the convener and organizing secretary of this international conference, that is Dr. Prakash Gaudna. Thank you very much. Man behind all the movements and brought to the final conclusion and to support for successfully organizing this conference is none other than our principal, our beloved principal, Dr. B. Ramesh sir. Thank you very much, sir, for your guidance. On behalf of Institute, I extend my deep sense of gratitude to APR, CEO, and all other members who have supported us and for assisting to hold this conference and guiding at each and every moment for this conference. And I also thank the media people for gracing the occasion. Thank you, one and all.
Thank you, sir. Here we are at the end of the session. I request all the delegates to be prepared for the upcoming keynote sessions. High tea has been arranged in the college quadrangle. I request dignitaries and delegates to proceed towards the card. See you all after the tea break.
Anything else?
Total pharmacology.
Share, right? Yes.
I request all the delegates to gather in the auditorium. The session will start now. Welcome back. Now, for another exciting session, we have Dr. Mahadev Rao, Professor, Department of Pharmacy Practice, Manipal College of Pharmaceutical Sciences, Manipal Academy of Higher Education, Manipal India Coordinator, Center for Translational Research, co-founder, Global Cancer Consortium. Sir is here to talk on integration of genomics and big data for precision medicine. I request Sir to take over the session.
ಗೀತೆಯ ಬಳಕೆಗೆ ಕೇರಳ ಹೈಕೋರ್ಟ್ ತಡೆಯನ್ನು ಕೊಟ್ಟಿದ್ದಲ್ಲ, ಅದಕ್ಕೆ ಈಗ ಸುಪ್ರೀಂ ಕೋರ್ಟ್ ತಡೆಯನ್ನು ಕೊಟ್ಟಿದೆ. ಹಾಲು ಬಳಕೆಗೆ ಇದ್ದಂತಹ ನಿರ್ಬಂಧವನ್ನು ಸುಪ್ರೀಂ ಕೋರ್ಟ್ ತೆರವುಗೊಳಿಸುವ ಮೂಲಕ ಮತ್ತೊಂದು ಮಹಾ ಗೆಲುವು ಕಾಂತಾರ ಸಿನಿಮಾಗೆ ಸಿಕ್ಕಿರುವಂತದ್ದು. ಕಾಂತಾರ ಸಿನಿಮಾ
Thank you very much. Very good morning to all of you. And next, I think 30 to 40 minutes, I'm going to talk about some of the interesting topics, the impact of genomic or big data for precision medicine. I'll show some of the work also we did with this big data. So this is a disclaimer notice. And if you look at this slide over the years, most of the treatment given to the patient, time versus data, is in the last century is basically based upon the guessing. So I think in the last century, most of the treatment was given to the patient based on the data, which was not there enough to give the best treatment to the patient. But since the invention of human genome in 2003, in the last 25 years, what data we have collected through the big data system in the genomics and others, I think it is unbelievable for the last maybe hundreds of years. So thanks to the the new technology, what we are going through now, I think first speaker was talking about the same thing, big data, AI in the healthcare. So again, human cognitive is very important to treat the patients. I think now it is more technological evolution compared to what happened maybe in the last 100 years. So when you talk about the treatment to the patient, now we talk about not the patient, we have gone to the cell. Everything we talk about a single cell, we have gone to that far because of the technology. You can see here, these are the different technologies available in the genomics, like chip sequencing, methylation sequencing, DNA sequencing, RNA sequencing. So when human genome was invented back in 2003, it was a human draft genome, but actually the full genome was released last April 2022. So if you look at in last 20 years, the invention in the genome has happened in every disease. I think there is 1,000 genome project, around 500 different pathogens they have sequenced. So with the dramatic invention in terms of cell I'm talking about. So with these inventions, different technologies, one can see the mutation of the genes, and one can see the genetic signature, biomarkers, and molecular subtypes, etc. And eventually, they are going to treat the patient. That is what we call the precision medicine. And to do this, there is a lot of consortiums are formed. For example, for human genome, there were almost 20 institutes started with, but now more than 500 institutes participating in the understanding the genome in the different diseases. So it's a very significant advancement. So this is where we are talking today about the precision medicine. So what is the precision medicine? It's basically enough one. So usually, I think you all may be knowing when we talk about how many patients are there in a group, but in this case, we are talking about enough one, a single patient will be taking. So providing the right treatment, right patient, right dose at the right time. These 4 mantra is derived by the European agency, EMA, and I do believe that it's very important, you have to give the right treatment, right patient, right dose at the right time. As previous speaker mentioned, multidisciplinary team is very important. That is where this whole precision medicine concept came. So earlier, the treatment was a physician will be the person who will tell what to do others. He doesn't care patient actually. That's a traditional system. He will tell nurse to do this, pharmacist to do this, and other healthcare workers. But here, patient is patient-centric treatment. Here, we will be together. All the healthcare disciplines are together, and we are treating a single patient. So this is a new kind of a treatment, called precision medicine. As you know, in the tailored approach, what we do, group of patients will give the medicines for the treatment of the diseases, and end up with 80% of the time drugs just don't work for them. 20% success. But in the case of precision medicine, you stratify the patient based on their genome and other, health, other profile of the patients, and eventually treat the patient. So this is a very interesting. You can't find this kind of studies or papers. Rarely you can see. This is the one paper published in the Nature. They talk about imprecision medicine. So that means this is a study from the United States where you can see the different drugs which are sold in the market. They are all the blockbuster drugs. So if you in this figure, if you see the blue means the drug which will work in that patient, red means it does not work. For example, the first one is here. I'm trying to show the aripiprazole, which is used in the treatment for the schizophrenia. And 1 in 5 patients only it will work. 20% success. Esomeprazole, probably every pharmacist will see that, omeprazole or pantoprazole in every prescription. It's 1 in 20 chance that drug will work for the patient. Similarly, rosuvastatin, I can, keep listing many drugs here. So what I'm trying to say that in this model, what they do is they give it is trial and error method. So you don't know whether drugs works or not. So the failure is very high. Now, what is the consequences? This is very important. So this is the article published in the Wall Street Journal about the drugs which is used in the Medicare Part D. This is basically for retirees in the United States. Based on the data, 80% of the money is just wasted. Drugs don't work 80% of the time, so you lose. So in this case, they show around $15 billion Medicare is just wasting. Because those drugs, anyway, you are prescribing to the patient, and they will take or not sometime, because if it doesn't work, patient will go into non-compliance. You may not adhere to the medications. So this is very important in terms of pharmacy, what I'm talking about. Why there is a adherence is not there, non-compliance is there, because one of the reasons is drugs may not be working to that particular patient. That's what we call, when we go to, clothes shop, we'll have different type of dresses, small, medium, XL, double XL, etc. But in the case of drugs, it's very different. So patients will respond to, differently to different drugs. So that's what we call the problem. Drugs don't always work. For example, if you look at here, antidepressant 38% failure. Whatever the existing drugs, if you give to the patient, 4 in 10 the drugs won't work. And then anti-asthma drugs, diabetic drugs 43% failure, arthritis drugs 50% failure, cancer drugs you all know very difficult disease to treat. And by the way, I'm not telling the drugs are false here. Actually, target is not the right. So we don't know whether drugs works or not. So in the cancer treatment, yes, 3/4 of the patient don't get any benefit because of the drugs. So that is why the way to go, one of the key area where people are working now in the cancer research or cancer treatment is the precision medicine, or we call precision oncology. So we founded a global cancer consortium based upon that. Around 9 institutes from India and United States, try to sequence our, genome also, cancer genome, how exactly work in India. They have done a lot of work in the other part of the world, and eventually, try to go to the precision therapy. And again, quite honestly, 95% of the, 90,000s of diseases we have, but we have very few drugs, quite honestly. If you go and list the, different type of diseases, for example, cancer itself, there are several stages, several types. Now, for example, in the lung cancer, non-small lung cancer, small lung cancer, adenocarcinoma, squamous cell carcinoma. So they need a different treatment because of the genetic mutations. So we have very few, drugs are available. So that is why I always, when I go and put up this slide, is translation science or research. So that's what we call bench to bedside, and you develop a drug, go to the bedside, see if it works or not. If it doesn't work, we come back to bench and find out why these drugs are not working. So that way, more research and more science could be done. That way, you can treat the patients. So this is the, in a very separate note, I want to say that most of the FDA approved drugs in India are not done much of the trials in our population. Before 2018, there was no mandate for how to do that. CDSCO came in 2019, the new, Clinical Trial Act in India. Before that, we did not have clinical trials in our patients. These are all drugs are, invented and manufactured in other part of the world, and manufactured maybe in India now, as previous speaker said, 30% of the drugs are manufactured here. But these drugs are not, tailored for us. These drugs are invented for them, and then all these drugs are marketed in our country and other countries too. So will there be a difference in the drug response? Remember, when these drugs given, they do a clinical trial, there is a system of doing phase 1, phase 2, phase 3, phase 4 trial, and they monitor the patient whether these drugs works or not. Of course, I think this is my personal belief. I think yes, mostly the reason is, if you go to West, you know what the diet they take. They have a different diet, they have a different lifestyle, and they have different seasons of, for example, they have spring, autumn, winter, summer. But and different genetic makeup too. But when you come to India, you have a different kind of food. We take very different food, and we do different lifestyle, and we are maybe having a different genetic environment. The reason why I'm telling you, diet and lifestyle is important for having all these diseases like hypertension, diabetes, etc. So my personal belief is, I think these drugs sometimes may not work us because of one of the reasons maybe, we have a different diet and lifestyle, and also different genetic makeup and environment. So with that, what I'm trying to say is that many people ask me this question, we don't have the regular, primary and secondary or tertiary healthcare system, how the precision medicine will help. But we were practicing hundreds and thousands of years ago. You go to Ayurveda system, they follow the same thing. Enough funding, you go to the Siddha physician, you won't, take 10 people at the same, scale. They will tell you, you're enough fund, they'll check you and they'll give the treatment. I think it was there for a long time, and I think it is a way to go to get a better treatment to the patients, and particularly difficult diseases such as cancer and other diseases. So I just want to connect this big data into precision medicine in the way that how big data will help to, solve one of the, or maybe easier to have a precision medicine for the patient. I think you all know, I think first speaker was talking about, large amount of different types of data produced from various types of sources such as people, medicine, of sensors. And you get this big data from the different sources. It's not necessarily in one system. I think Vice Chancellor talking about how Google Maps and Google things are generated is basically day-to-day life. In the healthcare also, I think big data is very important. And I'm not going to talk much about this. There are 5 V's of data, big data, like value, variety, velocity, veracity, etc. And these are all the different types of data required to collect, get the big data so that you can implement in the system. So, and again, this slide was shown by the first speaker. Data never sleeps. We are, as all in the social media, every second you speak, like WhatsApp, maybe you keep texting, keep sending the pictures, like in YouTube, Google, all those websites are already there in the system where you can get the data continuously in the system. And if you look at in the last 5 years, the global internet population is 35% more than 2016. Now, around 5.5 billion people using the internet system. So to collect the data from various systems, it is quite easy in terms of social, very easy. But in the healthcare, a lot of ethics is involved, especially if you want to get the genetics data from the patients. Ethics is the most important. You need to follow the country's rules and regulations, etc. And in the pharma chain, now big data is playing a very important role. I think you are maybe knowing there are different perspectives in the pharma chain. So they target like patients, where you get the signals about data analysis from the patients, from the drugs, from the business and innovation. So if you see this, success plot where, patients, you can get the data whether the drug will work them or not. Even you get the data now, there is a system they have done, whether there is a drug rating is there now, whether this drug is better than the other drug. So this all, kind of, I'm not explaining whole stuff. So there are websites or there are big data portal systems where you can collect the data at one point and then you can do the analysis. So in the case of genomics, what we call phenomics. Phenomics means pan-omics, all kind of data which is derived from the omics, like genomics, transcriptomics, where you measure the RNA, in the genomics you measure the DNA mutation, proteomics, where you measure the proteins, metabolomics, epigenomics, genomics, etc. So all these multi-omic profiling is done through the big data system, and then you can integrate those data into the healthcare analysis, where now again the health records are becoming electronic. Most of the Western countries is already electronic, but even in the India also, the EHR, medical information and patient health information, when you integrate all this data, you get the pool of data from the different patients from all over the world. Then you can stratify and see what exactly happening on those patients. Then eventually you can do this precision therapy to the patients. So these are the publications. There's a lot of interest in the big data. It is exponentially increasing year by year. I think one of the speakers, one of the guests today was talking about how we are moving. I think we are there in the millions and billions data collection systems. And what are these applications in terms of healthcare? It is that one can understand the genetic, diet, and lifestyle of the patients or the other normal healthy individuals also, and you can do based on that personalized care to the patient. And in the medical research, reduction of adverse medication, and particularly pharmacy, you can get all this data. You can see what are the different drugs process area. WHO already have a lot of websites now. They want to integrate into the global level, and that eventually, reduce the cost of the medication. And that's a very important point actually when you talk about our country, and you can have a better health to the people. So with that, when I came to India, I wanted to start a big data in the tuberculosis. So when I saw the patient data, it is very flawed. Means you go to a medical record and we cannot find a clean and neat data. Remember, if you want to develop any kind of artificial intelligence system or better medications or better health treatment, you should have to have a good source data from the patients. And you know that we have a lot of this particular disease is stigmatized and very difficult to ask some of the questions to the patients. They may not answer. For example, if you ask them, do you have a tuberculosis? They are still stigmatized, unfortunately. Then if you ask, do you drink alcohol? No, they won't answer. What is your salary? They won't answer. So it's very difficult. When you have flawed data, very difficult to develop a system in the way where the people are talking about a lot of artificial intelligence and big data, etc. But it is not like Google Maps. Somebody has to give the answer to you, which should be correct as best as possible. So that is the one issue we think, and even WHO says you need to work together and make a strong data system. So that is how we thought. We went to clinicians. Why can't we start a project which is basically funded by National Institute of Health from the United States, and This is a system called, TB portal ecosystem. So I'm the one principal investigator for India. We are the only institute that does this. So they fund this entire program. And this is an open web-based platform to facilitate meta-domain and domain-specific TB data exploration and analysis. Around 21 countries are participating in this particular program, including United States, Canada, China, and most of the East European countries, because they have, multiple drug resistance tuberculosis patients. And this type of TB portal data contains clinical, bacterial, genomic, and medical imaging data from TB patient cases. So you are all comprehensively, you are collecting the data from the patient in here. So what we do here is, we go to individual patient and we ask, there is a, I think, interest of time, I may not be, if I have a time, I'll show that website, how exactly we are doing. You can go to, website is written in there, you can go there and you can check how we are collecting the data. Remember, if you want a good outcome, for the data is the most important. So many people, EHR is very common in the United States, but in India, we are still not yet there. So doing this particular kind of a research, very challenging. But we recruited 800 patients in this particular program. And you can visit that website, written in the video, or I can show you that. So this is about 9,000 patients, from all over the world. And we have in the back end another 10,000 patient data, approximately 800 plus patients from our site. So here in this particular browser, you can see on the top, data.tbportal.nia.gov. If you go there, all our data will be there. And it's open access. Anybody can go and check that and see. So what we do here is we collect all socio-demographic data, and we take the patient sample, sputum sample. We do the microbacterial tuber genome. So I think I'm going to publish that paper in American Journal of Microbiology. So we already paper got accepted. So we have around 300 plus patient whole genomic data, and then we are comparing this data to other parts of the world. There are a lot of countries have a different types of strains. So it's very interesting if you look at every country has a very different diet and lifestyle. The outcome what we see in India is different than Moldova, different than Georgia, different than the China. So that is a very interesting part. So you can browse through the website. There are a lot of, even you can play different cohort. You can check which country is doing better in the INH treatment, or even individual drug you can go and check, or ATT drug treatment. So that is one of the paper we are going to publish that. So there are a lot of public, these are all the public data TB portal tools. You go there and you check and you can find, and even we integrated AI into this big data system. So we have derived a AI software where it will distinguish healthy lung versus the tuberculosis lung, 95% accuracy. You put the X-ray, you upload it, it will tell TB yes or no. This is the AI software we have developed. So you can check through this system. In interest of time, I may not be explaining all of them. Like we have a depot where data exploration portal, all the data is available in here. Gap where the genomic analysis portal, you can compare different codes from the different parts of the world. RAP is a radiomics analysis portal. You can see the chest, how the X-ray looks like from one country to other country, and you can see the CT, the computer tomography when we take from the patient, how it looks like. So they developed a lot of software where you can upload your X-ray, you can find out within no time whether this patient TB or not. Second thing is, you can go and when you put up this one X-ray, it will go and scan all the 18,000 samples, and it will tell which is the closest X-ray to that, and it will classify what is the chance of outcome, whether cured or maybe patient have MD or XD. So this is the one, the 3D, you can see in that website, the baseline, relapse, and 1 month treatment. So you can, in the 3D, you can see the lung, how exactly looks like. So I just, this is the one big data analytic precision therapy tuberculosis we have started in Manipur, and our ultimate aim is to, go holistically, see the host genome, microbiome genome, comorbidity. Even we have put drug-drug interaction, the new thing, because a lot of patients in tuberculosis patients in India, we found that 40% are diabetic. It's a very significant. So there is a lot of possibility of drug-drug interaction, and eventually we want to get into the precision therapy. And these are not only in the tuberculosis. We have started another CBio portal, which is done by US, but Cancer Consortium looks into this. So these are different databases where are there where they might implicated in the precision medicine. And again, big data is important in the pharmacy services also. I leave it. I think the first speaker explained much of that. And EHR is dramatically changing in India now, and not only India, all over the world. So there is a possibility of integrating the EHR data into the other systems. And again, a lot of money spent in the big data and AI. Now every companies want to get the most of the data out of this. And you guys know that ChatGPT what is doing now. Even top will tell what we have to do. So this is another new thing. And time is money. You need a lot of time to save to do the research. So by big data, I think 1/3 of time is saved and the cost is saved. It is a patient affordability will be good. And again, this is a slide again. We all talked about evidence-based medicine. It is not that easy now because of this big data, AI, and genomic stuff. So dramatic, shift is going on. We had a triangle shape. Now it's all getting an iceberg where evidence-based medicine based on the group of population may not be working. So clinical trials are dramatically going to change in the future. So to conclude, in pharmaceutical product development, what drugs we have invented in the abroad may not be suitable as for us. This is my thoughts for the most of the patients. But in the future, having a big data, I think we could give a better precision medicine to the patients. Thank you very much. I think lack of time, I'm not able to show that, but still I'm happy to interact with you. If any questions, then you can email me. Thank you. Any questions?
Thank you, sir, for a wonderful session. I request Mr. Rudra Bhanu Sarpati, sir, to felicitate Mr. Dr. Mahadev Rao. I request Judy, sir, to join. For the third keynote session of the day, we have Dr. Umesh Banakar, Professor and President, Banakar Consulting Services, Westfield, Indiana, USA. Founder, Goa Center for Excellence in Intellectual Property, India. I request you, sir, to conduct a session on invention to market and commerce, role of IPR.
Slides.
Good afternoon. It's a very precarious position because I'm standing between lunch and dinner. So, but nonetheless, instead of my entire 45 minutes, I take 44. You get at least 1 minute extra. Okay. I'm very happy to be here. Thank you for inviting me. I always cherish coming to Adichunchanagiri University. Very nice campus. A lot of things to do inside the university rather than outside. It's something that is very few places we have. Okay. The theme of our conference, and if you look at the title, two words are very critical: innovation and radical. Okay. So we are going to talk about a little bit about what happens or what is the logical next step after innovation. Because you have a lot of ideas, you have a lot of innovations that you are going to hear about.
Oh, here you can.
And there is always this question as to, well, what do we do next? That next part is going to be something that we will focus on for the next 30 minutes or so. Okay. And then the last part of the presentation is going to be, I'll give you a little bit of 5 to 7 minutes on what we do at our center for excellence in IP. So those of us who are here from outside, they take the message back to their colleges or universities or workplaces that would be useful. First and foremost, what we are talking about is intellectual property. Intellectual property is essentially you have your intelligence and then you apply that intelligence. When you apply the intelligence, you create something, and that creation is intellectual property. We are in the business of creating intellectual property, so we are the ones. So no mistake about that. If you look at IP as such, intellectual property refers to creations of the mind, which are in various categories. The most tangible category is patents, trademarks, copyrights, trade secrets, geographical indications. But patent is the one which is most tangible, for which we need to start to look into that, particularly in India, because we have not yet recognized the full potential of IPR. I just got stuck. Out and come back in. Okay. So what is a patent? Patent is an official document, which is a legal document giving exclusive rights for a term of 20 years from the date of filing. Exclusive right to the inventor to make, use, or sell his or her invention. So the key aspect of intellectual property, or so-called patent, is invention. And invention is what you innovate. We talked about that a little bit this morning. Innovation is what you know and you apply what you know and then you create something new. Okay, so that is innovation in very simple terms. And that innovation, if you are able to get a patent on that, then that is known as the rights that you get, which is IPR. So now the key important aspect is our innovations. We would like them to be inventions, okay? And that's what we should strive. Why? Because if you look into the patent itself for half a minute or so, the entire domain of IPR is broken down into getting a patent, what to do with it, and protecting your rights for 20 years. Getting a patent is prosecution of your invention in order to get a patent, which gives you exclusive rights. In those 20 years, what you do with it, people have talked about today, this morning, that well, 12 years are spent in going forward and then getting it to the consumer or market, so on and so forth. So we are going to talk about that, which is a logical next step. And the last one is in those 20 years, how we protect it, not for today. The other category in case of IPR is incremental innovation. There was a mention of that this morning. Second is how to commercialize it. Before commercializing, you have to take it to the market first, and that difference will bring it out because there's a big confusion in India about taking your invention to market, which is many times misunderstood as commercializing it. Then policies, procedures, IP data, we just heard about that, big data, IP data, intelligence, competitive intelligence, and IP intelligence. Get stuck again. What is the current scenario? This is the current scenario in India. Patent le lo paisa banao, patent le lo paisa banao, patent le lo paisa banao. There is a lot of difficulty in understanding how to get a patent first, let alone how to make money out of it. How do we see that? We have too many inventions. We have too many inventions. We have to do a lot of innovations. Very few or many of those get patented as well because of this slogan, patent le lo, patent le lo, patent le lo. And far too few are commercialized or taken to the market. Far too few, meaning you can really count them on your fingers. The scenario is the reverse in most of the Western countries, where yes, there are too many inventions, there are many patents, but there are quite a few products that come to the market. And our pharma industry, which is big, is no new to that because we work with copying those or so-called becoming the generic versions of them. We still have to produce one first single innovation going all the way through. So we need a lot of learning to do, but make no mistake that yes, next step to innovation is taking it to the market and commercializing it. Why? Because if you look at the top 20, 25 brands in the world, first is Apple, second is Amazon. We'll not spend time on that, but where are we? We are not there at all. We would like us to be there somewhere, sometime, very soon. We want to become $1 trillion economy, $5 trillion economy. All of those things are good things to talk about, but we have to take steps to that. Even US is not number one in top 25. China is number one. US is number two, and then the other countries. Along with this shocking emoji, the other emoji is, what do we do with it? That gives us the idea to start working towards getting better or trying to get into the so-called big league. If you really want to make it big, and if you want to be one of the top, among the top in pharma, forget about any other discipline. Why? Because the road to sustainable prosperity goes through IPR. And in our case, the prosperity is many times misunderstood as just making money. Prosperity, making money is the last in the list. So we have to start looking at how do we enrich ourselves. When you go from first year pharmacy to second year pharmacy, you are educationally prosperous. When you go from a scooter to a four-wheeler, you are economically prosperous. When you are going from master's to PhD, you are advanced in that prosperity. So get out of the mindset that prosperity means making money. Okay. So now let's see that, we have innovations, and we are talking about innovations today and tomorrow. And now what is the thing that is radical that we need to understand? First is, what is the next logical step? And innovation has given us a proof of concept that we have something new in our hand which is working. We have solved a problem. And we need to take it to the consumer or the end user. And our end user is patient or the doctor or the healthcare insurance company or the nursing homes or whichever our target is. We'll take it there. Okay. So now we have made an invention or we have an innovation in our hand which is at a level which is prototype. There are so many nanoparticle projects that are going on in India with natural products. So many of them do get filed for the patent. So many of them get PhDs and masters with it. But now we have to look beyond that and taking them to the market. So the genesis is, if we have an invention, we get a patent. From there, we need to take it to the market, and then we'll need to commercialize. These are two different things. So now let's get into the radical approach that I'm going to present in the next 20 minutes. Number one is, I'm not going to talk to you, but I'm going to ask questions and then answer them, so that's how that should go, question and answer, as opposed to, oh, you do this, you do that, you do that, you do that, because I'm the one who can tell you, I'm the one from whom you can get that direction. That is fine, I can help you, facilitate you, but really, the questions that need to be asked, regardless of your innovation, that's what we will focus on. That way, it will give you an idea. When you're constantly innovating, you will start to ask these questions, which will help you facilitate taking it to the next level. Invention to market and invention to commerce are two different things. Okay. First is, you have a product in your hand at a prototype level, then you develop it. What is your batch size? Simple question. What is your batch size when you develop a formulation? 1,000 tablets, 50, 100 capsules, so on and so forth. That is your PDR, product development report. Now from there, you go to regulatory approval, which is from 100 tablet batch to 100,000 tablet batch. 3 of those, do stability, do your clinical trials, then you file your data to regulatory agencies, whichever FDA, and then get it clear. That's taking it to the market, regulatory approval. That takes time, that takes money, that takes resources. From there on, still you have not sold a single one. Regulatory approved products will then have to be taken to the consumer, which is the next step, which is marketing, sales, so on and so forth, creating markets, so reaching the appropriate targets, and then selling it. That's when you start to so-called make money. So that process is the next step. So invention to market and market to convert to commerce are two independent steps, which are time-consuming plus resource intensive, all types of resources, and very much intensive in terms of technical inputs. Okay. All right, let's start with question and answers. That is the business that we are in. We are going to take our innovations to market and to commerce. Some of the questions and very brief answers I will provide you, because these are the questions you should ask. Number one, describe your innovation. We need to know what our innovation is. Okay, that's number one. First thing, you need to know what your innovation is, combined with, if you are the inventee or inventor, the patentee should make sure that what has originally filed in the patent application adequately defines the full parameters of the invention. If you don't know our product, we are in trouble. We have a general idea of our product, we are in trouble. Okay? We are talking about individualized medicine. I'm talking about just getting a product defined. That is the first step. Person of ordinary skill in the art should be able to reproduce your claimed invention based on the specifications and written description along with examples. That's what your entire business is in your PhD work or your master's work, which you need to know which of those product or formulations you are going to take it further. Where does your innovation start and where does your innovation end? You cannot have an innovation which has no starting point and no end point. Okay. So you need to know where your innovation starts and where your innovation ends. Okay, so that's very critical. which most of us forget because we think that our product is useful for everything and anywhere and everywhere, in every patient, in every condition, in every disease state, all of that. It is not. Okay? So we need to know that. So when we are going to the market, we know exactly where, whom, why, and how we are going to reach the end point. So scope and limit of our invention, we need to know where does it start and where does it end. Sufficiency in details provided in written description and specification should be there before we start to make these lamp-blasting statements that, well, this is my product and this is what it will do and this is my innovation and that is what it is going to be used for. We need to know this. What is the version of your product? When you are in the lab, when you are completed your PhD work, when you have completed your filing of application, you have even got a patent. What is the version of your invention? Your version of your invention is a prototype. Is a prototype. What is required for securing a patent? That's enough. Does it have to be stable? No. Does it have to be effective? Possibly may not be required because you're not looking for a therapeutic new indication. Is it useful? Yes. In India, there are only 3 requirements for getting a patent for a product. Number 1 is novelty. It has to be new something. Number 2 is inventive step, which is obviousness. This is not a platform for that. And then an industrial use or application. That's all you need. So you can have a 100 capsule formulation or a half a liter suspension or an injectable, a lyophilized 50 vials. Good, you got an invention. Now you have to take it to the market. For getting an exclusive right for those 20 years, that is enough. The patent to मिल गया, now the पैसा बनाना है. Invention to market is nothing but getting it through the regulatory approval process. Our generic companies, when they go outside, as well as coming into the Indian scenario, we have to get that prototype or so-called product in development through all the steps, stability, efficacy, determination, and then getting it submitted, all the data to any drug regulatory agency and getting it cleared. So all of our innovations that we are going to get in treatments with new products, new formulations, new approaches, fixed dose combinations, etc., ultimately have to get through regulatory agency. Once that is approved, then that is your market, taking your invention to market. That takes at least 2 years if it is generic. It takes at least 7 years if it is a new molecule, new treatment first time. Today, we do not know about individualized medicine, how that is going to be worked out in terms of regulatory approval, let alone taking it to individual. What we are working on most of these innovations in radical approaches, AI and all, is what we know. We are trying to use that background information which is stored in big data and trying to apply that in various ways that are secretive as opposed to going to the library. So that's a big step ahead, but still we have a long ways to go. Can a regulatory agency approve individualized doses?
So we can start with the.
Is it ethical to do that? Those questions come up, so we have to be aware of that. But nonetheless, regulatory clearance is the end point of taking invention to market. Invention to commerce. Today you have a new way of treating. You have a new mechanism that you have come up with, which is not in AI. Because AI, keep in mind that AI has known information which is being put to use for your benefit. AI does not create new information. What it does is, whatever is there, it will give you options. Yes, it will give you a less number of possibilities, so your chances are getting better. But today, if you come up with a new way to treat a tumor, mechanism of action, that will be the first point in getting into data. And then if multiple things happen, then it will start to generate its work. But right now, we have nothing in that. So either way, once you have that innovation, regulatory approval is there. Now your next step is taking it to the end user, which is the consumer or the commerce part. Who is your target? Yes, you can have in AI built into it, your how to get to the stakeholders, how to get to what are your targets are. That will help you rather than doing a big market survey and sales survey and have sales people that gathering data and then using that to build up a program. Today, if you have a new way to approach treatment, again, it is something new which needs totally from zero level up. But nonetheless, again, it is a time consuming, resource intensive step. Innovation to market to commerce. So innovation to commerce is not the same as innovation to market. I hope that gets cleared up. We need to evaluate what our innovations are, invention are. Your competition will be there, your IP data will help you, so your IP audit and valuation has to be done. So that probably will come in handy. We don't want another Me-Too product and then fail on the market, getting approval but could not sell. All these time-consuming two processes, steps have to be analyzed for cost. And who is going to bear the cost? If we as an inventor or a researcher or so-called IP generator, we don't have that resources. So we always look at somebody to invest in us. We do that. So validity, viability and decision. 5 inventions in ACU. Which one I'm going to take forward, even if I have 50 crores to take it. So those are the things that we have to answer. Every single one is very important, but which one we have to take? So that all made. So once that happens, then you have something to look forward to. The analysis have to be done. Some of AI can have, we can take benefit from there. For that, we need investment from others. Shark Tank is something that people got excited in India because it was something that is done in US. It is now coming in here. This is where Shark Tank come in. You present your work and let's see if they can invest in it, because you can present your work and its utility potential, etc., etc., etc. The first question they ask is, do you have a protection on it? If you are not protected by IP, your innovation, there will be 90+% investors will lose interest. There are startups here, there are ICIAPS here in India. I have been asked to many times over the last 7 years to evaluate them. And that's where the difficulty is. We need to get IP on that. But nonetheless, from research stage, which is your patent stage, to market, you need an R&D agreement, whoever comes in with you or invests with you. But you need to know that you have to protect your IP. Second is a commercial agreement. You get regulatory approval. You want to take it to the commerce. That is another agreement, which is known as a commercial agreement. Who will get what? For their policies are required when you start to make crores and crores. How to handle that? Okay, there have to be policies. Today, let's say the first product from ACU gets to commerce, and now there is a windfall of 50 crores per month. How is that money going to be used up? How distributed? Who gets what? Inventor gets what? Department gets what? College gets what? Who gets what? So all those things have to be in the commercial agreement. The whole process of taking your innovation to the market and then to the commerce is customarily known as tech transfer. People talk about tech transfer so loosely. Oh, what are you doing? I'm doing tech transfer. This is the whole process. So it is not easy. Market version, commercial version, and the claimed invention, there has to be a link. Patent to mil gaya. We have to know our scope and content, scope and limit of our invention. Where does it start and where does it end? Why? Because when you go from your patent stage to market stage, 3 lots you have made with 100,000 units as an example. Every single unit has to be within the limit and scope of your invention that you have got as a patent. When you take it to commerce, then you are going to make millions of lots, batches of million units. Every single unit in that million has to be within the claim. Otherwise, that product is not covered by a patent. Your competition will come in and trash you. Okay. So that's the role of IPR and importance of taking innovation to the next stage. So this is a very critical slide which will summarize what I was talking about. Your research or innovation gave you invention. That invention is your patent, which is your claim. That invention, now you want to take it to the market and then to commerce. That is all tech transfer. This is where we have to ensure that the IP is not only maintained, protected throughout that process, as well as taking it to commerce. Because this process is time consuming, resource intensive, you need startup investment, you need entrepreneurs. All of them are not interested in your product, they are interested in their investment. So you have to not only protect your IPR, but also make sure that that investment will lead to the appropriate outcomes. So we need to have policy, procedure, protection throughout this journey. So that basically is something that I want you to start thinking about is innovation is okay, but what to do next? Okay. One thought is take a patent. Okay, patent to mil gaya. What to do next with that? Because worldwide IPR is important. Last 5 minutes on what we do. Current scenario in India is we have yet to recognize the full potential of IPR. Prosecution, preservation, and protection. Because there is lack of motivation, lack of policy procedures, publish and perish, it has to be converted to patent and prosper, or although prosper has to be not just money. Inventor should be the focus, which is you folks, researchers, students, research faculty. Syllabus talks about basics of IPR. Nobody talks about what the inventor should do with his or her work. So we need to integrate IP. In 2016, that was our motivation, where we set up the center, self-finance, non-profit center. Okay. In 2016, we set that up with. We are the first ones in the country, and we are also the first ones who are registered under Societies Act, so nobody can take the name. So we are the first ones in the world as well. Vision is to recognize and protect scientific discovery with the rights it deserves, and we provide professional services at highest level to scientific discovery through IPR globally, not just India, to globally. Our objectives are advanced programs that we have, academic advancement programs, which will help us understand this IP, this whole domain. 6 courses we have. Second objective is very important for all of you folks. Every year in November, we have an international conference on IPR, where subject matter experts come from the world and speak. More important is, along with it, there is a research showcase presentation. It provides a platform for presenting your research work and get it evaluated for IP potential, your innovation converted to invention. And number 3 is we also recognize 3 most likely to have highest IP potential. show research showcase presentations, and we recognize them, we award them. Where we have developed the 6 courses going from basics all the way to how to write a claim, taking the invention to market, commerce, techno-legal considerations, how to protect your invention, and going beyond that, strategies, so on and so forth. Whoever completes these 6 programs, they will get a postgraduate advanced training certificate in IPR. You will be the hot potato for industry. Okay. We have a elective also, a 5-day elective. In summer we offer where we have a comprehensive understanding of the entire IPR just for our scientists who want to check it out if IPR is what you want to do further. In 2021, we have launched a 5-year dedicated campaign to enhance the knowledge base and awareness of IP to the scientific communities across disciplines, across state boundaries in the entire country, not just 1 or 2 conferences in the entire country, with the sole objective to induce spirit of inquiry in translating theoretical science into tangible products, is basically IP. Where we are, so far we have covered access 14 states, 1 union territory. These are the states. We have covered from Kargil to Trivandrum, from Gujarat to West Bengal. So we are translating it into 30 plus universities, 14 disciplines, over 110 presentations at no cost to the institution. No cost to the institution. We go there and we do these presentations to aware. So far we have touched over 17,000 individuals to spread the word that well, IP is, awareness of IP is required. 2016 we started it, 2020 Indian government has introduced the NEP, which requires IP in their curriculum, across curriculum in all universities. 2021 they have introduced Kapila program, which is talking about training in IPR, but they only talk about law. So if you look at the chronology, you can get the picture. Last 2 slides, please. We have over 25 MOUs. Where do we wish to be? We wish to cover all 28 states by 2025, which will cover over 1,500 institutions, over 2,000 presentations at no cost to the institutions. We will need your support and well wishes. We are ready to help. Thank you very much. Any questions? I'll be more than happy to. I know it is lunchtime. My email address there is, by the way, we have a website now. We have launched www.gceip.org. www.gceip.org. If you take a look at it, you'll see what we do and how it will help you take your innovations further. Yeah. Question is on an institution based, how we can help in terms of filing IPR. 2 or 3 ways we can do it. One is these MOUs help where we do training. Plus, we then help them identify their invention from their work, and then we help them file also.
It starts from searching the file. We provide also.
That is very small part of it. It starts from identifying and training the people, because most of these institutions we would like to have the faculty also involved in this. So then they can take the message forward to the next set of students. So I need to talk to you about a lot of stuff. Okay, There is innovative lunch today, which is what is known as have you ever thought about a tablet bath? You take a tablet and you get the bath. So in this case, you just breathe and you have your stomach is full. That is our innovative way of having lunch. Okay. Thank you again. Thank you all.
Thank you, sir. Please accept our thanks for a great session. I request Mr. Rudrabanu Satpathy and Dr. Prakash Gowdanavar to honor Dr. Umesh Padak. Now we have lunch break. I request the dignitaries and delegates to proceed to the college quadrangle and savor the same. All the oral and poster presenters are requested to go to the allotted seminar hall and hand over their PPTs to the respective coordinators. Go to your allotted seminar halls and give your PPTs to your coordinators. Hope to see you guys all at 2 o'clock. We also have an exciting session session in the afternoon from Malaysia. So we hope to see you guys at 2:15. 2:15. Thank you.
Last one. Sir. Money. Yes, sir. Next speaker. ಇಲ್ಲ ಅಲ್ಲಿ ಕಲೆಕ್ಟ್ ಒಳಗಡೆ ಸ್ಟ್ರೈಟ್ ಹೋದ್ರೆ ಅಲ್ಲಿ ಪಾರ್ಕ್ ಐತೆ ಸೆಂಟ್ರ್. ಎಲ್ಲೇನೋ ಒಂದು ಐಟಮ್ ಇದಾಗಲ್ಲ.
ಕ್ಯಾಮ್ರಾಗಳು 8 ಕ್ಯಾಮ್ರಾ ನೋಡಿಲ್ಲ, 1 2 3 4 12 13 ಕ್ಯಾಮ್ರಾ. ಲಾಸ್ಟ್ ಎಲ್ಲಿ ನಾನು ಇಲ್ಲ ಮುಂದಿದೆ ನಿಮಗೆ ಸ್ಕೂಲ್ ಚಾನಲ್ ಸಾರಿ ಕಾಲೇಜ್ ಯಾವುದು ಯೂಟ್ಯೂಬ್ ಚಾನಲ್ ನೇಮ್ ಏನು ಯಾವ್ದು ಲೈವ್ ಬಯೋಲಿಕ್ಸ್ ಅಯ್ಯೋ ನೀವು BI
Yes, ma'am.
Hello. Hello.
On the 8 o'clock. ಗೊತ್ತಿಲ್ಲ ಕೆಲಸ ಮಾಡ್ಬೇಕು ನಾನು ಈಗ 4 ಗಂಟೆ ಕೆಲಸ ಮಾಡ್ತೀನಿ.
Hello.
Good afternoon, everyone.
Hello. 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 Am I audible? Hello. Hello.
Good afternoon.
Good afternoon.
Yeah,
Can you,
I'm Miss Aparna. Hello.
yeah, can you help me to join room A? I'm not able to join. I'm coming out once I'm trying there.
Yeah, one second.
Yeah. Hello. 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 Hello.
Hello? Hello? Anyone is there? Hello? 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 No, kidding, forward. भेजो तो फिर व्हाट्सएप करो जल्दी से ये वाला लिंक करो उसका ज्वाइन करो जो मैंने भेजा है व्हाट्सएप पे व्हाट्सएप करो ये लिख करो ये लिख करो
Hello. Hello. Hello.
अब आवाज तो नहीं आ रही मेरी?
Color change.
Says, go. Color.
Good afternoon all. Now for keynote 4, we have Mr. Maniam Arul Pandian, Manager, Formulation Development, R&D and CMO, Northern Pharma, Petalin Jaya, Selangor, Malaysia.
Sir is here to educate us on the emerge of 3D printing technology in pharmaceuticals. I request sir to take over the session.
Remove and keep cable.
Thank you. Am I audible?
ಫಸ್ಟ್ ಎಲ್ಲ ಕಡೆ ಹೊಸ ಕೇಬಲ್ ತರ್ಸಾಕ್ಸಿ
ಮಸ್ತ ಇದು
ಕೊನೆ ಮೂಮೆಂಟ್ ಅಲ್ಲಿ ಅದು ಬೆಂಡ್ ಆಗಿದೆ ಇದು ಬೇಡ.
ಅದು ಹಳೇದದು ಸ್ಟಾರ್ಟಿಂಗ್ ಅಲ್ಲೇ ಹಾಕ್ಸಿಲ್ಲ.
Hello.
So virtual, right? Is online now. What's good? There is something.
First of all, thank you for giving me an opportunity to present my livable technology throughout my career. I have joined a formulation development department in the year of 2010, but this emerge of technology of 3D printing has been evolved in the pharmaceutical from 2015 and 2016. So from that point, this technology has been close to my heart, and I am very proud that I can present my technology to all over the participant over here. So shall I continue?
Yes, sir.
Okay, thanks. So 3D, the topic what I selected, 3D printing technology in the pharma, it can be new, but this technology has been evolved in the other industries from late 80s. So the disclosure of this presentation is that whatever whatever things that I'm going to share is completely a non-profitable, and the information which I have gathered is based on my previous knowledge, books, and the internet from the internet, which is solely for the non-profitable and educational purpose only. It is not related to my present or the last organization. So as the I would like to start with the thought from the Daniel H. Pink that the beginnings stay with us far longer than we know. Their effect linger to the end. So as we know that whatever beginning that we have together in the pharmaceutical industry, which is in the traditional way, that is staying longer, and to adopt a change that may take some time. So what you can expect from this presentation is introduction of 3D printing technology, history, why 3D printing technology is needed, what is the principle involved in the 3D printing technology, what are the classifications, how does the machine works, what is the advantage and disadvantages, and what is the future in the prospects of 3D printing technology, which is another term called as an additive technology. So as we know the history of tableting, to manufacture any unit dosage form, we started with tablet, and this we know that from BC 140 we know in the form of pill, where it's been given in the dosage form. In a traditional way, how we develop our formulation in terms of solid dosage oral form is nothing but we have an API, excipient or a diluent, which we will be incorporated into a different technology or a different process, which may be a dry granulation, wet granulation, direct compression, or any other common processing step where it involves an n number of processing technology in between, then we will be taking out for the tablet compression, which gives a final pill that lead to an patient patient compliance. Now, when we take it, before taking it into an compression, it has to go through and different processes. To adopt that processes, we may require some time for the development, and there are limited technology or the limited resources where the industry carry, where you can take it up the whole processes. Now, what all the things that we normally do, all sort of compression modes or the forms and the different dosage form require different machines to evaluate. We have a robots in manufacturing. We have an analytical tool for the manufacturing, and now in a traditional way, we able to manufacture a complex pill within the pill, bilayer, tri-layer, and we have evolved to a multi-layer polypills also. Now we are in a stage where we have a different dosage form where it can be adopted for patients in the buccal, vaginal, osmotic pumps. Now different extended release, I can say the modified release dosage forms are available. So that's how the whole solid dosage form has evolved. Now, what is evolving in the advanced pharmaceutical industry is that the evolution of modern diagnostics test has been evolved. Now, the new concept has been developed where the patients require a patient-centric dosage forms, where, for an example, gene therapy, DNA or RNA-based therapeutics, and a personalized medicine. Now we know that the industries has a different kind of patients, races across the different global condition. Now, what we are doing traditionally, we have only one pills or one dosage form where it has been given to the different people where have their own complexity of having it, or a person which has different races has different complications. Now, what is the advantage that we will be getting with this technology is more towards the patient-centric treatment. Now, how this evolution has happened, 3D printing has given a flexibility to adopt into that. It is adopted in the food and auto industries in earlier than the pharma. Why? Because in the food industry and auto industry, you have to develop your product or an machine part in a large volume. When you want to produce that in a large volume, you require a mass scale of production units, where the requirement can be as per the market expectation or cannot be. So when you have a mass production, to absorb in the market may take time and it requires a huge investment. But when you have additive manufacturing or 3D printing technology, you can produce your things as per the requirement, because it gives a flexibility to have a one part production, or it can be multiple part production, and whatever the flexibility that you want to put it into in your outcome. But we have adopted in pharma from 1996. And from 1996, when we started observing it, now we have a one dosage form which is available in the 3D printing in the market. Now, if you see the history of this, as I said, the earlier the industries have started adopting in the food and the automobile. But the first patent has been filed for this is in the year of 1984. On a SLA technology, then followed by a different principal machines by Dr. Carl Becker on the laser sinting technology, followed by the fuse method, followed by the first invention of sheet lamination. These all are the machines which has different principle but works on the only 3D printing technology. And later in 2000, we have developed our first bio absorbable. And after that, after 2000, then the evolution has happened, and we started exploring that how this 3D printing technology can be adopted in the medical as well as in the pharma field. We have gone through a different processes, and we found that 3D printing technology has given an artificial kidney, printed vessels, and finally, as I said, we started evaluating in 1996. From 1996, we have adopted a different procedure principle to adopt to a final dosage form, and in 2016. US FDA has given a first approval on the 3D printing technology, which is currently available in the market. It means a 3D printing technology is a future of the formulation, where you can give a flexibility to your formulation. Now, what are the different principles that are available and how that can be adopted in the pharma field? That could be a first thing that we must know. So there are 7 principles that is available in the additive manufacturing. This starts with the binder jetting, then vat polymerization. Binder jetting is nothing but where it involves only solid particles to formulation. Vat polymerization, where your whole formulation depends on the liquid. Where powder bed diffusion is completely a solid particle involved in the formulation. Material extrusion is nothing but a filament from the hot melt extrusion where it's involved in the formulation. Material jetting is nothing but an inkjet printing or polyjet we call. The formulation involves completely in the liquid. Followed by the directed energy deposition. Here the metals or the wires will be used as a raw material to give a shape to the formulation. And finally, the sheet lamination, where different sheets of the different viscosity, different composition are fused together to make it into a final formulation. Now, what is the principle involved in this technology? Now, in the 3D printing technology, we will have one platform. In the platform, we will have one nozzle where the feeder can be a liquid or a solid material or a filament. This nozzle will move across in the 3 different dimensions. It can be in any axis of X, Y, and Z axis where it will move as a printer, and that will give you a complete dosage form. And later, once the product is printed as per the requirement, then this platform will be removed, and then you will get a final dosage form. Now, how does this work? What are the things is required to make it into a 3D formulation? Now, first you require a modeling. In the market currently, we have a different software where you can feed what you want to. Now, this is we call as a CAD, a computer aided design, where you will be giving in the different axis of what is the length of the tablet you need, what is the height of the tablet you need, what is the width of the tablet you need, or it can be a different dimension of the tablet that you need. And then that will be put it into a computer aided design, and that design will be put it into a different principle of the machine, which I have discussed earlier. There are different principles which works on. Then that software will start printing a tablet for you, and that after printing, the stage will be removed or the platform will be removed, and then you will get a final product. So that's called, this is how the whole principle of 3D printing works, irrespective of whatever principle that you're going to adopt to the industry. Now, I would like to start with binder jet printing, where the principle goes like this. You have a bed, which is made up of a different powder, and you have a liquid binder. It could be any binder according to your need of the formulation. Here, you have a nozzle where you will be infusing your liquid binder into a nozzle. In this nozzle, you can apply a different kind of a pressure or a heat where the viscosity and the feed volume can be changed. So when you have this access to change different processes, then your outcome can change. As we know in the traditional, if the binder is thicker or the higher binder, your tablet hardness as well as the outcome will be different. If you have a less viscous binder or a different vehicle use binder, your outcome will be different. So in that way, you can control over here by choosing a different liquid binder or by modifying the binder jet. So droplet will decide your outcome of the tablet. So now the principle goes like this. When you add your droplets through your injector and over the powder layer, so it forms one mold first, then later you can stop a roller. A powder delivery platform will roll another layer of a powder, then against the droplet will be dropped, then it will give a final shape to the tablet. So this will be as a layer of a different powder, and that will give into a tablet. Now you can, in this way, you can formulate a different formulation. Now you can, as I said, you can have a different droplet size. You can control the viscosity of a binder. You can make a heat. You can give a heat to the binder. So with that, different viscosity, different size, different feed rate can maintain. so that you can have a complete different profile of the dissolution, whatever you need. That can be controlled in 0 and any other dosage form. So this is the technology first adopted by the industry. And now from this technology only, our first US FDA product is available in the market. Now, as I said, you can have a different design in your software. So you can have a different powder region. You can decide your viscosity and the thickness of your each layer of your powder. So with that, you can have a completely different profile according to the need. Now, this is a SLA. We call it as a serial lithographic. Is a generic of 3D printing, where here the liquid photopolymer will be there in a tank or a platform, where you will be applying a high laser beam or any other light which is suitable for this photopolymer. It depends on what kind of a photopolymer that you are deciding. So if the photopolymer is using a high laser beam, then you can select that kind of a laser beam, or you want any other laser beam like UV or any other, you can select accordingly. It completely depends on your photopolymer. Now, this is by applying an UV light or any other light, you will be applying a lower localized temperature, and this technology is more suitable for thermal labile. And commercially, there are different resins and photo initiators are available so that you can adopt your technology or the formulation or the outcome according to your need. So how do this works? So you have a build plate and you have all the photomerizable resins. Now, when you apply a laser source, it depends on what kind of a polymer that you have decided here. So the whole principle goes like this. So your photopolymer resins, you have a platform, it moves up and down, and you are applying a laser over here, and it can be a 3 or it can be n number. It depends on the scale of the machine that you have decided. And you can control the frequency of your laser beam. So once the heat is applied over the surface, so it gets solidifies. Once it gets solidifies, the moving apparatus moves up and down to make a further layer. And as the layer, this process continues up to make a final formulation as per your design. And this continues for a different layer also. A powder bed diffusion is based on the same technique, but instead of selecting a liquid polymer, here you will be selecting a solid powder jet, but you can this powder jet you can make a fusion to make a finished dosage form by applying a laser, and that completely depends on what kind of a metallic or ceramic layer that you have decided. This is a complex version, is a mixed version of the previous technology and SLS technology, where you have a powder and there is a roller which moves to make a layer by layer. Then you are applying here laser. Then based on this layer, the roller moves and it makes a different layer, and you get a final formulation where you can tailor your formulation according to your need. Now, how does this forms? Because it's completely based on your solid material where you are applying your laser beam, and the temperature which generates, and temperature, it gives you a mold of a tablet what you need. Now, initially there are grains. Why? Because you have selected a powder. Powder can be of any shape or a size, but it will have a void space, which we call a pore. As you are applying a high beam laser on the particle, it starts fusing together. And when it starts fusing together, the void space or the pore which it has, it fuses and it forms a solid liquid material where the fusion can happen completely. And further applying a more high heat or the laser, where it can completely fuse together, and that decide your final design of your formulation. So you can use this technology where you have solid, or you can use this technology where you have solid liquid also. So where you have a powder and you want to make some semi-liquid on this, and you can have a complete outcome of it. Now, this is a filament technology, and this filament fabrication is depend on a hot melt extrusion, where these filaments are made up of hot melt extrusion. This hot melt extrusion is well known in the pharmaceutical industry, where to convert the most of the polymorphic form into an amorphous, we use it, where it has different chambers, where you will be feeding an API and the polymer, which is thermostable, and by mixing together, we will be getting a filament. And this filament, we can use it for our 3D printing. Now, how does this work? Because these filaments we can get it, or you have to manufacture according to your need. Once these filaments are incorporated into the nozzle, this nozzle is having a temperature sensor where you can apply a temperature according to the need of your filament. Either you are using a one filament or a multiple filament. Now, when you apply a filament, as you know, this filament has a tendency to melt. So you can start doing in printing by computer-aided design. Then you will be getting a complete tablet. Now this can be applied through a filament, and this you can do a tailor formulation, not only in one formulation. You can have multiple drugs in this. Why? Because you, as I said, it's completely depend on your machine, where if you can add a further filaments, and you can have 3 or 4 multiples in one single tablet. Now, this semi-solid extraction, we can do it by the solvent evaporation technique also. Now, this technique, instead of having a filament, now you will be injecting your input material using a syringe with semi-solid paste or gel. When you inject this using a nozzle, when the nozzle is there, and depend on your kind of a solvent that you're using in your technology, that gets evaporated and then it forms a mold. That molds, as I said, it's depend completely based on your requirement. Either you can make it a immediate release, or you can make it as an control release also. And the solvent evaporation technique is well known in the pharmaceutical industry, where you can change the polymorphic form of a tablet. With this technique, you can change the polymorphic of a tablet as well as the appearance of the tablet also. Now, with this technology, we have a bioprinting also, where this first time it was introduced, so fabricated material according to the human need and in a different organ also. And it has been proven in the industry that the selection of a polymer adopted in the industry, in the medical device, it can be used and accepted well. Now, energy deposition is a kind of a similar technology, but here what happens, we have a laser beam material where we will be scintillating a different polymer or a different metal over a zone where it has a high temperature, and with this the printing happens. It's nothing like you have a powder or a liquid on the platform. It completely depends on the feed, and there is a laser beam focus on the center where the feeding of the material will design your formation. And this can be modified according to the laser that you're going to apply, or the metal that you're going to select. So with this technology, now is working most of the automobile industry, where you need only one part. So with this technology, you can make that part, and you can tailor as per the need. Sheet lamination is almost related to our hot melt extrusion, where we are using a thin filament. Instead of a thin filament, here we will be using a sheet. Now, in this sheet, it will be a tailor-made sheet available in the market, or it can be made as per our need, where the metal powder nozzle, we will be applying a high power laser and change in the different frequency of the applying waves. So you can modify a layer of your formulation, and you can tailor your formulation, and finally you will have a finished product. Now, with this all principle, because we are using a 3D printing, and it depends on your one platform or the kind of a raw material that we are using, whether it can be a powder, or it can be a filament, or it can be an liquid. Now, while doing this, there may be a defects. As we know, in the traditional way, the tablet has some defects in a traditional way of manufacturing, like capping, lamination, and all those things. So while adopting this technology, there are some defects which isn't identified. So what is that? The first defects, which we call it as a banding, where because the axis moves, your nozzle moves to print a tablet. So because of that vibration, the banding can form, and that banding can lead to defects in the tablet. And leaning, warping, this all happens. Why? Because the different principle are adopted by the machine, where you applying in heat or in vibration or the movement to the nozzle. So this kind of defects can happen. So delamination by missing a binder solution. For an example, you are applying a roller and the roller is not moving properly or is moving randomly, then the deposition of the binder cannot happen to fuse that both the layer. So delamination can happen. Drifting in the base layer. And finally, a collapse can happen if there is no porosity. If there is no porosity, the wicking property of a tablet will not be maintained, and there may be a defect in the tablet when we do a dissolution. So this is the major defect. So what is the advantages when you adopt this technology? As you know, in a traditional way, in the start of a presentation, I said it's a long process. You require n number of resources to manufacture one tablet, and it has to be in one particular release or one particular formulation only. Now, when we want to adopt this technology to the different rays across the globe or the patients which has different complexity, so that time you cannot change the formulation for each patient. But this technology has given a flexibility to adopt that. So the first advantage is dose personalization. Dose personalization, for an example, one person needs 100 mg of a dose, where the another person needs only 50 mg to ensure the therapeutic efficacy. This technology has given a flexibility to adopt for children as well as for the elderly, and specifically for the narrow therapeutic index drugs. And multi-drug combination. Why multi-drug combination? As we know today, we are having an n number of pills to cure different diseases. What in case if we have only one pill which takes care about everything what you want at the moment? This technology will give you a flexibility to adopt this kind of an dosage forms, where the market is working on all the different formulation like atenolol, pravastatin, amipril. This all can be put it together with a different release into only one pill. So that gives a huge advantage for the patient's compliance. Now, tailored release profile. What do you mean by tailored release profile? With a single formulation, you can have a different release profile where you can alter wherever you need to. And this 3D printing is not only giving you a different release profile, you can design however you want to. So it's completely depends on the patient compliance. Now, for an example, the patient cannot have a star shape of tablet, and he or she needs a tablet which is in the round shape, which is more palatable. So it is doable without changing any formulation. Improved patient's acceptability. As I said, for doing a clinical trial for any formulation, it requires a huge mass production for each stage of a clinical trial. But when you have a 3D printing technology where you can manufacture one tablet to 100 tablets, that gives a huge flexibility to the market or the industry, where they can save a lot of resources or the investment. On-demand printing. Now, if the product has very less stability, where the product shelf life is only for 12 months, so the market will not give that kind of an acceptance to that kind of a product where very less shelf life. But 3D printing will give you a flexibility where you can manufacture immediately and give it to the patient as per the requirement, and the numbers also can be maintained. And as I said, in the earlier phase of a drug development, we have to go through the n number of phases, and to go through the n number of phases, you have to modify your formulation in each phase. When you want to modify each phase, a mass production at each phase, it means a huge investment on the industry. That's why the generic industry is doing very good, but in the NCE molecules, where we have a very less molecule. So in this technology, if you adopt very less investment is required, so we can have a future where you can, with the less investment, you can have more molecules in the market. Dose flexibility, as I said, you can have a different doses as per your requirement. So what is the thing that we have to take it into consideration? Now, product complexity is a very complex free. As I said, different principle has different adaptability. If your product is thermostable, you adopt one technology. If you're thermolabile, you can adopt one technology, and where you can manufacture a tailor formulation or a multiple combination or any design that you want to, and you can start doing your development, or you can start doing your production. So no assemblies is required. It's just a simple printer machine where you can start doing your development. Infinite shades of material. As I said, there are n number of materials available in the market, the principle that you want to develop, and unlimited design space. As it's a computer aided design, so you can modify design however you want to. So this is the product complexity that makes it. And manufacturing and the process scale out is parts can be printed on demand. It's not required that you have to keep an warehouse where all the material has to be ready and you have to do a mass development. The product can be printed as you require, more compact, and is a very layman process because you need not to depend on a manpower where there may be and chances of human error. It's completely a computer aided program where a layman can only input a software design, whatever they have done it, and start doing a printing. What is the disadvantage or what are the things that we need to consider when we are evolving in the 3D printing? Now we doesn't have any regulatory body control on this. So when we want to adopt any technology, like I want to buy an lamination or a sheet lamination, I want to buy a filament, who will maintain? Who will be this distributor? What are the specifications will be maintained? So these are the small questions that need to be answered before adopting this technology into a market. For an example, in cartridge containing API and excipient and their distribution, who and how will be regulated? Who will dispense? Because the machine is available in a very simple and the smaller manner. So who will dispense? Either the doctor can dispense because it's a computer-aided design, only they have to fit in filament or the powder layer and they can start dispensing it. Or a pharmacist, that question need to be answered. And who will maintain this regulation for a medical device or the pharma? Manufacturing process medication can be done at home or not, because it's, as we know, is a tailor-made formulation. So if I need that kind of a formulation, so who will monitor that? That can be produced at home also if you have a machine. So specification, what kind of a specification that has to be put forward for any raw material on a finished dosage form, that has to be discussed. How can we define a shelf life for any product which is printed in the 3D additive manufacturing? And shelf life for the drug loaded print cartridge to be monitored, means whatever cartridge that we are using, how does the shelf life is defined? What is a product collection and the rejection? Material carryover, what to do with the material carryover? How to set up a specification for a 3D printing process? What is the scalability that you have to apply when you want to do a mass production? And post-processing and packaging, because it can be more fragile, and if you want to make it a tailor formulation where you want to make your formulation more faster, it can be more fragile also. So how this post-processing and packaging can be done once it's been manufactured either by the doctor or the pharmacist, how it will be given, that thing is not answered in the industry. And equipment cleaning, as the same machine can be used for the different formulations, so how do you maintain the cleaning of the machine? So achieving optimized drug release, these are the small things which is the future. Fabricate and engineer various level of novel dosage form, achieve an optimized drug release profile, because you need not to change anything, only the feeder has to change and the software will give you the release profile, so you can adopt any profile that you want to. New excipient, as we are using a different principle for the different machine, so we are limited to use an excipient which is useful for this technology. So we need a new excipient which can adapt to this technology. And if we wanted to avoid incompatibility between the multiple drugs, we never know that you can have a multiple dose in one pill, but how does the multiple combination of an excipient and the API in one pill have the stability or the incompatibility? That need to be studied. And limited degradation of biological molecule or helping resurgent cues, these are the things that we can expect. And in the future, if you remove the platform, then we have a 4D printing technology also. So the industry is working to make it as a 4D printing also for in the future. So that's it from my side, and thank you for your listening. And if you have any question, then you can write it to my mail ID, or you can WhatsApp me at any time.
Thank you, sir, for the session. I request all the delegates to be seated for next 10 minutes. We'll have an inaugural function. Swamiji is going to be arriving, so please wait.
Welcome. ಮೇನ್ ನೋಡ್ಬಿಡಿ ಸ್ಟೇಟಿಂಗ್ ಸ್ವಲ್ಪ ಬ್ರೇಕ್ ಕೊಟ್ಬಿಡಿ ಫೋಟೋ ಬಿಡ್ತಾನೆ ಇಲ್ಲ ಫೋಟೋ ಲೇಟ್ ವೇಟ್ ಮಾಡೋದು
ಅಲ್ಲೇ ಸ್ವಲ್ಪ ಕ್ರಾಸ್ ಆಗೈತೆ ಅಲ್ಲೇ ಫ್ಯಾನು ಫ್ಯಾನ್ ಇಲ್ಲ ಸ್ಟ್ಯಾಂಡ್ ಫ್ಯಾನು ಫಾರ್ಮಸಿ ಪ್ರಾಕ್ಟೀಸ್ ಕೊಟ್ಟವ್ರ ಚೇಂಬರ್ ಅಲ್ಲ ಯಾವ ಚೇಂಬರ್ ಅಲ್ಲ Yes, sir. File name, sir.
Folder name, conference material. You change that message, you make it as main call. Instead of name, it has come as mail. MAIL it has come.
Okay.
Spelling is mail, MAIL.
Okay, thank you.
People are getting confused.
Okay.
Where you are changing,
Sir.
Hello. 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 Respected everyone, I am Preeti Sharma from Amity University. My research topic is, my presentation topic is
correlative study of iron content with genetic diversity in Moringa oleifera lamb assembled from 4 different agroclimatic zones of Rajasthan, India. Moringa oleifera lamb, mineral and rich tree, family Moringaceae. It is originated from northwest part of India, belong to family Moringaceae, and there are so many names of Moringa oleifera was drumstick, ban oil tree, and horseradish tree. The Moringa oleifera is characterized by tripinnate compound leaves, hanging three shade pods, and grey corky bark. Moringa oleifera and Moringa concanensis both are closely related to each other. Methodology for the study of morphological, genetic, and diversity. I have collected samples from 4 different agroclimatic zones, like minimum 5 and 10, maximum 10 locations. For the study of iron content, bed digestion method was used. In this method, concentrated HNO3 is used. Then after making solution 25 ml, it is used for layers. Determine the iron content. For the maximum, for the value of atomic absorption, photometer is 190 to 900. For the study of 248.3 nanometer. Study of genomic DNA. Here, reagent it used to remove the polysaccharide. Here, HCl is used to maintain the pH buffer, and EDTA is used for killing agent. It binds with magnesium, and so DNAs don't work, and then become protein.
No, Doctor. Yes, welcome.
Jai Shri Gurudev. By seeking the blessings of His Holiness Paramapuja Jagat Guru Shri 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 Dr. Balagangadharanatha Mahaswamiji, and rendering our humble pranams to Paramapuja Shri 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 Dr. Nirmalanandanatha Mahaswamiji. We welcome Puja Swamiji, dignitaries and delegates to the International Conference on Innovation and Advances in Pharmaceutical Sciences, organized by Sri Adichunchanagiri College of Pharmacy together with Association of Pharmaceutical Research. The function of prayer is not to influence God, but rather to change the nature of the one who prays. A day without prayer is a day without blessing. I welcome Deepthi for invoking Almighty's blessing through invocation song.
जटिल कुटिल तमांतरंगा जटिल कुटिल तमांतरंगा भावविहीन संचारी भावविहीन संचारी तरतमनि हे आत्मतमोहारि जनुम जनुम शतकोटि संस्कार जनुम जनुम शतकोटि संस्कार परम चरम संस्कारी परम चरम संस्कारी तरतमनि गुरु हे आत्मतमोहारि हे आत्मतमोहारि आत्मतमोहारि
Thank you, Deepti. Now I request Dr. B. Ramesh, Organizing Chairperson, Dean and Principal, Shri Adichunchanagiri College of Pharmacy, to take over the dais.
Jai Shri Gurudev. My humble pranams to the lotus feet of Parama Puja Jagadguru Shri Sri Sri Dr. Balagangadhar Swamiji, and seeking the blessings of Parama Puja Jagadguru Shri Sri Sri Dr. Nirmalanandanath Swamiji, Honorable Chancellor, Adichunchanagiri University. I take this opportunity to welcome our Parama Puja Swamiji because during morning inauguration he had preoccupied the event. That's why he could not bless all of us. In spite of busy schedule, he is here to bless all the delegates here. Big round of applause for our Swamiji. I request Organizing Secretary Dr. Prakash Gaudamohan to seek the blessings of Puja Swamiji. And I also request Mrs. Stapati, the organizing chairman of this APR Advanced Pharmaceutical Research, to present the plan for Puja Swamiji. And with us we have Dr. E.S. Chakravarti, who is Vice President of TCS as Tata Consultancy Services. He is Vice President and also he is Global RMG Head, Resource Management Group Head. He is also here for the scheduled meeting with the Honorable Chancellor and Adichunchanagiri University officer here. On behalf of all the 472 delegates, 427 delegates, I would like to extend a warm welcome to Dr. E.S. Chakravarti. I request our Organizing Secretary to honor Chakravarti sir. I request Puja Swamiji also to bless Chakravarti sir. And without much ado, morning we have witnessed and we have welcomed Stapati and Organizing Secretary. And for the information of Puja Swamiji, the Honorable Chancellor, for this international conference, which is first of its kind on hybrid mode, it is a 2 days international conference, wherein 427 delegates across India have registered, and 262 are attending physically, and 165 are attending virtually. And around 212 scientific papers have been will be presented in 2 days of time. around 8 keynote speakers, national and international reputation. They will be also presenting their keynote address physically and also in virtual format. With this brief introduction, now I request Sri Dr. E.S. Chakravarti to address the delegates with his motivational words for 5 to 10 minutes. Over to Chakravarti, sir.
Jai Shri Gurudev. Jai Shri Gurudev. This is one of the best and the inspiring habits or the culture I picked up after visiting this BGS group of institutions for many years to start any meeting or any meaningful conversation with Jai Shri Gurudev that gives us energy, that gives us the purpose of that particular discussion as a blessing. So Jai Shri Gurudev, and thank you so much for giving us a wonderful, wonderful opportunity. And to Ramesh and all the teaching staff and non-teaching staff of this Adichunchanagiri College of Pharmacy for putting together a wonderful, hybrid program, especially after COVID, putting together both online and offline programs coming as hybrid program. It's very, very nice to see the people coming together. Hats off to them and give a big round of appreciation. So especially for the eminent speakers and participants who are part of this particular program, welcome to BGS group of institutions. I'm sure the blessings of Swamiji are going to get extended to all of you, including us, for doing better and better for the future of the society that we belong to. One thread that I would like to mention today to all of you as my sincere submission to each one of you is there are many articles about pharmaceutical industry, how the trends are coming, what kind of changes that we need to bring, what kind of future we are foreseeing. And one of the threads I really enjoyed from the literature is pharma is changing from a dispenser-centric activity to a patient-centric activity. And that is one of the most inspiring lines that I read on the trends of pharmaceutical, pharmacy, etc., etc. So I think that is something very, very powerful statement to see. The trend is from a dispenser ecosystem or a dispenser-centric education towards a patient ecosystem education or patient-centric education. And I also have seen a couple of other statements in that article that every student or every practitioner of this particular stream of education or profession is how individuals can play 3 roles to make this shift happen from dispenser-centric to patient-centric. Number 1, can you become an optimizer? So whatever the processes we have, which is the dispenser-centric processes, can we pick them and try to bring a lot of change in the process and apply them towards a patient-centric system, that is optimizer. Take a process, look at the process, how this process has to be changed, how this process has to be improved, and kept on to the new way of thinking. That is optimizing the existing processes or shifting the existing process. Not only what we do, we say shift and drop. Instead of it, you shift it, look at the process from a patient-centric system and try to modify it and start deploying it. So some of us have to become optimizers or transition agents of picking up every process and dropping it in a patient-centric environment. The second one that we spoke about is, are you a transformer? Are you a transformer? So can you look at how to bring transformation from various entities of pharmacology from a dispenser-centric to a patient-centric? So you can play a role of an optimizer. I don't want to go deeper into that because we have some other meeting to attend, very important meeting. Because today a lot of us are coming together and reviewing the progress of ACU under which this particular institution falls under, and what is the plan for next 5 years to make ACU one of the benchmarking universities in this country is what exactly the meeting what we are going to have once after
this session is over. So very important session. So I will not take much of time. And the third one, which is very important, is the role what we are supposed to play is innovator. So one is optimizer, one is transformer, one is what is that, innovator. So how to bring innovation, both incremental innovation and innovation play at large, small changes and large changes. That's what exactly called as incremental innovation and innovation at scale is what that article spoke about. How all of us can come together and start behaving like an innovator. And that is the role which the article says that all of us have to play, who are part of the profession, who are part of the education, to start visualizing ourselves as innovators and try to bring lot of ideas, innovation, progress, and change into the overall what is that, ecosystem. So boys and girls, if I look at the title of this particular, what is that, International Conference on Innovation and Advances in Pharmaceutical Sciences. I think underline the statement innovation and what is going to happen as part of it, how the ecosystem is going to get into act, and what are the important events of the ecosystem, elements of the ecosystem today and tomorrow. Go through it, understand it, and try to play the role of innovator in the pharmaceutical industry. Once you are done with the degree, before you get into the profession, having the mindset of an innovator, and also who are already in the profession of teaching and also pharmaceutical as a profession, how they can start behaving like innovators. So we don't want too many change agents, we don't want too many transformers, but we want many more innovators that has to come to shift the business from dispenser-centric, what is it, sciences to patient-centric sciences. With this, thank you so much for giving us a wonderful opportunity. But let me tell you, I visit a lot of institutions in this country. People who are into biotechnology, pharmaceutical, etc., etc., etc. It's my personal compliment. I said this before. The way Mr. Ramesh understands what change is coming in pharmaceutical education ecosystem, I don't think any other person understood it so well. Give a big round of compliments and appreciation for one of the wonderful leaders that we have in this institution, and I'm sure it is going to go levels after levels in the future to come. Jai Shri Gurudev, thanks for giving the opportunity.
Thank you, sir. Now we have the release of our college newsletter Udan and the souvenir, which is a compilation of scientific abstracts.
Now I request Pooja Swamiji and the dignitaries to release the Udan and souvenir. Thank you.
The power of concentration is the only key to the treasure house of knowledge. I request our Pujya Mahaswamiji to shower his blessings on us.
ब्रह्मानंदम परमसुखदम केवलम ज्ञानमूर्ति द्वंद्वातीतम गगन सदृशम तत्वमस्यादिलक्ष्यम एकम नित्यम निर्मलम अचलम सर्वधि साक्षिभूतम भावातीतम त्रिगुणरहितम सद्गुरुं तं नमामि। श्री विद्यां जगतां धात्रीं सर्गस्थितिलयेश्वरी नमामि ललितां नित्यां त्रिपुरसुंदरी। On the occasion of the International Conference on Innovation and Advances in Pharmaceutical Sciences, I submit my humble pranams to the divine soul of His Holiness Guru Padma Bhushan, Dr. Bal Gangadhar Mahaswamiji, who is the reason for the existence of this institution. And today's chief guest, Mr. E.S. Chakravarti, Mr. Ramesh as a principal, and all the different dignitaries who are present on the dais and off the dais, and dear students, and pharma entrepreneurs who are here. Already, Mr. E.S. Chakravarti has spoken bit of the conference. As we do have one more very important meeting, we have to move over there. Something like we are intruders now. Morning, this occasion supposed to be inaugurated. It is inaugurated. Time doesn't wait for anybody. As we did have another program, we will make it, though this is our own campus and our own institution, as a priority, we are supposed to be here. Mr. Ramesh had postponed this event twice, so got confusion, perplexed. As a result, we have missed it. As yes, Chakravarti has mentioned, he has a great vision towards the domain to which he committed to. This physical body is very, very important. Sharira madhyam kalu dharmasala. Though it is said, the main purpose of human birth or any birth in this world is to attain the ultimate liberation. Correct? If not, just to sustain this physical body, not necessarily to come and to go universities for 25 years, studying quite hard, earning, only to sustain this physical body for next 100 years. This kind of university education is not needed. If you think that it is needed, some other animals and insects and birds are also living without going to university and learning and not conducting these kind of international conferences and innovating and making an advancement and all. When you look quite seriously and intently inside, and when you listen to some great words of great wisdom, people, then slowly we'll understand the very purpose of our existence over here. It's because of that I said, Sharira Madhyam Kalu Dharmasala. To attain and to achieve ultimate, it is said that human physical body is very, very important. So whenever problems would come to us, to this physical body, obviously it should be fixed. The way how we fix the engine similar to this physical body also should be fixed. Then your pharma will come into existence. So there is a possibility even when your pharma knowledge drugs means medicines are not at all needed. So whatever the kinds of innovations and the advancements have been happening in this field, in line with that, if you can concentrate on certain methods wherein practicing which human physical body, not only at physical level, though you have been inventing medicines and all these things to sustain and to take care of the physical body, if we have such medicine wherein that is going to take care of our intellectual and mental body also. As we know, gross body, subtle body, and causal body. What all things are happening in pharma industry or pharma domain is going to take care of only the physical body. If you can invent and if you can have the advancements to treat our inner bodies also, then we can appreciate. Let this kind of international conferences would concentrate on those aspects also. Something like medicine is going to give physical body. Meditation is going to make us to understand our inner reality. So medicine through tablets and through such drugs, it's not possible for us to understand what is that. There is a saying that if you take this kind of drug, so in ecstasy you can be for some hours. I don't know how it's going to happen. Please, you don't invent such kind of medicines or the drugs which is going to bring humanity to the lower level. So whatever the intention and the vision this conference is started, I wish all the best because innovation is very, very important. World is moving so fast. If you are going to stick to the old ideas and old methods, obviously there will not be any advancements. If innovations would happen in newer way, physical body that can be sustained for more years, such physical body is going to be useful for the service of the society. So today we are here to attain such kind of knowledge, if that can be converted to wisdom. Eventually, with the blend of knowledge and the wisdom, if we are going for society to render the service, such society it can be called as a evolved society. So I wish all the best, because not just because that we have time constraint, we should not come in the way, because this is hybrid conference which is happening. I wish all the best for the best possible things to happen here, but in the conference. And those who have come to this institution from elsewhere, from different institutions, I wish them also. I wish the comfortable stay as long as you are going to be here. I am damn sure that our system, particularly around Mr. Ramesh system, will take care of you. Feel comfortable, feel at home. Wishing you all the best. Beseeching Karberar to best of best possible blessings upon you. Technical notes. Jai Shri.
Thank you, Pujya Swamiji, for gracing us with your presence and showering us with the Almighty's blessings.
By this, we come to the end of the stage program, the first day of APIA conference.
I thank Pujya Swamiji for gracing this occasion. I thank all the speakers of today's session for carefully carrying out this session to the delegates' utmost benefit.
I thank all the delegates for their attentive and patient listening and making this day a grand success.
Thank you all. Now, oral and poster presentations will be carried out in the respective seminar halls. I request the volunteers to help our delegates to lead to the same.
I request the presenters to proceed towards the same.
The high tea has been arranged. Please savor the same and then go to your respective seminar halls. Delegates, for tomorrow, I request all of you to be present here by 9:30 a.m. If any of the delegates require any accommodations, I request whoever requires to contact the hospitality committee. That is Dr. Rupesh sir.
ಸಾಮಾನುಗಳು ಇಸ್ಕೊಂಡ್ಬಿಡಿ ವಾಪಸ್ಸು ಏನಕ್ಕೆ ಈಗ ಇಲ್ಲ ಅಲ್ಲಿ ಮೇಲ್ಗಡೆ ಕ್ಲಾಸ್ ರೂಮ್ ಅಲ್ಲ ಹಾಕಿರೋದು
In this all the bits and all pin is bended. It's damaged.
Good afternoon, madam. Madam, my name is Deepthi Mathur. Shall I present this?
everyone. I'm Aparna. From Kerry College of Pharmacy Background, Department of Pharmaceutical Chemistry, and my oral presentation code is APR 020147061, and the title of presentation is Virtual Anti-Diabetic Screening of Selected Phytoconstituents of Embellia rice drum and the Pipermethrin. Now we move towards the introduction part. Embellia rice drum and the Pipermethrin plant have various therapeutic effects like antioxidant, anti-diabetic, anti-tumor, and anti-inflammatory because of the presence of several phytoconstituents in it, and numerous synthetic anti-diabetic agents which are already present in the market with the potent hypoglycemic activity, but with some of the limitations like hypoglycemia, weight gain, and the nausea, bloating, and these are overcome by the polyherbal formulations, and there are polyherbal formulations which contains these two plants are present in the market. And the drug likeness and the ADME parameters are mainly used in the drug design to optimize the drug-like properties exhibited by any of the chemical molecules, and in terms of the oral bioavailability, and to convert these lead candidates into effective drugs for the human use. And then network pharmacology is one of the important in silico approach, which is mainly utilized for detecting the mechanism of action of a drug in terms of a biological network by means of the genes, targets, and pathways by which all these chemicals and the phytoconstituents act. And then molecular docking, which is also one of the important in silico approach, mainly utilized for detecting the best fit orientation for the ligand that binds to the particular protein of our interest. Now move towards the aims and objectives. First aim is to screen the physicochemical properties and the pharmacokinetic parameters of the selected phytoconstituents from the Embellia rice drum and the Pipermethrin by the in silico methods. And then we proceeded with the biological network via the network pharmacology approach, and then molecular docking again is the selected anti-diabetic targets by the in silico methods. Now move towards the material and method section. In the current research work, we have chosen total 100 phytoconstituents from each of the plants from the IMPPAT database and the research articles for the virtual screening purpose. And the PubChem database was utilized for the collection of the canonical files and the downloading the SDF files for the ADME further studies. And then most of software was utilized for the detection of the drug likeness. And then Schrodinger software for the via Maestro 13.9 interface for the ADME studies and the molecular docking purpose. And then Digifred database for downloading the genes. And then String database for downloading the click pathways, and Cytoscape for the final construction of the network. Now we'll move towards the result and discussion portion. Here total we have screened 100 phytoconstituents from both the plants, and you are represented here in the table number 1 and the table number 2 with respect to the physicochemical properties and the drug likeness data. And we have covered all 5 parameters of Lipinski's rule of 5: molecular weight, hydrogen bond acceptor, donor, log P value, and molecular polar surface area. And most of the drug obeyed the Lipinski's rule of 5, and then we further proceeded with all those 100 compounds with the pharmacokinetic parameter analysis. And in the table 3 data pharmacokinetic parameters of the Embellia rice drum, and the table 4 contains the pharmacokinetic parameters of the Pipermethrin. Total we have covered 20 different pharmacokinetic parameters beginning from CNS to the rule of 5 and rule of 3. And is further subjected for the heat map analysis. In this, the green color ones are the highest values, and the yellow ones are the lowest middle values, and the orange ones are the least values, and the red color ones are the highly extreme values, which means the compound is violating that particular rule. So most of the compounds obey the Jorgensen's rule of 3, and then all of the ADME parameters lie within the recommended ranges. And then we further proceeded with the network pharmacology approach. The compounds which showed the positive drug likeness score were taken for the construction of the network. For the construction of the network, we have taken the canonical moiets and then added into the Digifred a database for downloading the mRNA up and the down regulation genes, and then added into the String database for downloading the click pathways, and then we have put a VLOOKUP formula in the Microsoft Excel sheet, and then we got the overlapping genes. From that we have constructed a network, and the compounds with the higher volume, those are the highly modulated compounds, and the highly modulated pathways by the highly modulated genes. And then we further proceeded with the molecular docking studies. These highly modulated genes like MMP2, FOXO1, PPR, gamma genes are highly expressed in these alpha amylase, alpha glucosidase, and then PPR gamma nuclear receptors. So we further docked on these enzymes and the receptors. So from the endolia rice plant, the pyrrolose is the compound which showed the highest binding affinity against the alpha amylase, and the methyl caffeic acid from the pipermethrin. And from the catechin and mopinamide drugs from the amblyopsy brum and the pipermethrin being against the alpha glucosidase, and diadine and the methyl caffeic acid from the PPR, which were compared with the standard drugs like metformin for alpha amylase and alpha glucosidase, and pioglitazine for the PPR gamma receptor. And now we'll move towards the summary and conclusion part. Out of the 100 screen phytocompounds from the 2 different plants, 83 compounds from the ERB plant and the 98 compounds from the PNL plant obeyed the Lipinski's rule of 5, and the 79 phytocompounds from the ERB plant and the 94 compounds from the pipermethrin plant obeyed the Jorgensen's rule of 3, and they possess the physicochemical properties as well as the pharmacokinetic parameters within the recommended ranges. And from the network pharmacology, we got to know that the luteolin 3,7,4-trihydroxyflavone and the quercetin compounds acted by highly modulated the TNF signaling pathway, estrogen and AMPK signaling pathways via MMP2, FOXO1, MDM2, PPR, SLC2A4 genes. And from the pipermethrin, camphor, methyl caffeic acid and pinene acted via PI3K AKT signaling pathway, AMPK pathway, and TNF signaling pathway via MMP2, FOXO1, MDM2 genes. And from the docking study, we got to know that pyrrolose, catechin, and the diadine compounds highly showed the binding interactions against the alpha amylase, alpha glucosidase, and PPR gamma. And methyl caffeic acid, mopinamide, methyl caffeic acid from the pipermethrin gene against the alpha amylase, alpha glucosidase, and PPR gamma receptors. So that we can conclude that the screen phytoconstituents could be proven as a potent drug candidates with a good membrane permeability and oral bioavailability. So this study can be utilized for the optimizing the marketed polyherbal formulations, and these data can be utilized for the repurposing of the phytocompounds. And these are the references.
It is time to ask question. Very basic question. Can you say Lipinski's rule of 5?
Lipinski's rule of 5 is mainly contains the 5 molecular weight if it is 500 Dalton. So the drug will not be able to produce the sufficient against solubility. And then hydrogen bond donors should be more than 10, and hydrogen bond acceptor should be lesser than 5, and then log P value it should be lesser than 5, and then polar surface area. So it should be in the range of 90 to 150 angstrom.
We know the 5 terminology has been used for the Lipinski's rule. Why they are saying as Lipinski's rule of 5?
Then it could be considered already bioavailable.
say if you are considering all the rules, you will be having the values of 5 over there for all other values. That's why we are having this term as 2 over 5. Yeah, good evening, Abha. So in docking, you selected 3. Which one? So why you selected 3? Any reason?
From the network topology approach, whatever the genes we have got, and then we do also one. These are the genes highly expressed in enzymes.
Are you have any raw data for this 105 constituents? Are you anywhere it's true? Are you have any value of it?
Taken from the database.
No, phytoconstituents I don't see.
I am the key database.
Question from what?
This is for selecting those 2 values.
Actually, this one project also in this we are developing the better development part. So for the everything from for that those plants we are planning for the synergistic anti-diabetic we are planning to do. And then after this in situ studies we are planning for the extraction, fractionation and the enzyme activities on this alpha amylase and alpha glucosamine. Yes, the compounds are reported from that plant, but there is no in situ work. Introduction for reference are mentioned. The all databases links are there.
Ma'am, presentation was nice.
Yes, sir.
But only one nice side. You mentioned that it's in your conclusion.
Yeah.
Good permeability and then oral bioavailability. Have you proved it? How you can confirm it?
From oral bioavailability.
Have you done that work?
Like from it is in silico approach only. From in silico approach we can conclude that, ma'am. But we will be further carrying out the in vitro and cell line study. From that we get to know, ma'am.
We cannot tell right? From the how many studies are taken bioavailability. So maybe we can say in the conclusion, maybe part of studies are there.
Yeah.
Percentage that you expect actually to the biology. So from in silico to practical one. So what is the practical?
Like if it is above 80%, then it is a good compound. Or lesser than 25%, then it is a poor compound for oral bioavailability. Thank you, everyone. Good afternoon, everyone. I'm Dr. Shreya Kosanam, Assistant Professor, Department of Pharmacology and Pharmacy Practice from Nirmala College of Pharmacy, and I'm also a research scholar from Kerala University, Andhra Pradesh. And today we are here to present the paper on network pharmacological approach and evolution of the approach in vitro antioxidant activity of Anthocephalus caramba against oxidative stress. Yeah, here we have used artificial intelligence approach to identify or to predict the targets. And along with that, usually the artificial intelligence can be used to predict the toxicity, pharmacokinetic parameters, and identification of targets and network pharmacology. And here we have selected the traditional plants, that is Anthocyclus caramba. It is being used for many other activities like anti-diabetic, anti-tumor, anti-diarrheal, hepatoprotective since ages. Our main aim of this study is a spectroscopical analysis and network pharmacological approach, and proving that approach in vitro antioxidant activity. And these are the, firstly, we have collected the plant leaf powder, and we have done the FTIR for the fruit powder, and followed by the GCMS of methanolic leaf extract. And whatever the data we have got from the GCMS, we have used that data to identify the targets. We got nearly 39 compounds from the GCMS, and out of which 16 have structures and targets. And out of 16, we have narrowed down to the 3 compounds using the cytohuba plugin, cytoscape, and degree value. And we have identified the targets for those 3 compounds using the Swiss target prediction. And followed by the identification of protein-protein interaction, we have built a string network of those 3 compound targets, and we have shifted that network from string to cytoscape, and we have again filtered using the cytohuba plugin. And based on the NCC value, we have find out the top 10 hub genes. And for the top 10 hub genes, we have undergone for the geo-enrichment analysis and tech analysis pathway. And we have shortlisted based on the false discovery rate and p-value, and followed by the in vitro antioxidant activity using the DPPH and ABTS standard methods. Here is the data. That is the FPIR graph. We have identified the presence of different types of amines, aliphatic amines, and from GCMS we got 39, and we have shortlisted to these 3 compounds: gamma cytosterol, beta cytosterol, and thymine, and they are the PubChem IDs. And this is the network we have built for those 3 compound gene targets, and that is the hub genes which we have identified from these number of, like, we got nearly 200 genes. From that 200 genes, we have narrowed down pathway using the cytohuba plugin. We have identified the top 10 hub genes, and for these top 10 hub genes, we have undergone the geo-enrichment analysis. Geo-enrichment analysis is a part, it's a website, gene ontology website, where we can identify the biological process, cellular compounds, and molecular functions of the genes which we have identified. For that, these are divided. Green ones are biological process, and orange ones are cellular components, and violet color one are molecular function. And followed by the KEGG pathway. These are the different types of KEGG pathways we have got, and majorly in these we have focused on cyclic GMP PKG pathway and calcium signaling pathway. And that is the cyclic GMP pathway, where these are different NAPK3 and nitric oxide synthase 3. These all genes act in this pathway, which can inhibit the cell injury, and it can also act as an anti-apoptotic activity. And same in the calcium also, because we focused on these pathways because during the cell injury, the ischemia and hypoxia condition occurs, and which first step is a decrease in ATP synthase and leads to the increase in calcium production. In the calcium content in the cell, in the mitochondria and the cytosol. So to prevent that, we have targeted these 2 pathways, and followed by the in vitro antioxidant activity. This is we have used 2 standard methods of DPPH and ABTS, and we have used standard as ascorbic acid, and we got the promising results at the 50% inhibition concentration. And these are the ranking. Based on the ranking of these compounds, we have selected the top 3 compounds. They are the PubChem IDs, and these are the MCC scores of those top 10 hub genes. And finally, I would like to conclude that based on the GCMS analysis, we have identified the top 10 compounds, top 3 compounds out of that, for which we have conducted gene enrichment analysis and KEGG pathway analysis, and for which we got through that pathway, we have identified the 2 pathways, like calcium signaling pathway and CGMP PKG pathway, which can act against the oxidative stress and which can prevent the cell injury in case of any ischemia hypoxia condition, example like myocardial infarction or any ischemic stroke in the part of the brain. Thank you.
Very nice.
You mentioned spectroscopical analysis, right?
Yes, sir.
So you're taking that.
That is just a part of the answer, but that is not the major concern. The major concern is about the network pharmacological approach, which we have identified using, like the first step is an GCMS. From GCMS, we have constructed network.
It is basic part to go for network pharmacology. Your title you didn't mention anything. And why this much confusion in your topic? You mentioned that you pharmacological and evolution of in-vitro antioxidant activity. Basically, you take natural extract.
Yes, sir. First, what we have is we have economic extract, and then we have constructed the network pharmacology based on the compounds we got.
Is there any reason to take natural extract?
No, sir, no particular reason. Like we got based on the yield, the percentage of yield we got, sir. Like we have done methanolic water chloroform 5 extracts. Out of this, we got good yield for the methanolic extract.
But nowadays, for application, they are not accepting. Either you can go for hydroalcoholic extract. Yeah, I am asking any reason.
Based on the yield percentage of yield we got, we have shortlisted it to methanolic extract. And also the components we but the DCM particular plant we got already and only for this extract. For the remaining we got only 10 like that.
Okay, can you tell about the geo network?
Geo enrichment and
advantages.
Geo enrichment analysis, it gives a clear pathway for us to predict the beforehand. Before going into the animal study or cell lines, we can predict what are the pathways it can act. So we can directly target to that pathway.
In the network pharmacological, you didn't highlight about inject antioxidant. You given some other activity like
different pathways I got.
give electron analysis of KE.
We focus on like apoptosis and the different like which we can get in the case of cell injury. So particularly in this cell injury, it will be caused because of excess oxidative stress, which can be like apoptosis and different. One is the clot formation, the cell focus on those two parts. No, sir. This is the basic study, sir. Actually, this is the part of the PhD. So this is the basic study, sir. We would like to isolate the compounds. Which what are the 3 compounds we got? Further, we would like to go for individual study.
Yield, right? You said that the
Yes, sir. Yield and also the like extract to be selected based on the yield.
right. So because mainly you select the extract based on the activity, right?
Yes, sir.
Similarly, if you are taking the yield, like in the chloroform extract, chlorophyll is more in that case. You get more amount of chloroform. So chloroform extract more. Then you will be going to the wrong direction, right? And another one, why you are going to the GCMS? Because GCMS normally gives the information to the fatty acids and some essential oils, but you are not going to get the information to the polar compound.
First, we thought it was the surface learning plan. So we thought that it will be having the high volatile content. So we can identify both number of compounds. That is the spectroscopical analysis.
However, I think everything probably done so much.
We might be like it might have the higher content, sir, not 100%. I can might have the higher content.
Like low what means you get condensation. But the flower means that they design any aroma.
Yes, sir.
Means like that much strong aroma is there like the flower.
Not like a flow, but sir, but it has a good
If you go for both extremes and extremes, you get the information about the polar and the non-polar compound. Based on that, is that some
basic like we have done till here. We would like to proceed it further also.
study of and other authentication
Yes,
study.
sir. We did that, sir. We planted
They mention any number over here. We need to estimate it.
like I thought this like the number of the site number.
This will be right. And one of it is how you rationally selected this one.
Not rationally, sir. Like this have like a different attributes like that protected. Everywhere not one if any disease occurs, it will be definitely a selection. So this has been using as a treatment for the particular because everywhere the cell will interact with the injector. So we thought that this plant might have importance in the particularly my product is based on trying to pull out the oxidative stress with the plant is against oxidative stress. Signaling establishing the mechanism. Yes, because though they are signaling pathways, we don't know the particular compound can all act in this particular pathway. So we are working on the individual compounds further. This prediction, we are taking these three compounds and we are going to further study. After that, it's like we are going to try
No, in extraction we don't go for extraction. Because extraction is very multi-component. So how we can take? We are taking it back.
Sir, actually, the thing is, what are the three compounds? Thank you.
Good evening, everyone. Myself Naina Javadi, and I'm from second year M Pharmacy, Department of Pharmaceutical Chemistry, MS Ramaiah, University of Applied Sciences, Bangalore. My title of the study is Identification of Novel Human Lactobenzyltransfer Derivatives for the treatment of tuberculosis, a computational approach. Tuberculosis is the worldwide dangerous disease, and according to WHO, around 1.6 million people have died because of tuberculosis in 2021. Mainly, tuberculosis is caused by some causative organism, that is Mycobacterium tuberculosis, and TB affects the lungs, and it can affect the other body parts also, like brain and kidney, etc. Some FDA approved standard drugs for the tuberculosis are isoniazid, rifampicin, and pyrazinamide. And these drugs have some side effects like skin irritation or skin rashes, hepatitis, etc. And the major drawback is the patient can develop the multi-drug resistance. So because of multi-drug resistance is in the patient, resistance will develop for the isoniazid and rifampicin. So this is the main cause, and this is the main reason for which we have to develop some. There's a need to develop some new molecule for the tuberculosis. Isoniazid, as we all know, isoniazid is the first line drug for the tuberculosis, and it acts actually on the body by interfering the synthesis of some major component, that is mycolic acid, and which is important for the synthesis of cell wall of the bacteria. So isoniazid affects the biosynthesis of mycolic acid. Aim and objective of the study: identify the ligands, correct binding geometry in the binding site to predict the binding affinity of the ligand with the active binding site, and to predict the ADMT properties of designed compounds. Isoniazid. It is the first line drug. So we thought of combine isoniazid with the 2-mercaptobenzimidazole because in some literature review we got to know that 2-mercaptobenzimidazole derivative also contains anti-tuberculous activity. So we thought of club the both of the molecules, and this is our proposed structure, and these are the substitutions we have on the proposed structure. Materials and methods what we are using. Firstly, camera we have used to draw those structures and in 3D format, and we have downloaded the structures in the SDF format. Then we have downloaded the protein, that is the mycolic acid synthesis protein from the PDB, and that protein should be optimized. So we have optimized the protein PDB software, and we have optimize the ligands, what we have prepared in the camera, that we have optimized, and we made the cluster of the ligands in the Discovery Studio. Later on, we have docked all the molecules what we have drawn with the protein in the Autodoc Pina and Pyrex. And Pymol we have used, Pymol and Discovery Studio we have used to draw the 2D and 3D structures of the molecules. And after all the studies, we have used Swiss ADME web server for the carrying out the ADME properties of this. Discussion. The protein data bank. From that, we have taken the IPKI protein, 1PKI protein, and as we all know, the higher negative value of the binding affinity, if it's there, the higher, then the more potent are the drug molecules. So as a result, it tells whether the identified drug candidate is really feasible to be druggable or not. So the binding affinity for the isoniazid and rifampicin we got as -6.4, and for rifampicin is -7.2 respectively. And for the proposed structure, the binding affinity lies between the -8.9 to -11.5. And by comparing the both standard and the proposed structures dropping score and these values, we can make the conclusion that some molecules, these molecules, if we can use as a drug for tuberculosis, if there is no side effect, then we can use, and it can be potent candidate after the synthesis. These are the binding scores in that molecule 3, that is BES-NRG3. This molecule got the highest binding affinity, -11.5. These are the 2D, 3D structures of first one. This one is the 2D structure of isoniazide, and isoniazide we have taken as a standard, and this is the molecule that is BBS enargitol. This has the highest binding affinity, like -11.5. Then we have carried out the ADMA study also, and we have taken here the 4 compounds, those who have binding affinities more than 10. So we have done the ADMA study for them, and these are the results. And all of these compounds are obeying the Lipinski rule of 5. Summary and conclusion. The isoniazide and rifampicin we have selected as a standard, and drug identification was carried out to treat the tuberculosis by interfering with the synthesis of mycolic acid. Based on the preliminary study, the 4 compounds we found to show high binding affinity with the target, and these 4 compounds we subsequently evaluated for the ADME study by using the Swiss ADME web server, and this compound 3, that is BBS energy, has the highest binding affinity with the better additivity property, and this molecule can be explored for the future activity after the synthesization. Thank you. We are going to continue this project by synthesizing these molecules. Not all, but we have got a 4 molecules that have a good binding affinity, like more than 10. So we are going to synthesize them in our lab. And this process is ongoing. I'm in 2nd year, so we are still doing this work. So this is just halfly done, so we thought of presenting the half. Yes.
But in docking, you should only
I said yes, sir. Because that for rifampicin also -7.2 is the docking score, but in the chart I mentioned only for the isomer. Yeah.
while you are novel drug, isoniazid is having a basic ingredient, you can say that in the assessment.
Yeah.
Why you selected for this standard cost?
Because it is also first line drug for tuberculosis, that's why it's
How many are the protein? How you can take
but for docking to just predict the activity we have chosen. Yes, we can't predict.
What are the parameters you are trying to select binding?
Yeah, we can have a break for 10 minutes. Then the session will continue. Okay, so you can break for tea and come back again.
So we have done literature reading.
Session will continue. So next one will be then. Okay, so we'll have here. Okay, so those who want to have tea and break for tea, so
yes, sir. Yes. Sir, I have got some articles in that they have taken mainly this protein, and they have talked with the isoniazid as well as the rifampicin.
No, I am asking the basic parameter, why you selected it? Because with the help of visual review of literature, we can't tell everything is correct, right?
Yeah, but it's not like only one. We got some more, like so many.
Actually, it is wrong.
Okay.
And what is the reason why you selected that specific protein? IK, 1 kp.
That is only based on the literature reviews.
Based on literature.
Yes. Mycolic acid, cyclopropane synthesis.
Let's expand 1 kPa. Let's expand. What is kPa?
Sir, that is a PDB ID for this protein. Yeah. I have to look after that. So.
Okay, that is the question.
Where is the active site for this protein? Where did you dock your molecule?
It's only the
Where is the
structure.
active site? Where did you dock? Where is this location? This is the zoom in.
Yeah.
When you look at the bar of the protein, where is that active site is located? You have docked your molecule, right?
Yeah.
Where is that active site? See, you have docked your molecule into the active site. Where it is located? On the surface, interior.
Yes, sir. Yes. But we have not added.
You show me the location. Where it is on the protein.
Sorry, sir.
Who did it? Right?
Yes, yes.
Which program you use?
For the 2D, 3D structures.
No, just docking.
For docking, we use the pipes. Auto docking.
Auto docking. No, the protein structure you use the ligand. This. So where did what part of the protein?
The binding.
Yeah, which part? It's huge. Just one so many. Where is the active site?
Actually, sir, that image was there. We have not.
What is active site of the protein?
Where the drug is binding with the.
That's what I am asking. It is binding in the active site of the protein. Where is the active site is located on the protein? What are the residues in that? So you put leucine, serine, and vitamin in the reference. You make a problem. You get very big. Is there a place to accommodate the big molecule in small pocket? See, big molecule in dark. Who is that one? See, that was very small molecule is the real structure has known. You have made a big molecule and it has gone to the protein with the autodactyl. How is it possible? How many other residues are interacting now? Instead of 3, there were 10 residues are interacting. Yes, yes. This is the reason you will this community values are increased. But there is no reason has to go and bind that particular. So whether we are going for doctor, we used to take only one standard. That will be related to other design structure. Second thing, if our drug is strong, it is not related to anything. So we used to related to that. So here is taken to that two is different. In this parameter is taken item. As per me, my knowledge, and ICIAPS is very different. One that we can compare the both sides. The energy that we can compare. So that is this character.
Minutes break ಅಂತ ಹೇಳಿದ್ರು. ಹೋಗಿದೆ.
So good evening, sir. So my name is Adichunchanagiri. I'm studying B.Pharmacy final year. Sri Shastra Pharmacy College in Chennai. My topic is about a dream machine MRI scanner.
Wait. Yes. Just we prepare my
So general introduction. In MRI machines, we use the MRI machines for analyze our brain activities. Other than in this dream machine, we are going to analyze our dream and visualize it. So scientists are
analyze our brain waves, that which is in our some synthetic codes. That are analyzed through MRI machine. So the main aim of this brain machine is to visualize our dreams. So then objectives, which is to the main goal of the is to visualize the dream only. So in this method, we have 4 sessions. So in first session, we are going through a Edinburgh hardness inventory, and then the dream recall frequency scale, Pittsburgh's sleep quality index, and 10 minutes TDCS test. In Edinburgh hardness test intensity, the person, that means a voluntary, the voluntary is going to be tested for some objects, and they repeatedly studied about that objects, and they visualize in that dream. In second session, accusation of 1.5T anatomical MRI, and there should be a homework. Homework means they have to bit questionnaires. After the MRI machine, after they sleep in the MRI machine, they visualize the dream. That could be there will be some questions after the sleep. And then in third session, TMS and sleep first night. TMS mapping of primary motor cortex, and then a placement of ECG, EMG, and TDS electrodes. So then participant falls asleep. Then after the 10 minutes of sleep, there should be a IEM. That means a rapid eye movement. In fourth session, except TMS mapping, we are going to do a MRI dream scanner. So, in ancient times, we are still considering we don't know how the dream should be visualized. So, in recent times, scientists have visualized our dream through MRI machine, but it should not be a proper way. It should be only a 60% of accuracy is there, and still there is no proper development in this dream machine. So scientists take 3 volunteers. So for each volunteer, they recorded 120 dreams, and they reported it for free. But finally, there is only 60% of accuracy. So the project is not clearly developed still now. So these are the brain frequencies when they are in a RAV. So this is my acknowledgement and reference. I refer only news articles. There is no any research articles or journals in this topic. So through MRI machine, we can visualize our dreams. But still there is no proper technology in our world. So that's why we have only 60%. We can now in this world
just hypothesis, right? Have you did any research? Some news or some researcher who published that you are.
So it's still from 2004, still now it's in process. But we have only 60% of accuracy. We shouldn't be able to visualize our dreams, but we can record the dreams in our brain. Yes. So only we can take it through only in MRI machines. Thereafter, I want to read.
Good evening, everyone. Myself, Mamata, 4th B Pharmacy from Seven Hills College of Pharmacy, Tirupati. Today, I am here to present about my topic, artificial intelligence in drug development. What is artificial intelligence? Ability of robot or controlled by the computers to do tasks that are usually done by the humans, but it requires human intelligence and discernment. AI principle is that approaching of complexing problems and breaking them into simple fundamental complexes through logical reasoning and give best solutions. As we know that we facing many obstacles in pharma sector. By using this AI, we come across from that obstacles. And what is the need of this AI is it has self-analyzing capacity and give results within a short period of time and give effective results and accuracy results, and it reduces time and cost of clinical trials and gives better results. And materials and methods. AI has various tools used in pharmaceutical companies in drug development. First one is in silico tools. It allows the assessing the affecting of amino acids substitution on the structure and function without conducting protein function studies. And it is a computational model. By using this computational model, we done pharmacological studies by analyzing data and by using analyzing data and data mining. And second one is microfluidic. Microfluidic is a chip and it is a miniature device which contains microfluid which resembles the human fluid. And this microfluid chip used as alternative model to animal model. And third one is admit lab. It allows to predict the 175 properties including solubility, pKa, pH, and toxicity studies. And various startup companies success through this AI by using different AI tools and methods. Some of the companies are Extensia. It is sensual chemist, which not only predict the drug, which not only predict the potential drug target, but it also build and send them to clinical trials. And so Stangin developed the Stangin bags, which intended to explore the latent chemical space to generate novel compounds with desired properties. And there are number of companies with success through these AI tools and by using these tools and methods. And applications of this AI in pharma sector is drug development, drug discovery, clinical trials, and other healthcare systems also. And as per global forecast, AI reached to 5.1 billion by 2023, and it has positive results in pandemic situations like COVID. And summary is many factors are influencing AI in drug development because of its rapidity and effective. AI is controlled by humans, sir, but not AI control human brains. No, sir. It is not control humans, sir. Yes, ma'am. Yes, ma'am. That you using AI only, no, ma'am. Thank you, sir. Thank you, ma'am.
No, black only is showing. See, black color only is showing. Yeah. Name. 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 Yeah. Yes.
Very good evening to everyone who are present here. Myself Jayshree Reddy, B.Pharm fourth year from 7 Hills College from Autonomous Tirupati. Today I am here to discuss about the topic method development and validation for the estimation of ambrobazole and terpitalin in pharmaceutical dosage forms by HPLC. There are various methods are available like HPLC and MV for development methods. We have to choose a particular, very sensitive method for the development of this process. Like the HPLC, in HPLC, we are mainly choosing a reverse phase because mobile phase is more polar than the stationary phase. Here, the compounds, the dosage forms which we have selected is ambrobazole and terpitalin. These dosage forms are mainly soluble in methanol, ethanol, organic solvents, but insoluble in water. For getting this, we need to maintain the theoretical plate. We need to make good theoretical plate that is about 8,000 to 12,000. Here we need to maintain the maximum peak. The peak for ambrobazole, after performing various methods, we get the good peak at for ambrobazole is 5 and for terpitalin is 9. The main important for development of this method is to development simple and sensitive and good retention and precision of this method.
So I want to ask you one thing. You have selected HPLC method. Or you are telling that its accuracy precision is fine with HPLC. Yeah? That you find. That you told. Okay. So apart from HPLC, any other method you can do? You can use UV. Comparing to HPLC, UV is the best method you will.
But comparing to UV, we will use HPLC. And we can use UHPLC.
Can we use HPLC for this? Maybe. And we get the results. No. Why professor is like that? Justifying on HPLC. What reason? Fine. Okay.
Why you selected these two drugs?
Because these two drugs are belongs to because these two drugs prevent the cough.
How many drugs are there later to come? And here is
Two drugs belongs to different classes. Revitalizing belongs to the beta agonist, and amroxine belongs to the sexualitis agents.
Is it correct? Amroxone?
Thank you.
Basic question. You are using a hydrophobic solution, right? That's also very important. So, what type of mobile phase you are going to use? You are going to use solar or non-solar? Yes. Polar is right answer, but why polar? Basic one actually, because you are developing a method.
So myself Sri Ram from College of Pharmacy, Revitalizing. So my title is As Related Diagnosis and Magic Breaks. So this is essentially approach targeting these four. So as we know about diabetes, it is characterized by increased blood glucose level. This is the idea of data. As need for study suggests that all of the marketed anti-diabetic drugs have side effects like hypoglycemia, secondary failure, BP disorders, and etc. So our aim was, it is just a pilot study. So our aim was first we designed the molecule. So in the designing of a molecule, at the one end we kept it as a lipophilic tail, and we linked it with BCAA, that is branch chain amino acids like leucine, valine, isoleucine. Then at the other end, we had to keep a thiazolidine dione nucleus because also we were targeting the PPR gamma genes. Then further, once the design, the core was designed, then what we did, we generated a molecular library of lakhs of compounds by R-group enumeration by using Schrodinger's. Then the whole library was screened by high-throughput virtual screening and further filtered by ADMET properties like violation of Lipin schedule. Then further we docked the molecule and PPR gamma target. Then after docking it, we got top hits which were showing better affinity compared to standard marketed pioglitazone, rosiglitazone, balaglitazone. Then further, those were characterized and anti-diabetic evaluation is still to be carried out. So in the first step, we treated thiourea with fluoroacetic acid in presence of HCl. We got a thiazolidine dione. This thiazolidine dione was further condensed with appropriate aryl aldehydes and heteroaryl aldehydes by means of Novengel's condensation in presence of piperidine acetate and toluene. Further, that the Novengel condensation product was then alkylated by using potassium iodide and potassium carbonate in dried EMF. Further, the synthesized compounds were then characterized by IR and the functional groups were at appropriate ranges. The proton and C13 NMR also were evident with the peaks, and further mass LCMS was carried out for the same compounds. Then we also the top hits we wanted to find out the probable mechanism of action we wanted to decode exact mechanism of action because of the hybridization of this molecules. So we constructed a network, and we found out that there were three genes which were highly being modulated. That is SLC2A4, which is mainly encoding the GLUT4 glucose cotransporter. Then INSR, which is responsible for insulin receptor protein, and then further the NR1C3, which as usual we know that it is a PPR gamma alkylation. Then further these three proteins were modulating the pathways like PI3 AKT pathway, which is a downstream signaling cascade of any anti-diabetic moiety. Further, P53 signaling pathway, as which is seen in PTQ1B. This was P53 signaling pathway was not expected for us, but we got it. Then insulin signaling pathway, which was expected, we got it by network pharmacology approach. So the conclusion, then we carried out the glucose uptake by means of in vitro yeast cells. Here, as such, we didn't get any better result for our synthesized compound compared to pioglitazone. So the results were negative. But again, still we have to carry out the in vitro in vivo analysis on glucose uptake on cell lines like skeletal muscles, adipose tissue, and further enzyme assays like PPR gamma estimation, PTP protein tyrosine phosphatase 1B inhibition enzymes should be carried out.
When you take an article studies, why you went for GCMS?
Not sir, it is LCMS.
Sorry,
LCMS. We wanted to find out the molecular whether our compounds have been found or not. I have just shown only one compound, but we have synthesized 8 compounds. But none of them were like active compared to standard drugs.
All compound is having less.
Activity is less. Yes.
What about energy? When for docking?
Yes, sir, we have done docking. In docking, at the time of we have not docked on GLUT receptor because that was accidentally we got through network pharmacology. But whatever the top 8 hits out of the million libraries we had screened, sir, those top 8s were greater than whatever marketed glitazones like pioglitazone, docetaxel, palaglitazone, we had added it into the library at the time of docking. And this was done on the PPR gamma library.
Good evening, everyone. Myself is Kamlesh Chaudhary. I'm from Department of Pharmaceutical Chemistry, JSS College of Pharmacy. As a topic of my project, design, synthesis, in silico study, as well as the biological evaluation of the AKT inhibitors as a potent anti-proliferative agent. As an introduction towards the topic, the PI3K AKT signaling pathway, which plays an important role in case of the breast cancer, which is because of the cell proliferation, cell growth, and survival and metabolism, which is into the AKT, which is known as serine tyrosine kinase receptor. As in my project, I'm targeting towards the serine tyrosine kinase receptor. And in this case, the HER, which is amplified triple negative, and the hormone receptor, which shows the positive, and degranulation of the system with the pigment, and targeting the AKT Therefore, the desirable therapeutic options for this method for this various cancer, which has the subtypes of the particularly shows that the resistance towards the standard therapies of the AKT inhibitors. Whereas the two competitive drugs for this, that is capivasertib and hyperlucertib, have undergone the testing into the phase 1 clinical trials and either alone into the combination and chemotherapy as well as the hormonal change or hormonal treatments. Similarly, in my studies, what I am doing as a series of the anti-cancer activity, that is the 6,7 dimethoxy 3,4 tetrahydroxy isoquinoline derivative, which I am preparing, which I am synthesizing those compounds, and with this in silico approach as well as the synthetic approach, I am further going for my studies, that is the biological evaluation study. And the characterization of the synthesized compound is done by the analytical instruments which are used. As the aim of the topic is to develop, synthesize, and evaluate the new AKT inhibitors, that is tetrahydroisoquinoline, as an effective anti-cancer agent. And the objectives, which are molecular docking studies, second thing, the synthesize, based on the molecular docking, we are synthesizing the compounds, and similarly, the purification and characterization of the compounds. And next towards the biological evaluation. The materials and methods, in this case, the instruments which are used for this my project, which is FTIR, NMR, mass, and micro pH meter and UV chamber, as well as the apparatus and the chemicals and reagents. In this case, the reagents which are used, that is the synthesis for the synthesizer, that is 6,7 dimethoxy 3,4 tetrahydroisoquinoline sulfonamide derivatives, and which were of the laboratory grade. And similarly, I am using for FTIR, NMR, mass, and LCM. This is the methodological approach of my study. That is, first I am going for the target identification of the compound in silico. Based on the review literature, in silico study, drug synthesis, analytical characterization, and next towards the biological evaluation, which is yet to be done because this is my ongoing M.Pharm project, where I am in the M.Pharm second year. These are the results and discussion towards the docking study. Whereas the protein structure, which is used as 5L2S, and the receptor ligand interaction. Based on the protein structure, I have did the docking for the co-crystal ligand as well as for the ligands which I am using, the aldehydes which I am using. Based on that, the binding energy for the co-crystal ligand is shown as around the -8. And in case of the ligands which I am using, means docking done with the co-crystal ligand as well as the ligands which I am synthesizing. The binding energy for the ligands is shown -135. These are the one compound which I am showing the spectral analysis of the compound. And in this case summary, the synthesized compounds are showing the potent I am going with the further studies with the biological evaluation, which shows the potent activity for the studies. I am planning for the anti-cancer activity of my compounds. As it shows the potent activity, it can be the drug candidate and it can be used for our useful in case of the anti-cancer drugs or research. As I'm thankful for my guide, that is Dr. Ravish Kumar sir and Principal D.M. Pramod Kumar, as well as Vice Principal Dr. Pujar sir, and all the JSS family. Thank you. These are my references. Thank you.
Presentations.
Sir, that is a cyclic means staining compound. There is cyclic PI3K inhibitor. There is cyclase means AK staining compound. AK staining. Sir, AK staining function of that AKT. Sorry, sir. Function of that AKT. Function of AKT. function of AKT.
What is it?
AKT is a tyrosine kinase receptor. Yeah, I told you, sir. TI3K.
Breast cancer, there are three things based on this, like beta and mTOR and there is any data.
There is the data, but I have shown only one project work it is directly published. We are planning to
Already existed data is available for sulfonides and isopropyl against the cancer. Because synthesis is compound is related to sulfonamide and isopropyl.
Yes,
that is because the isopropyl is an antibiotic. And sulfonamide is antibacterial.
yes.
Then which basis to select this drug against the cancer?
We are planning for the anti-cancer. For the anti-cancer. What will we plan for in case of antibacterial?
When you are going for the plan of study, either you have to select the activity, then you have to search that. Otherwise, you have to search that, then you have to go for activity. In which basis to select?
First in Sir, in this case, I have not done up to the biological evaluation.
And what you should know the energy value. What you have now, you have to for that. How much it cost?
In case of it was -8.
Video camera, trying to start the video camera, please.
See, doctor, it has
I am having doctor. Because of
No.
till now, there is no such interaction.
So in case of sulfonamides, what the
Leave it, leave it.
Standard you are. Take any standard.
Tomorrow it's on this one. Okay.
Then how you will come?
Not a cable. We are just reviewing the thing. Reviewing the power. So, projected only this one is matching.
But one thing, you just go with one relatively heavier literature. It makes this kind of answer.
Thank you.
You have not synthesized that? You synthesized that? Not synthesize, design only. Okay.
Babu. Very big.
Good evening, one and all. Myself, Nasila Khalil from the Department of Pharmaceutical Chemistry. Sri Adichunchanagiri College of Pharmacy, and I'm here to present the dissertation work of my senior Jayashree. And the title is Design, Synthesis, Characterization, and Pharmacological Evaluation of Indoor Dihydropyrimidine Derivatives. This is the introduction. There, the general methods for indoor have surveyed. They have surveyed the literature. They got to know about the general methods for the synthesis of indoor, like Fischer synthesis, etc., and the dihydropyrimidine synthesis, which includes a general method like condensation. And the synthesized compounds were characterized by spectral studies, which includes IR, proton NMR, mass spectral. These are the aims and objectives. Here, the docking study of the design molecules were examined through Schrödinger software, and the synthesis of indoor dihydropyrimidine and indoor thiopyrimidine derivatives were synthesized. And the conformation of the synthesized compounds was done by IR, NMR, as well as mass spectroscopy, and followed by analytic activity for the newly synthesized compounds performed by Eddy's hot plate method. Materials and methods. Here, this is the scheme where indole was combined with the 4-bromoacetophenol in potassium anhydrous potassium carbonate, which was refluxed for around 15 hours. And then the solvent was removed and the product was obtained. Further, the product A was again treated with the product A, which was obtained is 1,4 1H indole, 1I phenyl ethane, 1-ol. And the further compound A was again further synthesized by treating with the different aromatic aldehydes. Now, where the mixture was treated with the sodium hydroxide solution and was refluxed, followed by stirring for about 5 hours, and it was refrigerated overnight, as well as it was poured into the ice cold water, and they got the product B, that is 1,4-1H indole, 1I phenyl pro 2E 1-ol. And the product B was further synthesized with the help of urea as well as thiourea, where in the product B you can see the product obtained was a chalcone. That chalcone was treated with urea as well as thiourea, and they got the final products. And followed by docking studies. In docking studies, they have done for different aldehydes, where they got the highest negative value for the compounds like J7, J9, as well as J14. Among that, for those synthesized compounds, the spectroscopy was done, as well as the analysis activity was done for the compound J14, which showed the potent activity. Here in the compound J7, mass spectrum as well as the NMR spectroscopy was examined, where the molecular weight of the compound J7 was 400, and the fragmentations like 390 fragmentations at 399, 363, etc., as well as an NMR spectroscopy. Proton NMR was done, where the prominent peak was seen at 9.8, and it showed that the presence of NH as well as A6 to 8.6, which showed the presence of aromatic ring, as well as CH2 at 1.23 and CH1CH at 5.06 to 5.02. Followed by J9, the molecular weight of the structure was 455, and the various fragmentations was observed, as well as in NMR spectroscopy, the peaks was observed, which showed the presence of aromatic ring, CH, CH2, CH double bond CH, as well as OCH3. Here in J9, the aldehyde used was chlorobenzene, chlorobenzaldehyde. As well as in the compound J9, the derivative was trimethoxyphenyl derivative, which was attached to the compound B. These are the like in NMR, the compound was confirmed by the NMR as well as mass spectroscopy. For the J protein, the molecular weight of the compound was 396, and various fragmentations were observed at specific ranges, as well as NMR spectroscopy showed the presence of methoxy group, CH2 group, aromatic ring, as well as NH. Now here the reagent used was 4-methoxyphenyl ring was attached to the compound B. And among all the synthesized compounds, the activity was done only for a J protein compound because the negative higher score was observed for J protein. So for the analysis activity, they have divided the compounds. They have taken the 5 group of animals, like albino rats, and they have observed the analysis activity by hot plate method, where group 1 was controlled, and group 2 they have introduced a standard drug indomethacin, and group 3, group 4 as well as group 5. The activity was observed for 3 set of groups for 100 mg per kg, 200 mg per kg as well as 400 mg per kg, among which the group 5, that is indole dihydropyrimidine derivatives at 400 mg per kg showed the potent activity, which is matching with the standard, that is 9.9 plus or minus 0.34, and the group 5 animals showed the activity at 7.2 plus or minus 0.44 at the retention time 120 minutes. Summary and conclusion: the docking study of the designed molecules was carried out by Schrödinger software, and the compounds were confirmed by IR, mass, NMR, as I said. And among the 3 doses, 400 mg per kg showed the maximum activity at 120 minutes retention time, which is slightly lower than the indomethacin, and thus it shows the analysis.
Can you say any other derivatives based on the literature which have been already there with other than indole dihydropyrimidine derivative?
Target was for indole compounds, but the derivatives they used were different. Like as nitroaldehydes, they have taken and different different single methoxy groups they have taken. For that, they have done the docking studies where they have seen the highest negative score 3 or 4 compounds, among which they have selected for the activity as well as the spectroscopy.
What is the significant
the target was to see the analytic activity, sir. So they have taken a standard and they have went for docking studies, which have showed the good activity, good docking score. So they have proceeded with the analysis activity.
Those that showed better analytic activity. Optimal dose showed the we want to convert this into a human equivalent dose when we pass. This is the molecule. So you're going to prepare a tablet in grams. Yes. See, you are whatever you are saying here, the optimum dose is 400 mg per kg per day. If you are going to convert that to a human equivalent dose, it will come more than 10 grams per day or something like that. So do you think for a pain relief you are going to take 10 grams tablet? Thank you.
have chosen the doses 100 and 200 and 400. Fine, but choose. And also regarding the purity, any challenges you face for the purification? Your product is pure one, so I will study that. Yes, also. Oh, how will you get the analytical reports without the purity? Have you checked the purity?
Very good evening to everyone. I am Kirtana from Sri Adichunchanagiri College of Pharmacy. Here I am going to present on the synthesis and antimicrobial study of industry 3-carboxylic acid derivatives. I am presenting this research work of our senior researcher. Coming to the introduction, indazole is a heterocyclic compound which is a molecular formula of C7H6. Indazole is a heterocyclic. Indazole contains nitrogen atom functionalized with highly selectivity at different positions. For the polarity of the indazole ring, side chain length, and functionalization at different positions. The most frequently used method is the use of ortho-disubstituted benzene derivatives, where two substituted atoms reacts to form pyrazole ring. For example, diazotization and intermolecular cyclization of orthomethyl adenine derivatives. Next, need for the study. The study describes the synthesis and antimicrobial activity of the indazole, the novel series of a compound indazole 3-carboxylic acid. The synthesis of potent compound based on the docking scores obtained by the docking studies. Screening for the synthesis compounds for antimicrobial studies. Next, material and method. In step 1, synthesis of 1H indazole 3-carbohydride. In this 1H indazole carboxylic acid in the presence of sulfuric acid and ethanol to form 1H indazole 3-carbohydrate. In the second step, synthesis of benzene amino acid derivatives from benzyl chloride. In this, benzene chloride reacts with amino acids to form benzyl amino acid derivatives. In third step, both the product of step 1 and step 2 reacts to form final derivatives. In that final derivatives R represents amino acids. Next, results and discussion. The chemicals and reagents used for this product were analytical and synthetic grade purchased from the Loba chemicals. In this, azithromycin were used as a standard reference drug. Antibacterial activity on E. coli shows minimum inhibitory concentration on the compound 3A is 31.25 and antibacterial activity on Staphylococcus aureus is and the compound 3A is 15.6. Both the 3A and 3B compound shows the good antibacterial activity against the antibacterial activity. Next, summary and conclusion. Indazole 3-carboxylic acid and derivatives exhibit various antibacterial activities like anti-hypertensive, anti-thrombin, anti-neoplastic, and antibacterial activity. In this way, and mainly consider antibacterial activity of novel indazole 3-carboxylic acid derivatives. After the spectral studies of novel compounds have been screened for the antibacterial activity by cross-validation method. Both 3 and 3B compounds show the good antibacterial activity against
You have synthesized indazole 3-carboxylase, right? And is it possible to synthesize to go for indazole 4-carboxylic acid? Whether you compare? That's what any literature shows indazole 4-carboxylic acid. Why not we can go for any other substitution? Any other question? Why you are particularly focused for the third position to substitute carboxylic acid? Is there any specific reason? I guess third position. So,
antibacterial, right? Whether it is for gram positive or gram negative.
the models to see. Methods. No. Methods. Activity. Antimicrobial activity mentioned, no? So that one. The methods. First method.
Next.
Good evening, myself Madhushri from Department of Chemistry, second M.Pharm, MS Ramaya University of Applied Sciences. My title is Design of Novel Aminopyrimidine as a GABA aminotransferase inhibitor to treat conversion. It is a computational approach. Epilepsy, this convolution, it is a common disease characterized by chronic and recurrent seizure due to it is caused by sudden electrical discharge whenever the GABA, like gamma aminobutyric acid level decreases due to the enzyme gamma aminobutyric acid aminotransferase. It will metabolize the enzyme and GABA level decreases. It leads to convulsion. Need for the study. The currently available drugs like cycloserine, valproic acid, vigabatrin, which are used to treat the convulsion, which is associated with so many side effects like depression, sedation, ataxia, etc. So we decided to adapt the CAD technique of molecular docking technique to develop a new compound, aminopyrimidine derivatives, for effective treatment strategy for epilepsy. Materials and method. In materials and method, docking. Step 1, preparation of protein. In preparation of protein, the protein used is GABA amino, which is having a PDB ID of
2COI. This is the structure of the protein. And in step 2, the preparation of ligand. In ChemPro, I sketched these molecules, that is amino pyrimidines. And energy minimization is done using the tool Pyrex. Then step 3, grid generation. Using Autodoc Vina, the grid generation takes place. The grid, whatever I have generated, that is of dimension 56.715 angstroms. Then fourth step is docking. Here, protein is docked against the ligands. Yeah, I have taken the 26 ligands like chlorine substitution, bromine substitution, so many like aldehyde, aromatic aldehyde based on the availability of chemical. Then result and discussion. In result, among 26 ligands, JJMK 3, 6, 10 and 20 showed more binding affinity than the standard drug ligabactin and valproic acid. It shows interaction like interaction of pi cation interaction, alkyl interaction and conventional hydrogen bonding with the ligands. This is the 2D and 3D structure of the ligands with GABA aminotransferase. Summary and conclusion. In silico molecular docking approaches is useful and it could be potential to inhibit the enzyme GABA aminotransferase. And this is acknowledgement. So we are like I have designed and I have checked the
basic molecule. Actually, it is anti-nitrogen.
Yes.
And second thing, it is not polar. If you are using for any purpose or anti-nitrogen, it has to cross. And it is used to cross.
This is I have just designed the molecule, sir. So I did docking. I don't I am not I am not with IIAM. A computational approach is an in silico method of in computer we are going to design or will get the results of what we are aiming to know. The molecule is it is exhibiting any activity on that protein or yes, like one study, like single study I did, that is docking. So I'm planning to do synthesis also. Seven one five and the lens is
'Okay. So your molecule looks to be a very thin molecule. How many rotational bonds are there? Let's see. You have to draw from the axis. So you have to see where it is going to be flexible and where it is going to change the conformation. Where is the rotational bond in the molecule?
I think conventional hydrogen bond.
Where is the hydrogen bond?
The green color.
Green color only is hydrogen.
Yes, sir.
And why you are want to do darker? You can just move that. Why do you want to use the hard order?
Because this is the before doing the synthesis, if we do this one, we get an idea.
Then that's what. Get an idea, just get the molecule and put it. Why you want to use a software?
Without software, we can't do.
Yeah, you can move there. That means what? You can just put the molecule in the like this. So you have to find the rotational bonds where it is going to rotate and where it is going to fit into your that program. But where is your rotational bond?
It will metabolize GABA. Based on.
How you going to get that?
If GABA level decreases, it will lead to neuronal excitation. Excitation.
Sure.
Yes, sir.
GABA is the excited neuron or the
Excited. And this is the Department of Pharmacology, MS University of Applied Sciences. The topic is molecular docking assessment of anti-inflammatory potential of some phytoconstituents against ulcerative colitis. IPD is inflammatory bowel disease. It is the inflammation of the inner lining of the intestine. It is made up of two diseases, collectively Crohn's disease and ulcerative colitis. Crohn's disease is when the inflammation takes part in any part of the GIT, that is Crohn's disease. But ulcerative colitis is restricted to the mucosa and the submucosal region of the intestine, and in the anus and the inflammation takes place, then that is ulcerative colitis. When if it is left untreated, both of them can lead to colorectal cancer. So the aim of the study was to select few phytoconstituents from which is available in natural sources, and based on their antioxidant, anti-ulcer and wound capabilities, and to formulate into a suitable polyable formulation so that will be able to can act against IBD. Need for study. Some of the standard drugs which are used to treat ulcerative colitis, like tacrolimus or sulfasalazine, all of them have severe side effects. Like tacrolimus, if you use it for a longer period of time, it can cause diabetes, as it is reported in some papers, and it can cause disturbance in intestinal flora, and also the class of drugs used to treat ulcerative colitis, that is TNF alpha inhibitors, are not capable of treating all the symptoms of ulcerative colitis. There are chances of reoccurrence of symptoms, and also the medication therapy or any surgery is recommended is very expensive. The medication bracket is very expensive for ulcerative colitis, and according to survey, the people who are suffering, the number of people who suffer from ulcerative colitis is also increasing in the Southeast Asia. So the major goal was to develop low toxic any medication which is low toxic, which could benefit the mankind. Materials and method. First one was selection of ligands. The ligands which I selected for polyamide formulation were 4. One is fulvic acid, eugenol, naringenin, and linalool. All of them, but the structures are downloaded from Pumpkin, and the target TNF alpha was selected, and with PDB ID 2AZ5, it was downloaded from protein data bank, and the protein was optimized and purified during Discovery Studio and Swiss ADB, and then docking was done. After docking, it was compared with the docking score of standard cephalosporin, and which were better. And further, these herbs were evaluated for their admit properties using PKCSM web server. And coming to results, all the scores which are exhibited by these compounds when compared to standard, it was better, and admit properties also revealed that they fulfill the Lipinski's rule of 5, and the major property is intercellular absorption, and all of them showed good intercellular absorption with range between 50 to 90%, and none of them were CYP-PA4 inhibitors, and renal clearance was also good values. So, summary and conclusion. Since these any formulation which could be made with this, since this is still an ongoing project, any formulation which polyamide formulation like rectal suppository or gel which could be made with these herbs, it can perceive as harmless, and but no more safety studies are performed on this, which is still which we have to instill knowledge. Thank you. They are established for the antioxidant and anti-inflammatory, but none of them are over 90%. Inflammatory bowel disease, no activity is performed on any of these. This is just a prediction, sir. This is still an ongoing project. I have taken up, I'm not second year, sir. I've taken up this project. First objective was dropping. I'm going to make a polyamide form, rectal gel cell, and then perform in vivo studies. What is it? Animal model, sir. Animal model. TNT, trinitropentane sulfonic acid induced IBD. On that, or acetic acid or any DSS induced animal model, I'm going to, once I formulate the rectal gel, I'm going to test on them by groupings it, and then
any common
which are in the market, endomethacin. It is proven, sir. Ashwagandha is also proven. Turmeric, everything.
Okay, my presentation is although latest idea I have to present in front of you guys. Very good evening to one and all. Myself Sidesh, I am pursuing M.Com Pharmacy Practice Department. Today I am going to present my topic on regulatory consideration for xenotransplantation product in United States pre-marketing review. Yes. We have come across drugs and vaccines which are very essential for survival and maintenance of life. And another approach we have introduced is xenotransplantation or a transplantation. Transplantation is nothing but a surgical procedure where a cell or organ or tissue is transplanted from one region of the body to other region or from one individual to the other individual. Xenotransplantation is type of a transplantation where other types includes ABO domain transplantation, autograft, allotransplantation. We have particularly concentrated our topic on xenotransplantation. We have taken a regulatory consideration of FDA, that is the Food Drug Administration and regulatory body of United States. Our topic is xenotransplantation. Xenotransplantation is defined as a procedure which involves the transplantation of cells or tissues or organs from an individual of different species. Types of xenotransplantation include solid organ xenotransplantation, cellular tissue transplantation. In solid organ transplantation, whole organ is transplanted from different individual, like pig to a human, or we focused on the genetically modified pig because of the various reasons I'm going to mention in further studies. Cellular and tissue transplantation, where we only extract the tissues and cells from a genetically modified organisms, and we have transplanting recipient. Aim of our project is we are going to we are going to concentrate on the regulatory aspects of xenotransplantation products in United States. The objective of our study is we are going to study various terminologies which are under xenotransplantation. We are going to select the animal donors on what criteria, and we have also come across various preclinical and clinical data that are necessary for conducting xenotransplantation. And finally, we end our study based on the challenges and barriers that are occurring in conducting successful xenotransplantation. These are aims and objectives. Coming to the material methodology, in our regulatory project, our methodology and methods include the various guidelines that is put forward by FDA. Various guidelines are mentioned in CFR, that is Code of Federal Regulation. There are nearly 0 to 1,500 various parts of guidelines under CFR. For the xenotransplantation thing, FDA has opted under the organization of CBR, that is Center for Biological Evaluation and Research. Center for Biological Research and Evaluation provided the guidance on how to conduct xenotransplantation. So, for considering the guidelines for xenotransplantation, it includes two major aspects, that is characterization of source animals and characterization of xenotransplantation products. Coming to the characterization of source animals are nothing but the animals that are designed for the xenotransplantation, where we are obtaining the live cells or tissues or organs for the recipient of humans. We have mainly concentrated on genetically modified pig. Pigs are why we have selected pig as being the sense the FDA or any other governing body, they have mainly opted genetically modified beef because of the organ size and the very basic thing is that is the organs which are in pig, genetically modified pig, are also accepted in human body at the at least minimum percentage. So they have opted and also considering the ethical consideration, we may opt as apes or chimpanzees, but because of the intolerance and because of rearing and breeding, all those things are very advantageous in the pigs or the genetically modified pigs. For characterization, we have mainly focused on safety, purity, identity, and potency. In safety things, we check for the bacteria, fungal, and also virus test and endogenous and endotoxin test. Coming to the preclinical study, when the drug or a vaccine should market, it has to go through various clinical trials. Same way here, xenotransplantation products, when we want to get successful, people have to, or the FDA or scientists who are conducting xenotransplantation have to go to preclinical study. In preclinical study, we are going to test for the animal source xenotransplanted product, the autonomic and physiological the similarities to human analogs and determination of function of xenotransplantation products and animal model system integrity of device component. Device component in the sense when we combine the tissue or animals with the medical devices. Then xenotransplantation product host interaction. When a successful xenotransplantation means there should be no interaction between the transplanted organ and the recipient. But here in preclinical study, we are going to assess the xenotransplant product and host interactions. These are the IND application form and IND application form. These are mentioned in 21 CFR part 601. The sponsor have to look for the IND application and IND application form. He need to fill those form and he has to present to the FDA or the governing body. Clinical consideration. Clinical consideration here, we check for the clinical protocol review. The sponsor is filing the protocol that will be assessed by FDA governing body and xenotransplantation site where xenotransplantation should be taken place and criteria for patients, that's inclusion and exclusion criteria, like which patients have to be in and which patients have to be out. Then risk and benefit analysis. The risk and benefit includes whether the transplantation is benefit for the recipient or it is harm to the patient. Then screening for infectious agent like virus, bacteria, fungal microorganisms, etc. The result and discussion. In result and discussion, we have come across various barriers that medical safety and medical and safety issue, informed consent, issues in regulation in xenotransplantation, animal welfare. In animal, I conclude that saying that both vaccine and drugs are followed from like many years, but this is a new approach where we are for end organ failure, we are going to conduct the xenotransplantation. Thank you.
So, go Mike.
Any doubts?
So, do you have any regulations in India for this transplantation? Any guidelines are available for this?
Sir, actually, many countries are focused on
India.
1 minute, sir. Many countries they are following FDA for this project. In India, in 2020, the 2 doctors are arrested for transplantation of pig heart. So there is no certain guidelines that CDSO or DCGI they have put for xenotransplantation. This is the only FDA government they have come across various studies for the xenotransplantation.
They are approved for the studies only, no, but they are not legally marketed.
No, sir, it is not because the rate of successful rate is very less. So there is no like they did not done any guidelines for doing this.
It is not legalized.
No,
it is just a study.
Just a study, and trials are being conducted for to see the
That's what actually.
successful rate.
Studies are on.
Yes, sir. Sir, we have WHO census now, sir, like 300 deaths per day and 2 lakh deaths per year is due to a end organ failure. Because of that, so like suddenly liver cirrhosis or a heart failure, some like or then cancer, or sometimes there is we can't do anything with drugs and vaccine. The best idea is this, and for successful rates, sir, like we have selected genetically modified pig. Cornea obtained from pig, it survived for 2 years, and lungs and kidneys only they have been survived with 10 days. So because of that thing, we have concentrated, but intensive study on this, like immune suppression. If you do that, we may like get successful at least just for 5 years, or mortality may decrease. Animals, sir, they have majorly concentrated on pigs, sir. Um, Genetically modified pig. You may ask why they have not been for apes or chimpanzees, which they are really specially they are related to us. Because of the rearing thing, because of the intolerance, because of the controlling thing, which is very difficult in case of apes and chimpanzees and baboons. So they have selected genetically modified pig where they can easily control, and their rearing offspring will be very high. So, and even animal ethical committee have agreed for the genetically modified pig.
One last basic question.
Yes, sir.
Do you think this one will be successful?
Personally, sir, like if you like generally we can say if a person is dying, like at least the relatives or thing, like if you are coming to a big organization where we have to say that person at least for 2 years or 1 year, like we may go with this process. At least we can say for 1 year. That is very good. Like we can't do with drugs or vaccine, sir. At any moment.
Basically, why I'm asking you is that, so if you take one cell from the same person and if you transplant it to other one, or in the other organ itself, so it is actually showing allograft rejection.
Yes.
Rejection. Okay.
Then
how do you think actually the other animal will be transplanted successfully?
Other animal dishes are, as I mentioned, genetically modified pig. They have come up with CHCP gene, where introduction to that gene into a genetically modified pig resulted in a very less infection. Like commonly when we take any organ from pig or any animal and transfer it into human, there is a chance of hyperacute rejection, or there is a chance of endoporcine virus. So when these genes are extracted and transferred to the pig is resistant to all those viruses and things, and it is very matchable for the human too. So, and by giving immunosuppression agent, so we can at least save a person. No, sir. I need to go.
To the scaffold, partially they build the organ and they will transfer it to you. But in that case, it is not able.
Very it is not very economical, but still, and that is economical. Thank you.
today. Tomorrow is continuing. Okay. And your certificates will be distributed tomorrow. Okay. So you can understand after completion tomorrow session. Valedictory will be there. So during that valedictory session, you can be able to get your certificates. So that will performed and they can keep it up. Okay. They will improve. They will improve. It's like preliminary trials. Anyhow, so you tried. That is the best part you had done. Okay. So that again you can continue further improvements. Okay. All the best.